So this is our 13th year for the REU program, and it's grown from 8 during our first year up to now we have 25 students here that participated in the REU program. Let's see if I can. Did I just move forward? So the funding for the program comes primarily for the large, can you guys hear me? Comes largely from NSF, who supports the overall umbrella, and that supports 12 positions, and then we augment that with students that are supported through a whole bunch of other mechanisms. So the REU acts as an umbrella organization or an umbrella program for a whole bunch of different kinds of avenues for students to come in, and it's really has grown over the years. So we have funding that supports some of the Colby students. We have NASA funding, NOAA funding, ONR funding, Sea Grant funding. There's a whole bunch of different avenues that come in here. And thank you for those programs. The casing for this, so the casing for the sausage that is the REU program comes from a lot of organizational people in the background that you guys don't see every day, but they include coordinators. Roxana is priceless, and runs in the background, dealt with a lot with the financial parts of the back, any kind of food you'll ever see here, and this kind of organization. And really a round of applause there, because it is a huge, huge effort. Nicole Polton also participates in the program, thanks to IT, facilities, including Kim Knowlton, who also helps sets up all these things. There's professional work that happens. So I'm going to go through just a little bit about how this program works for the people who are at home or haven't seen this. We have a Cafe Sci, which really started in force last year, and that's kind of the programming arm of this for people that are either interested in programming with their data or just learning about how to deal with big data management. And so they put together a nice thing that they did every Friday. Ben Twining gave some on plagiarism and ethics in science. You guys remember that at the very beginning? Maura is just hands in all of this stuff, and Fritz takes pictures all over the place, and I'm sure he's hopefully doing that right now. And then again, the funders, which include some internal and external portions of this. So the program started out, we just came off of a pandemic, and this program started out last year with 16 students, all off-site. So this year we decided to start with 16, and they were going to be housed here. Then we had 5 that were commuters and 4 that were remote. And so these commuters came in every day and worked on the lab, but they lived off site. And we did that because we weren't sure what the pandemic was doing. At the end of the day, we got everybody here. So we had 25 people living on campus, all the REU students. And while not all of them are here, I used 2 photographs because some showed later, and there's still, I think, 2 missing. That's the cohort that you're looking at here. So here's where these students come from. So we had 25 students this time. 10 of them came from Maine, and the rest of them distributed around the Eastern Seaboard, 2 from California, and then we had some internal states. And I worked pretty hard to try to get more from internal states, but it's a pretty hard sell here. I'm going to superimpose this on all of our REUs over the years. So we've done 185 REUs have passed through this program. And again, you can see that we got a little problem up in the upper states here. Oops. In the upper states here. And we have an Alaska person that we had last year. And there are some down in these lower states in the Arkansas and Oklahoma area. But we're working on it. 185 students. We've supported over $1 million in stipend through this program for the students, about $300,000 to $350,000 in housing for these students. And clearly the money spent is not the measure of success in here. It's really the science that they've done and where they move on from that. So this is the program. Each of those lines underneath are funding the REU grants that we've gotten to support this. So we have to renew this every couple of years. And I think it's a little offset. But there's meetings, cruises, and publications. And as you can see, the meetings, we get about, on average, 10 people to a meeting every year. Some of them they are not listed on here, to smaller local meetings or virtual meetings that they've done. One year we had a great 17. Cruises, we average about 2 cruises per year, and these are long cruises that the students go on. We had 2 that went on this year, and they were 60-day cruises, and one was REU from last year or 2 years ago, and one was an REU Ben who was here this time. And publications, and we average about 2 to 3 publications per year. And so it's pretty amazing for an undergraduate program to be pushing things like that through. I'm not sure how I'm doing on time, but oops. Okay. So the program has a mixture of kind of these social events and also the programmatic events. And so the programmatic events, 80% of it is hopefully doing their science. That's what we try to focus on here. But we do take them on a research cruise. And so we go out into the Gulf, and we have a long-standing time series that we do, and they participate in that when they first got here. Some of them host tours. There's these professional developments, which include the Cafe Code, the Journal Club, the Science Communications, and then we give primers and some of the things that we've done. Excel primer this year, but we've also done stat primers and just kind of teaching some of the workings of that. There's also seminars. We did some diversity and inclusivity. Thank you, Beth. And we had some programs on applying to grad schools that we channeled in from other institutions that made those available to us. And then socially, we have these barbecues, these kayak trips, and then we did a staff cruise this year. And these are some of those pictures. They learned to hula hoop. Clearly, some of them got pretty good. We have outdoor showers there. And you can see Monsi and Tejas doing those barbecues. And then we took them on a kayak trip. And up above that was a ship cruise. It's really pretty amazing what happens in here. And I do this in part to remind you guys that over this 10 weeks, how much you got access to. These are some of the cruises that they participated on, and we give them a full CTD kind of experience. We sampled water, we sampled them for nutrients, we pulled them up and preserved them for bacteria and viruses. We did net tows for phytoplankton and zooplankton, the whole gamut. And then there was all the electronic data. And then at the end, the next day, we analyzed the chlorophylls, and some of you came up for that. And really, again, these were moments of opportunity. These are not required things. They were moments of opportunity. So what you are all here for today is to see the science. So all I did was grab their posters. I pulled off a little piece of it that I thought was emblematic as we went through the REUs and threw them on this slide. There are some really great science that you're going to see today. It covered things from oil spills all the way up to cows. All the way up to cows. We haven't had a cow on any posters in the REU program. There's some really beautiful kind of graphing stuff. There are these fiddle, is that what they're called? Or violin graphs that were in there, that things that I've never seen plotted like that. And some just some great explanations of both pathways, metabolic pathways, genetic from sequencing kind of information, and all the way up to organismal kind of studies. It's a sweet year that we've had this time. The program, it's going to be done into 4 blocks. This is a big, long day. So there's going to be 7 talks. We're going to take a 15-minute break, and then we're going to do them in blocks of 6. So there'll be a 6 talk. We'll go for lunch, another 6 talks. We're going to take a 10-minute, 15-minute break, and then we'll have the last set. It'll stay till 4:30, and I hope you guys enjoy the hell out of this because it's a great presentations that they have. And that is it. How much time? 3 minutes. So if the first speaker wants to come up, And if you want to load his talk, so you have to hold this here. And unlike what I was doing, that's who you're speaking to. Perfect. And you got it. And just watch for me if you're starting to open. All right.
Start now. Hi, I'm Ben Gustafson. I'm a rising senior at Colby College, majoring in environmental science and biology. And at Bigelow, I'm working with Dr. Barney Balch and Sunny Pinkham to look at diatom nutrient limitation in a Southern Ocean eddy. So water from the Southern Ocean, which is this small band here that's kind of circumnavigating around Antarctica, provides the nutrients for 75% of the ocean biological production north of 30 degrees south. For reference, that's this line right here. And so 75% of the productivity north of that is provided from this small band of water right here. These high nutrient waters are known as subantarctic mode water. So most of this water is subducted right now near the Southern Ocean and then is upwelled again near the equator. But a small portion of this also goes up in eddies and is pinched off from the current there. So eddies are a swirling water mass that pinch off from a current. This eddies specifically that we're looking at pinched off from the polar front. Eddies from the polar front often result in higher phytoplankton biomass, which is usually diatom dominated. So these eddies are about 1 kilometer wide and about 1,000 meters deep. And this one here is the one that we're referencing, Eddy A, from this cruise that we were looking at. And so you can see longitude here on the x-axis and latitude on the y-axis. And then there's these contours, which are all denoting height or altimetry. And so that's another way that we can locate this eddy. And then this color scale is looking at log chlorophyll concentrations. And so these deep reds moving into the yellows and greens are the higher concentrations. And then the blues on the other end of the spectrum are the lower end. And so we can see that this has a higher phytoplankton biomass right in the center of this eddy. And then it's a little bit lower, and then it gets deeper again on the outer edges. And so these are really important because we can look at the eddy water and experimentally evaluate it with nutrient addition experiments to understand what kind of nutrients are helping these phytoplankton grow. And so diatoms are often limited either by iron or by silicate. And so these are the two primary nutrients that we're looking at, as well as size structure changes. So what this means is that often in the phytoplankton community, when there's iron in the system, that means that larger diatoms are going to be growing. And then when there's a lack of iron, this means that the smaller phytoplankton are the ones that are going to be growing more. So to evaluate this, our research question was, what nutrients are limiting Southern Ocean diatoms? So we had two hypotheses. The first is that silicate and iron are the limiting nutrients for the diatom biomass. And then our second one is that in the low iron environments, there would be a greater biomass of the small size, which is 0 to 12 micron diatoms, than the larger size, which is greater than 12 micron diatoms. And so this data was collected from this 60-day cruise that I was able to participate on. We started up here in Honolulu, and we sailed south down to 60 degrees south. And then we went over to look at a couple eddies. So the eddies are right over here in this kind of corner. And then we went back up. And so the eddy water was collected and then incubated with different nutrient additions. So we used this system over here on the left, and this system helped us to collect the water, and it was a trace metal clean system. This means that there's not going to be any iron from the tubes that we're using to collect the water or from the ship that are going to be getting into the incubations. And then we incubated them in 20-liter bottles for 5 days, measuring every 48 hours. And then we ran them through the flow cam. So the flow cam is an imaging cytometer that takes very small pictures of these phytoplankton, which then we can use to look at and understand what's living in the water. So we had 6 different treatments. We had the control water, And then we had 2 natural water additions. So this winter mix depth, this is a cyclonic eddy, which means that water from the center of the eddy is going to be upwelling towards the surface. And so this 300 meters would eventually be reaching the surface. And so that's the winter mix depth that we're looking at. And so we did 5% of the winter mix step along with 95% of the control to get the nutrients from this depth, as well as one from the eddy edge. And so again, that's 5% of the water from the eddy edge with 95% of the control. And then we did 3 specific nutrient treatments. So the first was 1 nanomolar of iron, and then we had a silicate treatment because these are the 2 main nutrients that are limited by for diatoms. And then we had of iron and silicate together to see if both of those together would help promote diatom growth. So then next would be the data analysis. And so this was sorting the FlowCam images. So it's taking all these pictures and then they sort them into the phytoplankton groups. So things like dinoflagellates, diatoms, and ciliates along with size. And those sizes 0 to 12 microns, the smaller size, and 12 plus microns for the larger size. And then from these pictures, we can get the abundance and the carbon biomass using the Mendendorf and Lassard method, which is taking these algorithms to find the carbon biomass based on the plasma in the cells. And finally, we also can get the particle size distribution. So this is just the ratio of the large cells to the small cells to understand kind of what the sizes of particles and how they're distributed in the sample. And so here we can start looking at some results. This is the abundance growth rates. So on the x-axis are the different treatments. This is the growth rate from days 0 to day 3. And then this second graph is from days 3 to days 5. And so then on the y-axis, we have the diatom abundance plus or minus the standard error. And so we can see that from days 0 to 3, there isn't really any change. There is no significant difference in between any of the samples. They're pretty similar, nothing changing from the control. And then from days 3 to 5, we can see that there is some sort of jump in the iron. This is the one that is significantly different. And so this one changed from the control and had this jump in growth rate from days 3 to 5. Also in the carbon growth rates, we can see a really similar thing. So this is the carbon biomass growth rates, and it's a similar graph across the x-axis are the different treatments, days 0 to 3 and days 3 to 5. And then on the y-axis, we have the growth rate plus or minus the standard error, and that's on both of these. And so again, from days 0 to 3, no real changes from the control in any of the different treatments. And then from days 3 to 5, there is this major change in the iron treatment. Also interesting to note is that the iron plus silicate treatment together did not have the same results. These were not having this big jump that the iron treatment did. And this is the total carbon growth rates. And so this is for all of the cells put together. So not just diatoms, but everything that's growing in the water that we took a picture of. And so from this, we can see that there's pretty similar trends that from days 0 to 3, there was not really a significant change. And then from days 3 to 5, there's also this jump in the iron treatment. Finally, we're also looking at the particle size distribution function. So the way that we're looking at this is that we look at the PSDF slope. And so here on the x-axis, this is just an example. It has the log particle size on the x-axis, and on the y-axis is the log cell concentration. And so there's a bunch of really small cells, and then some of the moderate cells, and only one or maybe a few of the large cells. And then this would have kind of a negative slope that looks like this. But when there's larger cells, this means that a greater number of larger cells will result in a less negative slope. And so then if we're looking at this PSDF slope, here's the day 3 PSDF slopes and the day 5 PSDF slopes. And so day 3, there was no significant difference in between the treatments, and they're all pretty similar. Again, this is the slope of a line that would kind of look like this. And so when it gets closer to negative 2.8 and less negative, this means that there's a greater number of large particles. So in the day 5 treatment, we can see that there was a difference in the iron treatment again. And so this iron treatment had a similar result as the others, showing that there was a greater number of large particles, which potentially could be large diatoms. So in all, we found that the iron is promoting diatom growth as well as the growth of large particles in our samples. And then as we saw, there was kind of a lag time potentially post-nutrient spike. So those days 0 to 3, there wasn't really any change in any of the samples, but then from days 3 to 5, there was a change. So it's potential that just with incubations and everything, something was happening, and the phytoplankton are getting accustomed to the different system that they're in just the 20-liter bottle that caused kind of this interesting interaction. And then finally, there's a potential iron and silicate interaction effects that resulted in this less growth promotion. So the iron and silicate together did not have the same results as just the iron alone. And so it's potential that these two agents together either had a chemical reaction or all of the phytoplankton were promoting growth together and potentially rising up everything. And so there is less a growth rate of just the diatoms. But overall, it's not totally known what's going on in that sample. And that's kind of something to move forward with and take a look at as well. So I'd like to thank my mentor, Dr. Barney Balch, as well as the Balch Lab Group for all of their help and insight, Sonny Pinkham, Dave Drapeau, and Bruce Bowler. I'd also like to thank the RR 2004 Roger Revelle crews, especially the officers, the crew, and the science party, including the Team Trace Metals, Dr. Juliana Viglione, and Hannah Permiano, who all helped in conducting this experiment and running all of these samples. And then funding for this project was supported by the National Science Foundation, both for this REU program as well as the SAMW cruises. And I'd also like to thank the Linda Packman Lab for Biosciences for helping support me get onto that cruise. Thank you. I have time for a couple questions if anyone has them. Yeah, Molly.
Get a microphone.
So I apologize if I missed it, but what was the reasoning behind the 95 control and the 5% Eddy concentration that you used in the experiments?
That's the concentration just so that we're knowing that we're getting enough of the nutrients from the edge and from the deep concentrations so that we know that we're getting enough of the nutrients to support growth for the entire 20-liter bottle.
And 5% would have been enough?
Yeah.
Okay.
Nice work. Am I correct that this happened during the pandemic? You went on this cruise during the pandemic?
Yeah. So we flew to Honolulu and then had 14 days in a hotel room alone. And someone would knock on our door and then run with food. And we could not leave that room except for to go get our COVID tests, and then got on to this 60-day cruise.
That's a lot of work. Thanks.
Great job, Ben. I love seeing flow cam data, so as you know. So I have a quick question regarding the less than 12 micron fraction identification of diatoms. Was that pretty challenging? I assume you were using like a 10x objective. Is that what you were doing? And how did you have a challenge in identifying some of those smaller species? Or what was your cutoff for determining what was a diatom and what wasn't a diatom? Just curious.
Yeah, so our templates have the larger diatoms, and it's based on ABD. And then we're just, separating those. And so the smaller ones were more difficult to identify, definitely. So it was getting to a point where it was, only a very certain identification, like where you look at them and go, oh, that's a diatom. And then that's what you'd put into that category. Perfect. But definitely challenging.
Okay, thanks.
There's time for one more. Is there a hand?
Great job. Did you look specifically at the diatoms, like down to a species level? And did you compare like inside an eddy and outside of an eddy? Just curious to see if the population is differing.
Yeah, so this eddy edge, the idea was kind of looking at them, but not a ton of classifying on the species level just because of the quality of the flow cam imagery, just that we can't get quite that specific. But in general, it was more pennate diatoms than centric diatoms.
Great. Are there any questions in the chat? Nope.
No.
Thank you.
Thank you.
Hi, everyone. My name is Mike Stager. I'm a rising senior at Colby College, majoring in environmental science and biology. And this summer, I worked with Nicole Poulton and Laura Lebelchek, along with Steve Archer and Kate McPhee, to sort of evaluate this novel method for estimating phytoplankton growth and the grazing rates on them that we're calling the saturation method. So why do we care about phytoplankton growth and grazing rates? Well, we know that phytoplankton are one of the major drivers of global primary production. And in the ocean, the grazing on these phytoplankton can account for 12% of that carbon cycle in the ocean, or 5.5 billion tons of carbon per year. So in 1982, there was this really landmark paper by Landry and Hassett that set forth this standard method called the dilution method that's grown to near ubiquity in its use for the estimation of this growth and grazing rates. But as we'll see throughout this talk, it's not without its own issues. So we're trying to come up with this new method that can sort of augment this classic dilution method. So some really important things to keep in your head as we move through this talk of some of the underlying concepts of this is that we're assuming that what we're trying to do is decrease encounter rates between predator and prey. And we think by decreasing the encounter rate, we can decrease the predation pressure and therefore encourage growth. Because put very simply, grazing is equal to your gross growth, so your growth in the absence of any sort of grazing pressure, minus your net growth, so your normal community growth with all that grazing pressure. One of the key assumptions of this model is that as your grazer is moving around in the ocean, it sort of moves around randomly and it encounters a prey. So they're not actively looking for prey, but they're randomly encountering prey, and that's how they're predating on their food. So first, overview of what the dilution method does. So what we're trying to do in this method is physically separate the predator from the prey by adding space in between them. So what you do is you sample a whole bunch of water and then you filter a section of it through a really fine filter that removes all of the particles in that water. So you're able to remove all the phytoplankton and the grazers from that water. And then you add that filtered water back to the unfiltered water in varying concentrations in this attempt to continually reduce your encounter rate, therefore reducing your grazing pressure to zero. So some of the key assumptions here is that, again, planktonic grazing is directly associated with encounter rate, and that filtering and diluting water has no effect on phytoplankton growth, which, as we'll see, may not be entirely the case. However, with the saturation method, we're not filtering any water, so we're not changing any water chemistry. So what we're doing, instead of separating them physically, is we're putting lots of things in their way. So we're adding these surrogate prey items, which in our case were these 2-micron polystyrene beads that had been yellow-green fluorescently labeled so that we could tell what they are. And by adding these in increasing concentrations, we're putting lots of stuff in the grazer's way. So as the grazers are going around, they bump into these beads and they have to handle them. Maybe they eat them, maybe they spit them back out. It doesn't matter because what we're doing is we're stopping them from grazing on their natural prey. So some of the key assumptions here is that there's a point where you've added so many of these surrogate prey items that your grazing pressure goes to zero. So they can't find any of their natural prey because there's just so much of these surrogate prey items. And that adding all these beads and all the surrogate prey has no effect on growth or the light availability for the phytoplankton. So some of our the results that you'd expect to see from this, first looking on right-hand side in blue, these are your results from your, or I guess your left-hand side, excuse me, your dilution method. On your x-axis, you have increasing amount of unfiltered water. So your 0%, that's going to be all filtered water, and your 100%, that's going to be all unfiltered water. And on your y-axis is your growth rate. So what you expect is that your y-intercept, that's where you have all filtered water, excuse me, and that's where you would have no grazing, right? Because if there's no natural grazers in the population, that's theoretically where you should have your maximal growth rate. And then your 100% whole water, that's going to be your community net growth rate. And so by looking at that slope, you can get your grazing rate. Whereas on your right-hand side in orange, that's our expected results from our saturation experiments. So along your x-axis, you have increasing surrogate prey, and along your y-axis, you have either your growth or grazing rate, depending on which series we're looking at. So in blue, that's our growth rate. And as you see, as you increase that amount of surrogate prey abundance, you see increasing growth rate to a point, and then you've totally saturated the population. It begins to level out. And that's the point that we call sort of our gross growth. So if you draw a straight line back from that level off point, that's going to be your gross growth in the absence of grazing. And looking at this darker gray series, this is sort of your grazing pressure. So as you increase the amount of beads, you see a decreasing amount of grazing pressure. So our experimental setup, we ran 4 parallel experiments where we ran both a dilution and a saturation experiment over the summer, and we compared them. So in our dilution method, we have a series of bottles ranging from 10% whole water down to 20% or down to 10% whole water. And we expect to see increasing growth as we decrease the amount of whole water. Whereas in our saturation experiment, we have increasing bead concentration from no beads all the way up to as many as 10 million beads per milliliter, which is a huge amount. And we expect to see increasing growth as we increase the amount of beads. So just to visualize what 10 million beads per milliliter can look like, it's a little bit washed out on the screen here, but on the far left, that's our highest bead concentration, or on the far left is our 0 bead concentration. And as you move to your right, that's increasing amount of beads up to 10 million beads per milliliter. So once we have all these bottles set up, what we do is we take a sample at time 0, and then we incubate them under ambient water temperature and under this shade cloth that removes incoming solar radiation. So it's like they're being incubated at a 1 meter depth. And these are being constantly mixed by an intake of water that's trying to keep these beads suspended so they're not like settling out or clumping together. And then after 24 hours, we take another sample, and both of these samples are analyzed via flow cytometry so we can get really population specific numbers. So very briefly, what flow cytometry does is it channels all of your particles into a single file line and passes them by a laser. And then it can read both the fluorescence back from this laser and the side scatter of this laser. So what that lets you do is know what sort of pigments are in these particles and a general size idea. And from this data, you can get really fine enumerations of specific populations. So we're looking at here is a cytogram that shows side scatter, so basically a proxy for size on the x-axis, and then a fluorescence on the y-axis. And our red population here, that's going to be our Synechococcus, what we define as a Synechococcus population. Our lighter green, that's what we're going to be focusing on for the rest of this talk, sort of our pico-eukaryote population. That darker green is going to be a more nano-eukaryote population. And then in yellow along the side, that's our bead concentrations. So we can tell very obviously what is sort of a phytoplankton, what's eukaryotic, and what is a bead. So briefly to summarize our results, first looking at our saturation method, we have 2 different models of thinking about this. First, if you come from maybe a chemistry background, you can think of it almost as a Michaelis-Menten relationship, where you have your prey, either surrogate or natural, as your substrate, and then your grazer is sort of your enzyme. And as you increase the amount of substrate, this reaction rate increases to a point, and then once you've added so much of that substrate there, it's completely saturated the enzyme, and that reaction can't happen any faster. And that's what we're showing with this blue curve. This blue dashed curve is sort of our Michaelis-Menten fit, where you have increasing surrogate prey concentration along the x-axis and your pico-eukaryote growth along the y-axis. And as you increase that surrogate prey, you see increasing growth to a point, and then it begins to level off. Whereas in the red fit, this is where we're saying we're going to force this growth to come to an asymptotic relationship. So we're going to say, we think that there's a point where we've added so many beads that growth no longer increases. So we're just going to mathematically force that to be. So these models yield very similar estimations for both growth and grazing, but they have different conceptual backgrounds. So we've decided to compare them both. So basically, the only things you really need to look at when you look at these graphs is first, our controls on the bottom, that's going to be our net growth rate. That's when we've added 0 beads. That's our normal community growth rate. And then where it starts to level off, and that's our gross growth rate. And just by comparing these two, we can get a grazing rate. Whereas our dilution method, we had a little bit more inconsistency in our results. So first, looking on the left-hand side, this was one of our experiments where we think we may have had some sort of issue with our filtrate. So one of the main critiques of the dilution method is that when you're filtering all this water, you can have some negative effects and you can have really inconsistent results. So what we have here is something where we think that potentially our filtrate or our filtered water was negatively affecting our growth. So at our most dilute, so on our 10% on our left-hand side of this axis, we had the least amount of growth. And as we increased the amount of whole water, we had more growth, which is the opposite of what we expected. And then looking on the right-hand side, we had a lot of inconsistency in our 10% dilution level. And again, this could be due to just those effects of a high amount of filtered water, or it could be that we have such small populations to begin with, that if you dilute 90% of them out, it can be really hard to accurately enumerate them. So we looked a little bit at comparing what would happen if you remove this 10% level. And as you can see, in a lot of, or at least in this case, these are data from the same experiment. We just have removed the 10% level. And what it does is it can be pulling that whole relationship down. So as I said, this y-intercept is going to be our growth rate, and then our slope is going to be our grazing rate. So by removing this 10% level, we get maybe a much more accurate grazing and growth estimate, and we get a much higher R-squared value. So now to compare our saturation to our dilution results, looking on the left-hand side, this is our growth rate estimates, and on the right-hand side, that's our grazing estimates. In red and blue, these are our saturation models, and then in green and purple, those are our dilution models. And each dot here represents one of our 4 experimental estimates. And the key takeaway from this is that our 2 saturation models have much more tightly packed estimations. So it may be more consistent, and they're within the range of our dilution experimental estimates. So we think that they're likely just as accurate and more precise. So if you do any of the work with these dilution models or dilution methods, you may have heard of a 2-point dilution method. And that's this idea that you can streamline this experiment by basically removing a lot of the points and saying, we don't need all these extra points. All we need is a really dilute and a whole water. And from that, you can draw a line between those 2 points. And there's been some work done to show that you can get pretty accurate results by doing this. So we think we can do a pretty similar thing with our saturation method, because as I've said, all we really need is our controls at the bottom and then our fully saturated point at the top. And by comparing those two, we can get a growth rate. So hopefully all we would need would be our control bottles and a fully saturated set of bottles to do a 2-point saturation method that could decrease on experimental setup time even more. So some of the key takeaways from this comparison, some of the potential pros of this saturation model are that they offer more consistent results, as we've seen. And since we're not filtering any water, we're not changing the water chemistry in our experiment, it allows us to do chemical analyses of how grazing can affect the chemical constituents in the water. Also, it has a much easier experimental setup. In the dilution method, you're filtering tons of water, you have to wait for that, and then you have to deal with trying to measure out correct amounts of filtrate to whole water. In the saturation experiment, you have your bead stock, and you just pump in 1 mil, 2 mil, 3 mils, and then you're ready to go. It's much simpler to set up, and it has, again, this potential for a really easy 2-point method. And one of the advantages of using flow cytometry is that you can get really population-specific growth and grazing rates. Some of the potential cons, as you might have guessed, we're using these 2-micron plastic beads, and we don't want to be creating more plastic bead waste in the ocean. So we did a lot of work to try and scrub that from our wastewater before we dumped it. But there's some work going on as well. Right now, this is more of a proof of concept. Does this method work? But there's some work going on to say, like, maybe we can replace these plastic beads with silica beads or with some sort of biologic, like a labeled E. coli. But that's sort of the next steps in this process. As you get really, really saturated, so as you add huge amounts of these beads, you can start to negatively affect growth. So there's a worry that adding large amounts of saturation may alter growth. And then one of the key points of this project was to ensure that we were reaching that saturation point where that grazing or the growth rate begins to level off. So one of the challenges of this experiment could be making sure that you're on the top of that curve and you're not either on the downslope and you've added way too much, or you haven't added enough and you're still starting to come up. And with that, I'd like to thank Nicole, Laura, Steve, and David, and Kate, all for their help throughout the summer, and along with the whole REU cohort, the NSF for funding this project, and the Lynn Packman Lab for Biosciences Innovation for funding this internship for me. Questions? Monsi?
Could you explain how filtering exactly affects the water chemistry or negatively impacts the growth?
So what you do is you pass this water through a 0.45 micron filter. And when you're doing that, you're forcing all sorts of cells through this really small filter. And you expect that probably some of those cells are going to rupture. Maybe you're going to impact like the microbial community, so you can have alterations on chemistry there. So like what we think may have been happening when we saw that negative or that positive slope in our dilution method, we think there may have been something in the water that was producing like toxic chemicals. And as you passed it through the filter, that ruptured and released that into the filtrate.
Nice job.
Thank you.
I may have missed it in the beginning. How did you calculate growth rate?
So we had our time zero population numbers. So with flow cytometry, we can get exact counts per milliliter. And then we had our time 24, and we just compared those two and used like a factor because we have the times when they were run. So we can just divide that by the difference in time.
So it was based on counting your prey items. You looked at, how you had so many of them at the beginning and more.
Yes.
Assuming at the end. Okay.
Yes.
Just didn't know if you're measuring chlorophyll or anything.
Sorry. Yeah. Maybe
Yep. Great. Thanks.
Last one.
That was really interesting. Thank you. Can you offer any insights on microplastic pollution based on these experiments where you're adding microplastic pollution to the water in a predatory grazer prey system?
How do you mean? Like, do you think it's affecting grazing rates naturally, or
well, yeah.
how do we try
So when one adds, one of the arguments about microplastics in the sea are that they are getting in the way within the trophic system of predation, and essentially what you're offering there is the case where you're adding plastic items, non-natural plastic items that are affecting the predation on prey.
Right. I think it's probably unlikely that we get to these sorts of, or hopefully unlikely that we'd get to these sorts of concentrations where you start to see these effects on growth. But it's hard to say because this is a very like controlled experiment that are within bottles, and they're like identically sized 2 micron beads. So they're the perfect like prey size. So it's hard to say if natural plastics would have the same sort of effect.
Good morning. So I'm Brianna Mays. I'm an upcoming senior at Maine Maritime Academy. And this summer, I've been working with Dr. Leanne Whitney, Dr. Mike Lomas, and Deborah Lomas, and we've been assessing cellular polyphosphate in phytoplankton growth on phosphonates. So I want to first start off by addressing phosphorus and how it's an important nutrient for all cellular activity and function, no matter the organism. It's in our RNA and our DNA, our ATP and ADP, and it makes up our phospholipid bilayer. For phytoplankton specifically, phosphorus is found in a storage body called polyphosphate. And the focus of my research has been around this polyphosphate body, which can make up 10% of cell mass, if you look here along in this figure. So in the ocean, phosphorus is found in many different forms. The more preferable form for phytoplankton is phosphate. If you look at the graph I have up here, where in the purple, phosphate concentrations are considered low, and in the red, phosphate concentrations are really high. In the subtropical North Atlantic, waters are considered oligotrophic, meaning nutrient limited. In these areas, phytoplankton do not have direct access to their preferred form of phosphate. And so because of this, there's another pool of phosphorus that kind of influences primary productivity. So looking at this graph here, where you have in the x-axis years and dissolved organic phosphorus on one y-axis and primary production on the other, you can see that this is what fuels primary productivity in these oligotrophic regions. When you look at the peaks, like say here in 2005 and DOP, subsequently you can see a peak in primary productivity and a drop in DOP. So that tells us that this is what phytoplankton are using to fuel the primary productivity in these regions. In the dissolved organic phosphorus pool, you have P esters and phosphonates. P esters make up 80% of the dissolved organic phosphorus pool, and it's found in things such as ATP, DNA and RNA, and it's described by having a carbon to oxygen to phosphorus bond. Now, prokaryotes and eukaryotic organisms can utilize this in any shape, way, or form. But when we go into phosphonates, which is where my research has been focused, it makes up 10% of the dissolved organic phosphorus pool. And you have two main ones, aminoethyl and methylphosphonate. And they are described by having a strong carbon to phosphorus bond, which you can kind of see here. Oh, this thing is really hard to use. So it was originally believed that only prokaryotic organisms can utilize phosphonates to supply themselves for their growth. But research that's been done in conjunction with mine and is very new and was done by us in these last couple of years, was able to show that there are eukaryotic organisms that can utilize phosphonates for their growth. So in the experiments that we did, phytoplankton were grown in batch cultures and three different treatments. You have the plus P treatment, where cells have access to all the phosphate that they need to grow. If you look at the figure I have here, where the y-axis is cells per milliliter and the x-axis is days, this is like regular stuff that we see, where you give phytoplankton everything they need, they grow hardy and healthily. You give them a small amount of phosphate, which is known, but it's not enough to supply every organism or cell in that culture. And that's where you get this low growth here at the bottom. This is really hard to use. And then we have a batch culture where phytoplankton are grown in methylphosphonates, where their growth was a lot better than those grown in the low phosphate treatment. So in those treatments, we measured cellular P and polyphosphate on those at different time points. So this research has been going on for a couple of years now, and it started with Emily McDermott back in 2018, where she had generated this working list of eukaryotic organisms that are able to grow in phosphonates. For my research, we are focused on Emiliana huxlei and Isochrysis galbana, and you can see them on the side here. Oh my goodness, yes. Okay. Yeah, so Emiliana and Isochrysis. Does that? Okay. Oh, perfect. Okay, so yes, Emiliana and... So Emily McDermott first grew Isochrysis galbana and the three treatments I had described before in batch. And you can see here that those grown in the plus P treatment grew nice and big like we would expect them to. But if you look here between the methylphosphonate treatment and the low phosphate treatment, cells grew not way better, but 12% better than those in the those in the low P treatment. And so a trend that we noticed in these same cells when we took the cellular P measurements is that with time, cellular P decreases, especially in the methylphosphonate treatment, which is where our interest kind of lies, because it's decreasing, but that those cells are growing a lot better than those grown in the -P treatment. So then Kat Mahoney in 2019 did the same experiment with Emiliano hexali, and you can see the same trend here. It actually looks a lot better, where cells in the +P grew nice and healthy, those in the -P grew low, and then those in the methylphosphonate still grew better than those in the -P. And so it's important that I note that those in the methylphosphonate group, they have access to what's considered a contaminating amount of phosphate. And so that low amount of phosphate in that MPN group is what was put in the -P group. And so that's kind of what drives my research. And again, with the cellular P, it's decreasing with time. And so my research that I did this summer was focused on that drop in cellular P and trying to figure out what is driving it. So back to this graph, you have intracellular P pool, and it's pretty dynamic as phosphorus is found in many different parts of the cell. But going back to polyphosphates and how it makes up a large portion of that intracellular P pool, that is my focus. And we're curious to see is if cells that are grown in the presence of phosphonates, are they pulling from that poly P pool instead of actually utilizing the phosphonates given to them, or are they utilizing phosphonates and growing? So that's where I come in. I extracted poly P from all those samples that Emily McDermott and Kat Mahoney had grew a couple of years ago, and a pretty tedious, very tedious process. The first part was cell lysis. So we had to split open our cells, put enzymes in to degrade any DNA and RNA so that they wouldn't interfere with our spectrophotometer readings, which was done with DAPI. And as any of the interns know, DAPI is light sensitive, so I had to work in the dark all summer. And so it's fun, but not that fun. But I feel like we got some good results. So going back to the growth curve done by Emily McDermott, you can see here where you have cells per milliliter on the y-axis, that on days 6 and relative to that +P taken on this day 6, and the cells that I extracted from day 11 and 14, relative poly P actually increased in relation to the +P treatment. And it's interesting because a poly P value above 0 means that there is more poly P per cell than the +P at that time point. Below 0 means less. And so you can see that the +P is literally not really there, but you can see that in the -P treatment versus the methylphosphonate treatment, that they have high rates of relative poly P. So you see the kind of the same trend in Emiliano hexali, where the +P the relative +P that we're kind of comparing this all to was taken on day 6, and those same measurements as well, where I extracted polyphosphate from methylphosphonate in the -P treatments on day 6 and 8, you can see the same trend, that there is an increase in that polyphosphate storage rather than a decrease, which we were kind of expecting. So this suggests that cells may be regulating their poly P storage in different P stress responses. So to kind of conclude on that, to piece together what we've been putting together for so many years, and I'm kind of just like this piece right here, and this is what they've been working on. When you compare -P and methylphosphonate, those in the -P treatment in terms of cell concentration did not grow as well as those in the methylphosphonate, which is interesting because at least in the -P, they have a small amount of access to what they want. And in the methylphosphonate, they don't have direct access to what they want, but there's something around them that they can use. And then in cellular P, you can see that in the -P, it was a lot higher. And that a normal trend that we saw that it was that cellular P, when measured, was a lot higher in the -P versus the methylphosphonate. And in the poly P, it was similar between treatments, that it was increasing, but it's important to note that these poly P changes between IGAL and EHUX were different. And so we believe that how they regulate this polyphosphate storage may be species dependent. And then implications. Going back to oligotrophic regions, like especially the subtropical North Atlantic, these zones are expanding with global warming and ocean stratification. So this suggests that the role of dissolved organic phosphorus and its role and the role it will have in primary productivity in these open oceans will increase with time, and the utilization of phosphonates by eukaryotes may influence future phosphorus and carbon cycling in future oceans. So I want to thank my mentor, Dr. Whitney, and an amazing lab manager, Deborah Lomas. Like never come across someone so just particular with everything. She was such a big help. And Dr. Mike Lomas, who we just very needed him at times this summer. It was very, very difficult. The Bigelow REU program, there was never a point where I didn't feel supported. And the National Science Foundation grant that supported me for working this summer, and the Archer Lab for your spectrophotometer, because we did just take it over. No one else really had access to that. We were in there for hours. And also the Countway Lab for your incubator, and when I had a vortex. And all the REU interns, I loved meeting you guys, and Turner. Thank you. Questions?
It's time for one.
Sorry.
Really nice work. And it sounds like it was a lot of work. All right. So this is just 2 species. How do you think these trends will be apparent in the other species of these taxa that you mentioned at the very beginning, or do you think it's going to be wildly different?
So what we had noticed in the past, like looking in our past data, was that there was no obvious trend in terms of cells that were grown in phosphonate. You know, there wasn't something that we noticed in one group versus those that couldn't, because there were some that were tested that could not grow at all in that methylphosphonate group. So it's hard to say. It seems like everything's very species dependent at this point.
Thank you.
Hello, everyone. My name is Andre Martin. I'm a rising senior at Haverford College. I'm a biology and environmental studies double major. And I had the great pleasure of working with Robert Schmedke and Dr. Mike Lomas in the NCMA this summer. And we were studying if we could use EPS producing algae to filter microplastics out of wastewater. This picture here is spoiler alert, we managed it. This is actually microplastics captured in wastewater. The blue particles are fluorescing microplastics, and the red behind it is chlorophyll fluorescing. But to jump into why this is important, first of all, is microplastics are any plastics that are below 5 millimeters in one dimension or another. They can be longer in others. For example, microfibers can be many millimeters long in one direction and less than 5 in another, but they are a major pollutant that we as a society have yet to really come up with a strategy to deal with. And there are a ton of different sources. A lot of them are manufactured once larger plastics escape into the environment. But one class of microplastics that we really wanted to focus in on was microplastics that we wash down the drain ourselves. So this is stuff from laundering synthetic clothing, and any cosmetics that use scrubbing microbeads are actually just microplastics that we then wash off. And so they are all sent to a wastewater treatment plant that is supposed to be able to pull them out of the water so we don't release them into the environment. But they're hard to filter at scale. Many of the properties that make plastics good commercial products also make them really difficult to properly capture, you can capture them with smaller scale filters, but when you're working on the scale of how much water we flush away every day, it's really just not feasible with current technology, which means they're escaping into the environment and doing damage. Even the best wastewater treatment plants are missing 10% of microplastics that they are sent, with many other plants releasing a much higher percentage than that. 10% seems small, but at the scale of societal water use, it's a huge problem. And it's one that we actually have a chance to do something about, because we know where these microplastics are passing through. Bottles that get dropped in the middle of a park or something like that and get washed into a waterway are very dispersed, and we can't attack the problem in the same way. So there's a lot of potential to stem a big tide of microplastics in these wastewater treatment plants. But any solution that is designed also needs to be additive. We can't undo the infrastructure of currently existing microplastic or currently existing wastewater treatment. We needed to design something that could be an extra step. So something that could both naturally interact with these microplastics and could use the natural resources already there in a wastewater treatment plant, made the NCMA and its new branch, the Center for Algal Innovation, turn towards solutions in nature. Recently in the study of microalgae, there has been a lot of excitement about a class of chemicals known as EPS, or external polysaccharides. Certain algae and bacteria have been using these polysaccharides, which are essentially a sticky goo that they excrete, to help them grow on microplastics and other substrates in marine environments that otherwise a single-celled organism would have no hope of clinging to. So if you don't use this chemical, you lose access to a potential space to grow. And so it's a competitive advantage, but it also requires the use of these sticky microplastic binding goo chemicals. And we wanted to see if we could take that same innovation that nature has brought us and turn it towards capturing these smaller microplastic particles. So with that research question in mind, we came up with an initial sort of concept of what we wanted to do. We wanted to see if we could use, if we could grow a thick mat of a EPS producing microalgae. In this case, we chose Glossomastix chrysoplasta, and then run contaminated wastewater over it with these microplastics in it, and then test the water before and after using flow cytometry to count if we could reduce the number of microplastic particles. And this would be an exciting sort of filtration model that could theoretically be scaled and could be fed with the nutrients already present in a wastewater treatment plant, and could theoretically sustain itself and regrow over time, so there wouldn't be a huge resource expenditure. So it was an exciting method that, if we could prove that it worked, has a lot of potential to scale well into the wastewater field. So I was brought on to do a lot of that transition from this is a cool idea that we had to we would like to be able to test this and quantify it. So a lot of my work was building the mechanisms to even test this. That sort of broke down into 5 major categories. We had to cultivate the Glossomastix, which was a major limiting factor in my project. It's a fast-growing algae, but for those of you in life sciences who work with bacteria and things like that, people are used to like a 12-hour turnover time to like double your amount of algae. We had to wait 2 or 3 weeks to grow enough for a test. So in a 10-week project, it could be a little bit difficult. And then we also had to create our own methodology for creating microplastic solution that we could test removal on. There are a lot of labs that work with and study microplastics, but most of them default to just using microplastic beads that are all the same size, all industrially produced. And it's not the greatest allegory for the actual wastewater we produce with microplastics in it that have a lot of sort of broken down pieces that don't look the same size or shape. So we needed to build our own. So we used PET in this case. We'd like to in the future bring in more microplastics into our suspension, but we had to find a way to grind and shape those microplastics in and of themselves. We ended up creating a suspension that had microplastics between the size range of 20 microns and 500 microns. And they came in a variety of shapes and aspect ratios, although we weren't working with microfibers that have the much higher aspect ratios. We also had to build a settlement rig to transition the glossomastix from how we grow it in these sort of round carboy tanks into a runway where we could run the water over it and guarantee the maximum amount of surface contact time. So, and once we had built all of that, we had to build a way to bring them all together in a controlled manner. So we also had to build a lot of the mechanics of controlling the flow rate and the angle of the plate that this raceway sits in and all these things. And then we tested them with flow cytometry by testing and counting the amount and size of particles in the microplastic solution before it was run over this algal lawn and then after. It took us a long time to prep all those components, but once we got them all together, we ran our first run and we got an immediate sort of very positive affirmative result. We got this beautiful looking, sort of very gelatinous, jello-like EPS matrix that was like a huge source of celebration. And then as soon as we were running microplastics over it, we could see that they were being captured really effectively. They would stick and stay there, but as I mentioned, the growth of this glossomastix was really limiting and we only could run one test. So it was an exciting proof of concept, but we couldn't pull a lot of data points from it. So we wanted to add something that would at least give us a stand-in to indicate that the algae was responsible for this. So we began designing a second set of tests that would tie amount of microplastics captured to amount of time that this algal lawn had to grow. So we turned that settlement rig into a seeding rig that would deposit algae on a 300 micron mesh that we would then scrape to make sure algae was just seeded into the tinier pockets of that mesh. And then we put that mesh in media and allowed it to grow. And then we would run the same test on it at 2 days of growth, 4 days of growth, and 6 days of growth, and measure the particle count before and after it ran over the lawn. And then through subtraction, we could calculate the amount of microplastics retained. The results, once again, were really exciting and sort of confirmed a lot of what we had been thinking. We saw at day 2, when there hadn't been much time for this algae to grow, that only 15% of the particles that we introduced to that lawn managed to stick and stay on the lawn. But once we hit 4 and 6 days of growth, we had 60 to 70% retention. And the only thing that had changed in that time was that the algae had more time to grow in and produce EPS. We also ran saltwater elutions on these tests, where we basically ran filtered seawater over it to see if the particles that we had retained actually would stay retained or if they would wash off. And we also saw lower percentage wash off with these later ones, it also indicating that we might have a higher level of bound plastics, where we've actually managed to get them to adhere to the EPS instead of just sort of settling on this lawn and then not moving because we were running the water very slowly. So this was a very exciting proof of concept, but we still have a long way to go on this project. And there's a ton of next steps to jump into, but some of the main ones are that we need to develop a method to quantify that biomass more accurately than just number of days that it has been growing. We have ways to do that, but they would be destructive to the sample. So we want to find a way to capture that better. And we want to perform some microscopy to make sure we understand exactly why they are binding. We have an idea that it is the EPS, but we haven't been able to confirm that yet. And finally, we want to test if we can run sort of different chemical elutions that can compete with that EPS and sort of bind preferentially to that EPS and then allow those microplastics to be released. And then we also, as we move forward in this project, are going to look into ways to cultivate that seeded mesh to be scraped and then regrow. So it's sort of like a reusable filter. And we want to look at ways to grow the algal mats vertically, because that would make growth spacing much easier and much more applicable in the wastewater treatment plants. I'd love to thank Mike Lomas and Robert Schmedke, my advisors, and David Fields for sort of being the point person on a lot of the REU's other programming. The NCMA staff helped train me on so many different things, and the REU interns were my family through all of this. Thank you to the NSF for funding me, and the rest of Bigelow for a wonderful summer. Questions?
So one of the issues you mentioned was the scalability for industrial wastewater. Could you see this as like a way of cleaning, like an at the source way of cleaning like drinking water for people, like on the smaller scale?
I think there is a barrier of upkeep where you have to be cultivating this algae very deliberately that works better at sort of industrial scales where you can have a large tank and you just have a protocol of swapping it out. Obviously, once this technology is created, people can take it in a lot of different directions. Developing the ability to interact with these microplastics will inform a lot of other people and give them other opportunities to take that and spin with it. But this project in its current form is really sort of focused on that industrial wastewater treatment phase.
Big one more if it's short.
Great job, Andre. I'm just wondering, is these algae or like is it bacteria or is it like fresh water or salt water? You mentioned running salt water through it. Like
it is a salt. It's a saltwater algae, but we are looking for freshwater algae that can also produce this EPS.
Gotcha. Thank you.
Thank you.
So just talking this. Good morning, everyone. I'm Finn DeWorkin. I'm a rising fourth year at Grinnell College, and I worked with Bigelow Analytical Services this summer, and the title of our project was Expanding Options for Monitoring Diuretic Shellfish Poisoning, or DSP. So what is DSP? DSP is an acute intestinal illness caused by the ingestion of marine biotoxins from shellfish. So we care about this issue because, of course, we want to keep the consumer safe. People in Maine, they come here to eat seafood, they'll consume shellfish. We care about their safety. But we also care about the lucrative shellfish industry, and we want to test the shellfish so they don't establish any industries in areas of high prevalence. Oh, sorry. So where does DSP come from? So DSP are marine toxins. They're the result from microalgae. So here we have two microalgae, Dinophysis acuta and Porcentrum lima. So these two, we have, sorry, so these microalgae, Dinophysis and Porocentrum, are responsible for producing DSP toxins. And these microalgae, they can actually float in the water column, as seen here on the far right for you. And they can be sucked up by these bivalve mollusks that shoot out their tongues, bring in the algae, and inside the shellfish, these microalgae can produce the toxins that are lipophilic and get stuck in fatty tissues, which then can transfer to humans when we consume the shellfish. So here are some toxins. These are phycotoxins. The ones we care about, we are monitoring 3 phycotoxins: oxidaic acid, DTX1, DTX2. In reality, there are many more, but these are the ones that we know cause DSP for now. So these toxins are lipophilic, they're in the tissue, and they're also heat-stable polyether acids. So they're polar and cannot be cooked out of shellfish. So this is what we did. Our objectives for this study was to implement the Interstate Shellfish Sanitation Conference approved liquid chromatography with Tandem spectrometry method for testing for these toxins. And once we implemented that, we wanted to optimize this method to get a low sensitivity for testing. And so in the future, we want to use this method to compare with some test kits, because the LCMS it's very expensive and requires a lot of technical know-how. So we want effective kits that are cheaper, but also sensitive. So this was the extraction method for the LCMS. DMR supplied us with samples from across Maine, from areas where we expected low incidence of DSP toxins. So we received homogeneous bags consisting of 12 to 15 blue mussels. And we homogenized them, and then we extracted those polar compounds out with methanol twice. We would then hydrolyze that to convert ester derivatives back into the original toxins. And then we would run a hexane wash to remove interfering compounds like triglycerides. And then we would filter that and run through the LCMS. And so here's the first step in the LCMS, which is the liquid chromatography. We used reverse phase liquid chromatography, and we would pass that through a C18 column, and we use gradient elution. And as you can see here on this graph, where we have time of separation and intensity, we had these separated toxins, and we had good resolving peaks in intensity. So onto the tandem mass spectrometry. So after separating out these toxins, they pass to the first phase, which is ionization, which is a soft ionization via electrospray ionization, where we run a gas along the eluted compounds as they come through this nebulizer. And we also run a current through it to remove these ions, and these ions pass to the first quadrupole. And here we have oxidaic acid, which has a precursor ion of 803.5. So in the first quadrupole, these precursor ions, they can pass through. We have 4 quadrupoles with charge, and these charged quadrupoles produce an electromagnetic field that filters out certain mass to charges, but allows the mass to charge for 803.5 to go through into our collision chamber. So in the collision chamber, we bombard this precursor ion with nitrogen gas, and that fragments it and produces more ions, such as our quantitation ion and our confirmation ion seen here on your right. So this quantitation ion tells us how much of toxin we have, and the confirmation ion just confirms the presence of toxin. So that passes through to another quadrupole, where again we filter out certain mass to charges. And seeing here we have 255.2, which passes through for quantitation, and then the other ones for confirmation. And this passes through to a detector where electrons are isolated from the ions, and these electrons are amplified at each dynode to produce an intensity peak. So here we have an example of a standard where we have good resolved peaks. And as you can see on this purple line, the 803 to 255.2 for oxalic acid and DTX2, we can actually integrate under these peaks to get peak area and use peak area to confirm concentration because they are proportional. And then we can get a concentration in micrograms a kilogram. And so here were the initial calibration curves we ran at the beginning of the summer. And as you can see, these curves actually vary a lot. So we ran a start and an end curve and then samples between, but we noticed this percent difference of around 34.9. So we noticed a problem of instrumental drift where our sensitivity was changing. So to resolve the sensitivity issue, we tried out a conditioning measure to our procedure. So seeing here, where we have no condition in the blue, you can see an increase in sensitivity, then a decrease. And so there's obviously some issue with drift, and this makes our samples less reliable. But with here in the orange, with the orange squares, where we treated with a 3 matrix blank condition, you can see there it's fairly flat, pretty consistent, and you can see that the CV at the bottom is a lot less than the blue. Additionally, we wanted to optimize our extraction method, so we played around with the Hexane Wash. And you can notice here with the red and the blue, and the red we have with oxalic acid with hexane. And we ran the same sample a bunch of times, injection numbers on the x-axis, and then we got a peak area on the y-axis. And as you can see, these lines are fairly similar, the with and without hexane, and they have similar coefficients of variation. So we just removed the step, which made extraction a lot faster, a lot cheaper because we had less reagents, and we reduced waste of hexane. Okay, and so once we implemented these conditioning measures, we ran our curves again, and as you can see here, we ran a bunch of standards. Our y-axis is concentration standard, our x-axis is concentration of standard, our y-axis is peak area. And as you can see, we did a curve at the beginning and at the end, and then we ran a standard in between. And it may be hard to see these curves because they are so reproducible that they almost overlap. So we significantly reduced the sensitivity drift between our start and end curves. And as seen by the percents for each toxin at the top. And seen here, we then used these well-resolved curves to solve for LOD and LOQ. And our limit of detection for OA, let's say, was 1.6 micrograms a kilogram, which is about 1.6 ppb. So very, very tiny concentrations. And then we could quantify all these concentrations together as OA equivalents. And so here's some results from some naturally incurred samples that we ran that we received from DMR. And as you can see in this map, I chose a few stations where we had multiple samples. And so on this top graph, we have latitude and longitude. This graph actually corresponds to this map. And as you can see, this is each sphere represents the average annual OA equivalent concentration for 2020. And as you can see, the southern stations have a lot more than the northern stations. Sorry, it's cut off for you, but there's a northern station up there that's very tiny. And then below that is the average annual Dinophysis acuta cell count in cells a liter for 2020. And as you can see, these two graphs correlate pretty well, except for one outlier, which is Pine Point, the blue sphere at the very bottom. But we were fairly limited in our DMR data, so more should be looked into it. So another thing we noticed was that aside from spatial variation, there actually wasn't that much variation seasonally. We had pretty consistent low levels of DSP, most DSP toxins, mostly OA and DTX1. We have mostly low concentrations that were persistent throughout the year. And although that won't cause any acute illnesses, that does pose the question of chronic exposure. And I did some research and found that there are links of chronic exposure to carcinogenic effect, embryo cytotoxic effects that can be teratogenic for people carrying children, and then also apoptotic capabilities in the liver cells. And so currently there's a dialogue to reduce the concentration the regulatory limit concentration to 80 micrograms a kilogram from 160 to 80. And that poses a lot of issues on testing, which is why it's very good that we get an optimized method for testing, and also could cause issues for the harvesters and people selling shellfish from the US over to Europe. So conclusions. So we took this method. It's a precise method. We tried to optimize it. We got to very low concentrations that we could test for. There are some disadvantages to it though. It's very expensive, and over the summer I realized how much technical knowledge it requires for use and interpretation. But further study will use this LCMS method that we developed to test with test kits that we will eventually have some information to present to the ISSC and get cheaper and better methods for testing for DSP. So acknowledgements. I'd like to thank the whole Bigelow Analytical Lab, my mentor, Carmen Cardizano, Craig Burnell, and Stephen Archer for their support and mentorship. I'd also like to thank NOAA and the NSF for funding this project, and also the DMR for providing us with samples and cell count data. Questions? Okay.
Thanks for that analytical talk. How long did it take to run all these samples? How long did it take to run through a set of samples, and including conditioning it with several blanks in between?
Sure. So each sample we ran took about 23 minutes. On my slide with, let me see if I can find it.
Okay, but how long to extract?
To extract 16 samples, it took about a day, and then to run each sample, it took about 23 minutes. Seen here, it takes about 12 to 14 minutes to elute through all the compounds. So it took a lot of time, and through the whole summer, we got about 125 samples run. Oh.
Hey, while the switch is happening, is it possible to hold the mussels after the time to let them depurate?
Sure, that's a great question. I actually did some research into the depuration rate, and one paper that I read showed that after a harmful algal bloom where we expect the most toxins, it took about 3 weeks for the shellfish to release all those toxins. So there's like a lagging period between harmful algal blooms and the concentrations in the shellfish.
Good morning, I'm Matthew Yost. I'm a rising junior at University of Maine Orono School of Marine Sciences, and I work with Dr. John Burns and Baptiste Chanel on study of ethylene presence within Cyanophora paradoxa algae. So to start, I'd like to give a bit of background on what ethylene is and why it's important. Ethylene is a hydrocarbon that has great commercial importance in today's world. On the left side, you can see ethylene as a petrochemical, where each year it is the largest petrochemical produced by volume with 190 million metric tons being produced. And on the right, we see it as a phytohormone or a plant hormone, where it is used in the food industry to shorten ripening times and involved with fruit growth. So for the case of our study, we looked at ethylene as a phytohormone. So in the plants, ethylene is very well studied and its purpose is well known. It's used to make plants grow faster. And you all probably know it as when you leave fruit together in a bag or something and they ripen faster than normal is because the ethylene gas that's being given off by these fruit speed up the ripening time of them. But as I just said, we already understand ethylene very well in plants, so there's no point to do studies in that. But of all photosynthetic organisms, plants are just one group, and we wanted to see if they could be present in another one. So we choose to study Glockophyte algae, which you can see are right down here, and plants are all the way here. So we wanted to check this group that is very distantly related to plants to see if we could find presence of ethylene in these two. And Glockophyte are a freshwater algae that are very understudied, and little is known about them as they diverged from a common ancestor hundreds of millions of years ago. So they're kind of a weird outgroup, and we're thinking if we can find ethylene within these, this could lead to bigger studies in the future. So before going on, I just want to talk about why any of this matters. So why would it matter if we could find ethylene within these algae that are just kind of weird and distantly related to plants? Well, to start, as I said, they're related from hundreds of millions of years ago. So if we can find ethylene and a pathway conserved in these algae and in plants, we could maybe discuss or research the possibility of ethylene being present from a common ancestor that they diverged from however long ago. And then also on top of that, if there was a common ancestor that had ethylene, we could look into the potential of current day species besides these two that also have ethylene. So in order to study this, we decided to base our experiment off of the ethylene pathway as we understand in plants. So one of the things that ethylene regulates or mediates in plants is ROS. And ROS stands for reactive oxygen species, which is the production of oxygen species such as hydrogen peroxide within cells that's typically used as a way for cell communication or apoptosis. And how this typically occurs within plants is there's a stress that occurs outside of the plant, and then this causes ethylene to signal for ROS production, which would then have the communication for the cells or kill the cells for whatever reason. So for this diagram here, just an outline of it is, so there's a stress typically that occurs, which then this precursor to ethylene, ACC, is converted into ethylene by an ACO enzyme, and then ethylene signals for ROS production within the cells. So we decided with the algae we were studying that we were going to stress these algae and then see if we could get this pathway to follow, so then we could get ROS production as a result, and then we could observe that and kind of confirm the pathway. But in order to make sure that it's the pathway we're seeing that is producing ROS and not something else, we decided to check different points of the ethylene pathway from plants by the use of inhibitors that we know work in the plant pathway. So these inhibitors included cobalt, which we use in cobalt chloride, which we know in plants inhibits the ACO enzyme in the plant pathway. So this prevents the ACC precursor from converting ACC into ethylene. So then there won't be ROS production. We also used STS, which is a silver compound, to inhibit ethylene, so it can't signal for ROS production. Then we won't see that again. And then last, what we used was ABA, which is abscisic acid, which would inhibit ROS production. So we similarly wouldn't see any of that. And then kind of to tie everything together, to do one last check for everything, we use paraquat as an oxidative control. Paraquat is an herbicide, which is known in plants to cause ROS production separately from any ethylene pathway. So if it was the same pathway we were seeing in these algae as in plants, we would accept paraquat to work independently of this pathway and not to be affected by any inhibitors that we were using. So the experiment we conducted was as followed. With each of our treatments, we had one treatment of just the Glockophyte algae with no stress or anything in them. They were just left them over the night to incubate. Then we did one of the ACC, the UV, which was a stress, flagellin, which was another stress, and then paraquat, which is our control. And then with each of those treatments, we did one of them with the 3 inhibitors. So ACC got one of cobalt, another one with STS, and then one with ABA, and the same for all the other treatments. And then after we left them for whatever time period, for depending on what the treatment was, the next day we would stain them with the cell rocks dye, and then we would take pictures of them using the microscope we had by fluorescing the cells. And from there, we would quantify the green we had from those individual images, which would show the ROS production, because green from the stain that we had would be indicative of ROS production within the cells. And it's worth noting for the following graphs that'll come up that the data may be different in terms of what the numbers are, but what matters is the statistical difference between each treatment, because for the microscope, you could take it in a different setting. So one treatment may have really high numbers because it was taken with a higher green saturation than another one. But as long as the groups of tests we did were the same for the microscope, that's what was important for us. So for ACC, which was the precursor for ethylene, as I mentioned, if we saw ROS production after we tested with ACC, this would support the idea of ethylene and the ethylene pathway, because it would be getting converted into ethylene and then signal for ROS. So that would tie into the pathway. And as you can see here, what we were looking for is in this picture here with ACC, that green is, if you can see it from there, is kind of the idea of what ROS would look like within these cells. So any green we saw was this production of hydrogen peroxide within these cells. And from here on this graph on the y-axis, we had the mean, median green quantification, which is what the program gave us as a number for green. So the higher the numbers, the more green the image was supposed to be. And then on the x-axis here, we had what the treatment was. And as we can see here, the treatment with the highest green value of anything was the ACC treatment, which would support the idea of ethylene and the ethylene pathway in these algae, because this meant that this ACC treatment was producing ROS, and it was likely because ACC was being converted into ethylene. And then the 3 inhibitor treatments here, which had ACC but had the inhibitors, showed a significantly less amount of ROS production, which would tell us that they were likely blocking this part of the pathway, which further supported the idea of an ethylene pathway in these algae. Then next, we did a UV treatment. In plants, UV is known to cause ROS as a result of an ethylene response. So what we did for this is we would leave the algae under UV light for 2.5 hours, and then we'd stain them to see if we saw any results. So similar to the ACC treatment, we saw that the UV stressor alone had a significantly higher amount of ROS production compared to all of the other treatments we had, and you can see some green here in this image. And then the 3 inhibitors and the control all showed much less than that too. So that further supported our idea that this UV was producing ROS as a result of an ethylene pathway in these algae. And then last, for our stresses, we did flagellin, which is meant to mimic a bacterial invasion within these algae. And for our flagellin treatment, we saw that flagellin had a much higher amount of ROS production compared to all of our other tests we did. And the 3 inhibitors, similar to our last 2 tests, showed that there was a big difference in the amount of ROS that they were making, which was telling us that they were also being blocked too, which supported the idea of the ethylene pathway. And then last, to make sure we just kind of wrap up all our results and confirm that what we were seeing was an ethylene pathway, we tested with paraquat, which, as I said, works independently of an ethylene pathway within plants. And for this, the results we got supported that idea with the Glockophyte too. So we could see that the paraquat here had a high amount of ROS compared to the control, but the 3 inhibitors did too, which told us that the inhibitors weren't able to block any ROS production because paraquat's not working through this ethylene pathway. So with what we gathered from that, we were able to conclude that ethylene is present in these algae and that there is an ethylene pathway too that's very similar, if not identical, to that in plants. So from here, what we can do is we want to work towards looking at other species that could potentially have ethylene, or maybe if another species kind of changed the way that the ethylene pathway works and it has a different function now, as a different metabolic pathway in different species. So what we did just a couple of weeks ago is we took some diatoms and dinoflagellates from the dock here, and we were testing those with the same treatments. And hopefully soon in the future, we can work towards finding other species that have ethylene present in them in some form or another. And I'd like to thank John and Baptiste for all their help this summer. It's been great. It's been good work, and all the interns too for a good summer. And I'd also like to thank the NSF for their grant they provided. Questions?
Really nice project. I'm going to ask a kind of tangential question. Your introduction talked about how ethylene is a petrochemical that's really tied into our food industry. Do you think if we're trying to move to a fossil-free future, could there be like a microbial solution for ethylene production that we could take advantage of from experiments like this? I know yours is looking at ethylene use, but what do you think about that?
Well, the thing is, we don't know. We didn't run any tests with how much ethylene these would produce the algae. So I'm not sure on what scale you would have to have enough ethylene that would be usable to say for if you want to use for some alternative. So that'd have to be looked into for future if we did test with how much they gave off.
Hi, great presentation. I was wondering about what you think this might imply for the depth and evolution of the ethylene signaling pathway, because we see it as ripening in fruits, and we see it as this response to stress in algae and other plants. So do you have any thoughts on why it could have originally evolved? Because it seems like it's a deep pathway.
I'm not too sure that, because there's some species with nitrogen converting algae in the ocean that it's believed the ethylene pathway could have potentially evolved into their nitrogen fixation pathway. So the role of this pathway could have evolved into different things depending on the species. So it's not too sure why it's kind of evolved as it has throughout whatever group we looked at.
Any other? Okay, I got one. So if they're signaling molecules to either ripen fruits or things like that, why would a single cell have it? I mean, to be able to send a signal to another cell, right, would require pumping out a lot of ethylene to have it reach in a fluid environment, to have it reach another cell. I wonder if you'd want to comment on it.
I'm not too sure. I mean, with these algae, how we have them, they're typically in colonies or big groups, so they'd be close together there. You'd think that if something would happen, potentially they might want to signal to other cells, but outside of them, I'm not too sure.
Okay. Hi, everybody. Over here. Yes, to the Zoom camera. Okay. Hi, everybody. So my name is Liz Westbrook. I'm a rising fourth-year undergraduate studying biochemistry at the University of Maryland in College Park. And this summer, I worked with Dr. Brian Demento, Dr. Chris Affley, and Dr. David Fields on a project investigating the effect of dissolved organic matter on the bioconcentration of short-chain chlorinated paraffins in the copepod species Calanus finmarchicus. So first, I'd just like to introduce you all to our group of molecules of interest, short-chain chlorinated paraffins, or SCCPs. As you can see from the example of an SCCP that I have on the slide here, they're pretty simple molecules, which just consist of a carbon chain, 10 to 13 carbons in length, and chlorine substituents, which make up 30 to 70% of the molecule by mass. Historically, we've been using these since around the 1930s, primarily as industrial metalworking lubricants, but they also have a variety of other uses, such as paint additives, plasticizers, and flame retardants. And their worldwide production was relatively high, and so they were officially declared a persistent organic pollutant by the United Nations Environment Program in 2017. This classification came about due to the molecule's toxicity, their potential for bioaccumulation, and their potential as a carcinogen. Unfortunately, while their production has greatly decreased ever since, they are already ubiquitous in natural waters. They're very persistent, and very little is known about their fate in the environment. So the larger goal of our project is just to better understand what's going to happen to them now that they're already in the ocean. Because of their structure, we know a little bit about what we expect their properties to be, and so we expect them to behave in a certain way. So let's take a look at that right now. One thing you might have noticed about the definition that I gave for this group of molecules is that there are many combinations of carbons and chlorines that could technically qualify as a short-chain chlorinated paraffin. And this can make them difficult to study at times. But one thing that they all have in common is that they're all considered hydrophobic molecules, meaning that they do not like to be left alone in just water. So we expect that if we put a bunch of SCCPs into a solution containing water and some hydrophobic substance like oil, that we're going to see most of those SCCPs partition into the hydrophobic substance. And in our case, that hydrophobic substance that we're talking about is not something like oil, but rather living organisms or copepods. So when we're talking about accumulation in a living organism specifically, we quantify this by taking what's called a bioconcentration factor, or a BCF, which is simply the concentration of the molecules of interest in the copepods over the concentration of the molecules of interest that stays in the water. For our project, we're adding a third character into this mix, and that character is dissolved organic matter, or DOM. DOM encompasses a very wide variety of very large molecules, which come from the breakdown of living things and can vary widely depending on where they come from and what ecosystem, what creates them. So there's a lot of variation in dissolved organic matter in general, but most dissolved organic matter molecules are so large, they actually create their own little hydrophobic microenvironment, wherein smaller hydrophobic molecules like SCCPs can accumulate and therefore be kind of taken out of this binary water copepod system. So what we expect is that because we have these dissolved organic matter molecules that we've added into the system, we're going to see less bioconcentration because it's competing with the copepods to absorb the SCCPs from the water. So one thing I forgot to mention is that we can also quantify partitioning into dissolved organic matter. And it's quantified in a similar way. You notice these equations are very similar. This partitioning coefficient, KDOM, is just the concentration of the SCCPs that go into the dissolved organic matter over the concentration that stays in the water. So that'll be important for later. So again, we predict that when we add dissolved organic matter into the system, we're going to see lower bioconcentration factors in the copepods. We're particularly interested in understanding if and how the properties of dissolved organic matter and the environment from which it comes change the way that it affects this process. So some of the environmental factors that can affect the properties of DOM include the flora and fauna of the environment from where we're getting it, and also the water chemistry and some things that may weather the DOM, such as sunlight. So we ran these bioconcentration factor experiments on 3 different types of dissolved organic matter to investigate how these properties might change the way that it affects bioconcentration. The first type of DOM that we extracted came from a salt marsh near Popham Beach, right here in Maine. And we ended up with a couple 100 milligrams of dissolved organic matter from that extraction. We also extracted water from the Damariscotta River right off of the Bigelow dock here. And we got just under 200 milligrams of dissolved organic matter from that extraction. And the final type of dissolved organic matter that we used came from the Swanee River in Georgia. And we actually bought this DOM from the International Humic Substances Society. And the reason that we did that is because this particular type of dissolved organic matter has been being extracted and used in experiments for many years, and there's a lot of data out there that can help us make sure that we're on the right track, and we can compare our data to make sure we're on the right track with these 2 types of dissolved organic matter that we extracted ourselves. So one thing I did want to point out is that we got a lot more dissolved organic matter from the salt marsh than we did from the Damariscotta River, and that's because salt marsh environments tend to be much more rich in dissolved organic matter. So we actually extracted half as much water from the salt marsh, but ended up with like 3 times more DOM, whereas seawater just tends to not have as much dissolved organic matter in it. So we have our dissolved organic matter that we used, and we also needed to collect copepods offshore in the Gulf of Maine. And I'd just like to quickly introduce copepods in general. This particular species that we used, Callinisphon marchicus, is a species of subarctic copepod, and these are a zooplankton, so they're only a couple millimeters in length, but they're very important because they make up the base of the food chain. So they're not only a great model organism just to generally model how these compounds might be accumulating, but they're also important because when you see accumulation in a species like copepods, that can mean more as it goes up the food chain. So the classic example of biomagnification. So they're not only significant, but also just a great organism to use as a model for other organisms and just accumulation in life forms in general. So once we had our dissolved organic matter and we have our copepods, we dosed artificial seawater with short chain chlorinated paraffins. And then we added the DOM in the copepods and allowed those paraffins to equilibrate into those different hydrophobic environments for 24 hours. After the 24-hour exposure, we immediately removed all of the copepods from the DOM water, and they took any SCCPs that had sorbed onto them with them. We then added a hydrophobic solvent called dichloromethane, or DCM, to the DOM water, and we shook it for 24 hours. And the reason why we did that is so that the SCCPs could partition into the DCM. And they did that because it's more hydrophobic than the DOM water. So most, if not all, of the SCCPs are going to prefer to be in that DCM phase. We then removed the DCM using a separatory funnel. So you can see it's this bottom layer here, this clear bottom layer that we just drained out, and we concentrated it down and got our SCCP sample into a vial for analysis. And as for the copepods, we put them into vials containing a strong base sodium hydroxide solution and also another hydrophobic solvent called hexane. And then we added silica beads to this mixture and we shook them really hard in an instrument for 10 minutes, effectively decimating the copepods and allowing the SCCPs to partition into the hexane layer. So then we removed the hexane via a pipette, and we have these two vials now that have our water extracts and our copepod extracts for every trial that we did. And we put these into another instrument called the GCMS, or gas chromatography mass spectroscopy instrument, which essentially tells us what the concentration of short chain chlorinated paraffins in our extracts is. So what we get from this is that we now have the concentration of short chain chlorinated paraffins that were in the copepods, and we have the concentration of the short chain chlorinated paraffins in the water. And as we solved this equation before, we can calculate the bioconcentration factor using these numbers. So these are the results. And I'm just going to walk you guys through this really quick. So on the bottom here, you see three different compounds, tetrachlorodecane, tetrachlorododecane, and hexachlorodecane. And you can see the structures of these compounds on the side here. These are three SCCPs that we chose because they have varying carbon chain lengths and chlorine contents, chlorine distributions. So we felt that they encompassed a variety of different properties that you could see from a short chain chlorinated paraffin group. And so you see the three different compounds and you see their bioconcentration factors under different treatments here on the y-axis, the log of their bioconcentration factors, because these numbers tend to be pretty large. So you can see here's our artificial seawater control is here in blue. And then we have the three different colors are the different DOM treatments. So if you look at this, you'll see that we did see a decrease in log BCF for most of our DOM treatments compared to our control treatments, with the exception of this tetrachlorododecane, which increased in the DOM treatment, and we're not really sure why that happened. These might not look like very dramatic changes, but when you consider that this is on a log scale, these actually are pretty significant. So, or they're dramatic changes in the bioconcentration factor. So what they actually are is up to like around a 60 to 80% decrease for many of these different DOM treatments. So this means that we're seeing 60 to 80% less SCCPs in the copepods after when they're exposed in like a DOM water compared to just a plain artificial seawater with no DOM. And that's a pretty dramatic change. As far as the difference between the different DOM types goes, we didn't really see much variation there, and we're still working on characterizing the dissolved organic matter to kind of see what its properties are and why that may be. One other thing I wanted to mention is that there are differences in specific SCCPs that can affect how they partition into the DOM. So if we have a SCCP that's more hydrophobic, then we would see possibly more partitioning into the dissolved organic matter, and then a higher percentage of these SECPs being in the DOM phase means that we would expect their bioconcentration factors to be even lower. And so here's using this equation that was developed by a candidate all in 2003 that you can use to predict the bioconcentration factor based on partitioning into DOM and the concentration of DOM in the solution. We actually saw that our measured values for bioconcentration factor were lower than the predicted values. And we're not exactly sure why this is. We think it's because when we measured the partitioning coefficients of these compounds into DOM, we did it in millicu water, which is fresh water. And then when we did our bioconcentration factor experiments, we did this in salt water, which has ions in it that would farther drive the compounds into the DOM and decrease bioconcentration factor even more. So in conclusion, it appears that bioconcentration factor can be lowered by the presence of dissolved organic matter. And this is important because if we want to model what's going to happen to these compounds in the ocean, so we are interested in understanding the effects of dissolved organic matter on how it can accumulate in organisms. Finally, I'd like to thank my mentors, Dr. Brian Demento, Dr. Chris Hapley, and Dr. David Fields. And I'd like to thank Maren Nemesto and Erin Beering for answering my questions throughout the project and offering support. And I would also like to thank the other REU interns that were here this summer for providing a network of support, especially Hannah Sterling for being there in the athlete lab with me all summer. And yeah, so any questions? Long side. Yes, Molly.
I just wanted to know how you extracted the DOM in your project.
Thank you, Molly. That's a great question. So this is quite a process. We extracted DOM from they're laughing at me. Extracted DOM from two different sites. And so the first step in the process was to filter all of the water down to 0.2 microns. And that removed anything that was not fully dissolved. And then we pumped all of the water through these cartridges that have a chemical affinity for the dissolved organic matter, and that removed it from the water. And then we also pushed methanol through those cartridges, and that has an even higher affinity for the dissolved organic matter. So we ended up having this kind of methanol and DOM solution, and we just evaporated all the methanol out and freeze dried to obtain the final product that you saw before. Thank you for your question.
So building off of Molly's question, when you had your bottles with the DOM in there, they looked really brown. And if I pulled water up off the dog, I don't think it's that brown. So or maybe it is. How did you decide what concentration to put back in?
Well, we ended up going with a concentration that is much higher than what you would see in nature. But the reason why we did that is because it's an acute exposure for just 24 hours. So we were trying to model what we would see over a long period of time in a short period of time. So we kind of exaggerated the DOM concentration to see the differences that you might see just in that short 24-hour period.
Hi. So thanks for the talk. That was really interesting. So Copepods aren't quite at the bottom of the food chain.
Right.
Do you think there's the processes or organisms that might accumulate these compounds before they get to copepods or at an earlier level?
I can imagine that any organism that has these hydrophobic environments in them or presents a hydrophobic environment would see these compounds accumulating in them. So it wouldn't just be animals, but any like phytoplankton could also see this happening. So that could also biomagnify into the copepods. Unfortunately, we didn't have time this summer to do like a feeding experiment where we see how they biomagnify. So we just focused on sorption directly to the animal. Yes, David. Hi.
So in your DLM samples, I mean, it seems like they're these aggregating mechanisms that are shore bound and then get exported off into the ocean. I'm wondering if when you pulled them out of Bigelow and you pulled them out of the Swanee River, if you measured the inherent compounds that they were holding on to already, and was there contamination in them?
We did not do an analysis for that. I don't think that would be too difficult to accomplish if we were to look for compounds in the DOM already. But yeah, we definitely it's thought that DOM is a huge sink of some of these organic contaminants. So we would expect to potentially find some contaminants in there.
Could they have been saturated? And the effect you would have seen would have been even higher because they were already preloaded when you got it?
I'm not, I couldn't tell you for sure. I'm not sure that they become saturated or they kind of just absorb, unless they are full of these compounds, but they kind of like stick to the aromatics in the DOM. So I don't know that they would be saturated, but potentially there could be contaminants in there. Thank you.
So there's a 15-minute comfort break. So rush because there's limited bathroom comfort areas, and be back in 15 minutes if you will.
Alrighty, hello. My name is Owen Keller. I'm going to be a senior at Bates College in the fall. And this summer, I was working with Dr. Benjamin Twining, looking at feeding cows seaweed to reduce methane emissions, and specifically looking at iodine seaweed in milk. So my project starts with cows, not very common at an ocean science research center. But the reason that this is important is because cows contribute greatly to global warming. And they do so because they're the largest anthropogenic methane producer. So when you think about like humans producing greenhouse gases, cows are producing the most methane. And methane is important because it has a super high ability to retain heat. So when you think about obviously the sun pouring in, giving heat to the earth, methane is keeping that heat inside the atmosphere. So in the last 5 to 10 years, there's been a lot of work done that's shown that if you feed cows seaweed, that it can reduce the amount of methane that they produce, which was hugely exciting. And so the Bird Busters project is locally in Maine and New Hampshire, primarily a project that's looking to get these same methane reducing effects, but thinking about kind of the bigger picture of it. So not just looking at like the methane emissions, but also understanding like where can we get the seaweed from? Hopefully that's a locally sourced product, because if you're transporting seaweed from a faraway place, then obviously you're also adding to emissions gases. So the other component of that is like the animal's health and ensuring that the milk that the cows produce is still healthy. And so when you think about seaweed, has always been a really high source of iodine. And so that could potentially be an issue here. So what exactly is iodine? Iodine is an essential nutrient. So all humans, all organisms basically need it to live, and especially in thyroid function and developing children primarily. It's really important. And so this is relevant because as you can see in this table, the range between what's recommended that you have every day and the upper limit, at which point it could become unhealthy, is a relatively low margin. So for children, for example, the recommended value is 120 micrograms, which is, as you can see in the little cup of milk, just about a cup and a half of milk a day. And so the upper limit there is 200. So just the difference of a single glass of milk a day could push you over that threshold of what you should not have anymore. So for adults, it is a little bit higher with a recommended value of 150 micrograms. And then the upper limit being 1100. And for a 600-pound cow, obviously those values go up again from 3,600 to 50,000. So thinking about seaweed, it's known that there's a lot of variation in the iodine in seaweed. And so understanding like what caused those variations, and then thinking about different species of seaweed, different geographic locations, then also just between a single blade of a seaweed, there's been shown to show variation. So I'm going to get back to this, but on my little diagram there, you see how if you get further away from the holdfast, that there appears to be less iodine in the seaweed. So understanding like what causes that could be helpful in determining what seaweed to feed the cows. And then the second component is understanding how much of that iodine really gets into the cow's milk. So here's an overview of my research goal and questions. So again, starting with reducing methane, taking away from the contribution to global warming. And so starting with looking at brown seaweed, which is known to have a high level of iodine, but Saccharine latissima, which is sugar kelp, is incredibly abundant along the coast of Maine and is the most transported or exported and produced seaweed. So if you're thinking about doing this in like a sustainable way, and Burpbuster is getting seaweed that's locally sourced, the logical place is to start at sugar kelp because there's a bunch of it. And so again, understanding the drivers of what causes the variation in iodine levels. And then lastly, looking at milk, looking at the iodine in milk from cows that have been fed seaweed and then cows that have not been fed seaweed. So to start off with my seaweed methods, back in 2017, Lauren Chacho sampled seaweed from 13 locations along the coast of Maine. And I found those in the freezer up in the twining lab. And I took triplicates from each of the locations. I sampled 10 grams, thawed them, freeze-dried them, and then ground them to a fine powder with a coffee grinder. And that was probably the most painful part of my summer. Grinding coffee is wildly easier than it is to grind seaweed. But that was necessary to then be able to digest the sample in a microwave and basically get a liquid dilution of the seaweed, which was diluted in TMAH. And this is relevant because the TMAH as a base was shown to keep the iodine and preserve it better for a more reliable iodine analyzation. And so this was then put in the ICP-MS, or the mass spectrometer. And the last thing I'll say on this slide is that a certified reference material was used that had a recovery of over 80%. So that gives us credibility. We are confident enough that the levels that we're seeing in the seaweed are actually what are found out in nature. So here's my seaweed data. There was a mean of 1,360 micrograms of iodine per gram of seaweed. And on my graph, you can see on the left here is East Point's the southernmost point that was sampled, and then going over to the north end of the coast of Maine. And so with a standard deviation of 868, there is a lot of variation here, which is not necessarily surprising to us. But we have not yet been able to find cause for this. That's clearly something to look into to really understand, like, is there a reason why Stones Rock is significantly lower than Merchant Island? Or is that just a cause of the variation in the elements in the ocean? The range that we saw in the main seaweed falls within the range previously sampled. So the red line indicates the levels of iodine shown in the same sugar kelp in Ireland. And then the yellow line is the same samples up in Norway. And if you're thinking ahead and thinking about how much iodine a cow needs or should not have to get these methane reducing effects, it's previously been shown in other studies that 5% of the cow's diet would need to be seaweed. And so that would put you at 2,500 grams a day, which if you're doing the math at home, I'll get back to it, but it's relevant. The second part of the study, as I said, is I sampled from the same blade. I sampled 5 different pieces of it, so at 1 foot increments away. And you do see this trend from 1 foot where it's close to 3,000 micrograms per gram of iodine, or micrograms of iodine per grams of seaweed. It does decrease significantly down to just over 1,000 in the 5th foot, but they're at this level, there's still quite a bit of variation. So again, interesting that even within one thing of seaweed, it can vary that much. And this last graph that I have here kind of goes back to the variation shown on the last slide. These different elements along the y-axis, and then their percent of variation. And the same sugar kelp samples, Lauren, when she sampled them, she looked at these different elements. And so it's not as low as some of the variation that you see on the left, but it's also not nearly as high as like iron or lead, for example, that you see on the right. So what does this mean? I'm not here to tell you what you should or should not eat, but looking at the numbers, if you think about having just a single gram of this seaweed before it's processed, you're already way above the limit that's recommended for humans. And so getting back to my math question, if you take 2,500 grams of the seaweed and multiply it by the mean of 1,360 micrograms per gram of iodine, or per grams of seaweed, gets you over 3 million micrograms of iodine per day. And that's significantly higher than the 50,000 that's recommended for a 600-pound cow. So that then takes me to my next part of my presentation, which is looking at how much iodine that ends up in the milk. And so down the road in Freeport, Maine, at Wolf's Neck Center, there were 22 cows that were subject to this feeding experiment. And half of them were fed seaweed as a component of their daily diet, while the other half, the other 11, did not get fed the seaweed. And this experiment lasted for 7 weeks. After a 2-week acclimatization period, milk was sampled at 2-week increments. And so I had 3 different sampling dates. And in similar to the seaweed, I took the samples, I diluted them with the TMH solution, put them through the plasma mass spectrometer, and then again had a certified reference material recovery of over 80%. So here's a look at my milk data. Previously should have been in that box as a word. Surprisingly and contradictory to previous data, the cows that were fed seaweed did not produce milk that had a higher level of iodine than the cows that did not get fed the seaweed. And so that made me wonder, because there still was a great level of variation between the different cows and between their different milks as to what might be causing this. And so the first thing that we looked at was looking at different breeds. There were Holstein cows and Jersey cows at Wolf's Neck Center. And so while there were only 4 Jersey cows, so not necessarily the best sample size, these Jersey cows showed a significantly lower level of iodine in their milk than the 18 Holstein cows. So thinking about different physiological differences could be a good next step into understanding how much iodine ends up in the milk. So going forward, understanding the different components of the feeding methods and really being sure that the seaweed that was put in front of the cows was really consumed is important, because I can imagine that if you put the seaweed in a bucket next to their normal hay, that they might not be as inclined to eat it. And then also going back to my initial data from the seaweed is really understanding, like, you do see such a huge variation in the iodine in the seaweed. And so understanding which part of the blade, where it's grown, and especially different species of seaweed to get the methane reducing effects without pumping huge amounts of iodine into these cows. Secondly, thinking about physiological factors. So going back to the breed, does that really have a significant difference? And so therefore, is there a cow breed that will have like better results going forward with this? And then other factors, for example, days in milk. Does the age of the cow and how long they've been producing milk, does that play a role here? And so finally, kind of to bring it all together, thinking about this project as a whole, not only do we want to see these methane reducing effects and take away from the strain that we're putting on our environment, but also making sure that we're raising healthy cows, and then that these cows are then producing milk that can be used for consumption and ensuring healthy dairy products. So I'd like to thank a lot of people. I'm very appreciative of my opportunity here. The NSF grant, the Shelby Cullum Davis Charitable Fund, which funded my projects here, the Twining Lab. Thank you, Ben, Brittany, Sarah, you guys all helped me in the lab. Lauren Chacho for the seaweed collection. And then everybody else here. There are a lot of members of the Birth Busters Project. The cows undoubtedly need to be thanked. And yeah, thank you all. Are there any questions?
So what would happen to somebody or a cow that consumes too much iodine? Like what would be the consequence of like saying like doubling the amount you're supposed to have?
So that's a good question. In the most severe case, it can lead to health conditions as serious as thyroid cancer. And realistically, with everything in life, like there's only a certain amount of sugar you should have in a day, and people kind of go past that a bunch. So if you feed these cows for 7 weeks and they have too much iodine, I don't think they're necessarily going to have like instant effects. But if you think about long term, like if this iodine in milk is suddenly twice as high as it normally is, I think then you would see like generations of like maybe increased levels of thyroid cancer or other serious conditions like that.
So just to follow up on that real quickly, in the feeding experiment time frame, you didn't see any negative health effects in the cows that were purportedly eating the seaweed?
No.
Is there a reason that you chose dairy cows as candidates for the seaweed project over like beef cows?
That wasn't necessarily my decision. It's somewhat over my pay grade. That's a good question. I think the dairy cows were at Wolf's Neck Center. They have more of them, so I think they're somewhat more easily dealt with. And also you can throw away the milk, say it is dangerous. Whereas if you start giving this huge amount of iodine to meat cows, and you're not totally sure where the iodine goes, wasting all that meat would be a waste. So. Yes, David.
Is it possible to cultivate the seaweed in environments with lower iodine? And so you actually farm seaweed, or is the iodine level that they have necessary for their physiology? You know?
That's kind of its whole like separate realm of research, and that's absolutely a question that should be asked because already there are ways they like, for example, just drying them in the sun or bleaching them reduces the iodine concentrations. And so it would make sense to me that there would be a way to basically grow seaweed in a way that would decrease the iodine levels. And it could be as simple as like growing them in fresh water. And an issue there is thinking about how much seaweed you would need to produce to feed all the cows in the world, whatever level of seaweed it would be. But that's absolutely a question that's worth looking into.
Short one.
I've just forgotten my question. Never mind. That was great.
Thank you. Sure. You show different CVs for different elements in sugar kelp with iodine in the middle. What is known about what causes variation of other elements? Does this help inform hypotheses for what causes variation of iodine? So if you go back to this, I know I'm running out of time. If you go back to this, so in general, you can't see me. In general, the elements on the left are more closely related with growth, whereas the ones on the right, for example, lead and iron, just that variation depends more on like what's available in the surroundings. So I'm not, yeah, that's kind of the short answer is that on the left side, because the seaweed needs it to grow, there's less of a variation, but I'm not sure. Yeah, we need to look into that further.
Thank you.
I'm gonna leave it down. Okay. Awesome. Okay.
Good morning. I'm Lydia Duncan, and I am going into my final semester at the University of South Carolina. My mentor was Dr. Kath Mitchell from Bigelow Laboratory for Ocean Sciences. And my project was the estimation of biomass and desiccation of intertidal seaweeds using reflectance for future unmanned aerial system survey applications. Funding for this project was provided by the National Science Foundation. Bigelow Laboratory is both the undergraduate research experience in the Gulf of Maine and the World Ocean, and the Center for Seafood Solutions, and also the Maine Economic Improvement Fund Small Campus Initiative. Whoop. There we go. So here are intertidal seaweeds that we're looking at. We have Ascophyllum nodosum, which would be called Asco for this presentation. And we also have Fucus verculosus, which would be addressed as Fucus for this presentation. We have Asco is more of like a green, olive green near the holdfast, and then Fucus can range anywhere from like green to like little orange tips, and then more brown at the holdfast. The significance of these seaweeds is that they drive the intertidal food webs, and they are also very important for carbon cycling in the coastal ecosystem. Asco is also commercially harvested in the Gulf of Maine, so being able to monitor these seaweeds ensures sustainable harvests and harvest limits for the Asco, and also gives us a better understanding of the intertidal food web. The end goal of, and like the future goal of this project, and what my mentor is working for, is being able to use hyperspectral images collected from the unmanned aerial systems, or UAS surveys, over areas of Gulf of Maine to build machine learning models that will map the distribution of seaweeds and also accurately estimate how much seaweed there is. So my hypotheses for my project is changes in the biomass affects reflectance for samples of both Asco and Fucus, and also desiccation, or the time left drying, affects the reflectance for Asco and Fucus, because we want to estimate how much there is, and if these two variables change how the reflectance looks, that will change how we make our models in the future. So my methods, for my project, here we have a nice schematic of my experimental setup, and next to it is a picture of the setup itself. And then we also have a bulleted list of what I did during the experiment. We had blackout cloth covering everything, and the lights were off in the room to prevent any extra reflectance coming in that would affect our data. We had halogen lights suspended above to illuminate the sample. We had a fiber optic sensor that was connected to a field spectroradiometer that was used to collect hyperspectral data, or just the percentage of light reflected from each sample for wavelengths from about 350 nanometers to close to 2,100 nanometers. We had our samples had to be larger than 7 centimeters in diameter. That way, the sensor had a 22-degree radius of field view. So at the 7 centimeters that we kept it at, we needed to keep our samples larger than that, so it would only see our samples. We also chose to keep the sensor 17 centimeters above the top of the sample to ensure our ability to compare our results from both the drone data and our field site samples. And then we had an adjustable height prop to move the sample up and down to keep that 7 centimeters. We had 5 varying samples for both Asco and Fucus that just increased in biomass. And then we took 40 spectral measurements for each sample. That was taking 10 spectral measurements and then rotating at 90 degrees. So we got a nice average. And then we left them out throughout the day and took 3 different time points so that we got a good look at the desiccation for each sample. Here are some images of our samples. And this top row has all the Fucus samples all the Asco samples from 1 to 5. And the bottom has the Fucus samples from 1 to 5, 5 being the ones with the largest biomass. Up here is a table with all of the sample wet weight, dry weight, and canopy depths. Our dry weight was collected after leaving the samples in the drying oven. Here's a look at the hyperspectral data. As you can see, this is very, very messy. So here we have our wavelengths along the bottom and then our reflectance percentages up along the y-axis. Here we have fucus biomass, where these are all of the samples from the last slide shown for the first time point of fucus, so it's just increasing in biomass. And then here in the second graph, we have the ASCO2 time points. So we have the first time point, second time point, and third time point. And so as you can see, these either switch which one is on top or valleying and peaking at different points, or they're very close together. So it's hard to say what exactly the relationship is used for desiccation and for increasing biomass. To solve this problem, we use the normalized difference vegetation index. This is the NDVI is based off of Landsat 8 surface reflectance bands, and it's used to estimate vegetation around the world. And so we averaged the near infrared wavelength band reflectances and the red wavelength band reflectances, and plug them into this nice equation to get a value for each sample so that we can compare them in linear regressions. So these are our linear regressions for our ASCO biomass. As you see, we have the NDVI along the x-axis, and then dry weight, wet weight, and canopy depth along the y. All of these linear regressions are significant to the p-value of 0.05. No, 0.05. But there was a supersaturation point where we realized that any sample that was above our ASCO sample 4 did throw off our curve, so we excluded it. So we did find only a significant relationship for samples of lower biomasses. The same thing we discovered with fucus. Here again, we have dry weight, wet weight, and canopy depth against the NDVI. And so these linear regressions are also significant, but we had to exclude a fucus 5 sample and its time points because it gave a supersaturation point for our data. For our desiccation, we did find that there was no overall significant relationship between ASCO and fucus for our desiccation. Here are our plots for time in hours and NDVI for ASCO and fucus samples. And as you see, there is no correlation or pattern with these points because they're kind of scattered everywhere. We did attempt to look at individual samples and see if there were significant relationships. But we either think that either 3 time points was not enough time points, or there's just no consistent relationship between samples because we couldn't compare them or find regular, consistent relationships with our data. So in conclusion, we found reflectance has a significant relationship with increasing biomass measured by canopy depth, wet weight, and dry weight for both ASCO and fucus. Reflectance and desiccation were not found to have a significant relationship for ASCO and fucus, and these results show potential for estimating biomass of intertidal seaweeds with future UAS survey methods. But of course, we have a lot of extra future work that we need to do, such as we need to sample more intertidal sites. We only sampled 1 site and only ran 1 experiment, so that we need to do more to get more data so that we can have better regression so that we can understand these relationships better. We want to use the partial least squares regression model to have the full hyperspectral data be applied to the regressions instead of just using NDVI. And then we also want to investigate using machine learning algorithms to map that estimated biomass so that we can have something to show for this project that's real and applicable. Thank you. Any questions?
About time. That was great. Thank you. My question is about the relationship. So you saw this positive relationship between NVDI and DVI and a couple of the biomass indicators, and they were positive, but they definitely weren't like linear, right? You know, and I'm wondering, do you or does the broader project have a sort of a cutoff, or like, is there a level of relationship that needs to be reached before you decide that it's useful, or is just having a positive relationship enough? You know, like the R squared was probably not very high, but maybe that's good enough. Do you have a sense?
So for ASCO, the R squared values were actually very, very low. For samples 1 through 4, we actually found it went higher if we excluded 4. But at that point, we're just like picking points. Definitely we want to look at more values in between, because our values kind of jump from below 20 to close to 60 and then to 120. And so being able to get more data points in between those ranges would probably help make a better model. And then for fucus, actually, so like these are all like sample 2, but it's like time point 1, time point 2, and time point 3 that are like spread out. And so while it is graphed as linear, like if we only did like the first time points, it would be more linear. And so we're looking more into like seeing like what are all those differences, and then where exactly that supersaturation point is, so that we can say from our like UAS surveys being like, oh, we know here is like this deep, or it could be deeper, so that we can put a normal limit on like how much they're able to harvest there.
Thank you. David got food while you were
I did. I was eating while I was standing here. I have a question. So it sounds like you're trying to adapt this project to be using drones to fly over. Is it possible to transition that to satellites so you can get kind of broader global views of that?
Good question. So in the intertidal, it is very, very difficult to get satellite images that are clear and at low tide, specifically because you can't control whenever the satellite will pass over that time. There's also like a 3-day difference each time the satellite goes around. And so while people have used satellites in the past for ocean color or some similar like this, it's not super accurate. And so we're trying to get these models as accurate as possible. And so that's why instead of using also like planes, they've decided to use drones with multispectral or hyperspectral images, imaging systems on them. We have another question, David.
So would this same calculation still work when you're looking at seaweed that might be attached to a pale rock as opposed to a dark background?
Yes, so that's actually what the field samples are for. There's people that are going out and already flying drones to get those images, and then also going through the intertidal and looking like, okay, what is this area here, and what is this like patch that is like mixed seaweed or seaweeds that aren't ASCO and fucus, and looking at them and being like, oh, we know exactly what that looks like. And then we take the reflectance measurement of that in the field and be like, oh, this is what it looks like, and this is how we can build a model. But we were just trying to predict if we could like estimate what biomass is or desiccation, but we were looking more if we could actually estimate biomass from the reflectance data. Yeah.
Hi, my name is Hannah Sterling, and I'm a senior at Roger Williams University. Oh, sorry. My name is Hannah Sterling. I'm a senior at Roger Williams University, and this summer I worked with Chris Appley looking at the biodegradation of photooxidized oil products, implications for the fate of marine weathered oil spills. On April 20th, 2010, the Deepwater Horizon oil rig exploded in the Gulf of Mexico, killing 11 people. Over the course of 87 days, 4.9 million barrels of light crude oil, which equates to about 210 million gallons, were released from a depth of about 1,500 meters, forming both a deep water plume and an oil surface slick that persisted for 102 days. This environmental disaster spurred a new wave of research and funding for the fate of oil spills. So there's a number of factors that impact the fate of oil in the environment once it has been spilled. Some of these factors impact the oil physically, others impact the oil chemically. Some of the factors that physically impact the oil are dissolution, evaporation, emulsification, and sedimentation. So dissolution is, as it says, when oil dissolves into the water. Evaporation is when some of the more volatile compounds evaporate off the top of an ocean surface slick. And emulsification is when water gets integrated into the oil and increases its viscosity and density, which can then allow it to sediment onto beaches, coastlines, and the ocean floor. The two factors that impact the oil chemically, of which I focused on for my project, were biodegradation and photooxidation. So biodegradation first is when microbes break down oil compounds either into biomass or metabolically to form carbon dioxide. Photooxidation is when the oil in a surface slick reacts with light and becomes oxygenated. So on the left are some examples of some crude oil compounds, and on the right are their photooxidized counterparts. So at the top left, you have N-alkanes, then isoalkanes, and then polycyclic aromatic hydrocarbons, or PAHs. So these compounds interact with light and react and become oxygenated. So oxygen functional groups will kind of add randomly all over the molecule. You can get carboxylic acids, ketones, alcohols, and aldehydes. Prior to Deepwater Horizon, it was thought that photooxidation was not really a major impact on the fate of oil, but research after Deepwater Horizon has shown that up to 50% of the oil mass in a floating surface slick can actually become oxidized in the first couple of days to weeks. And so because Deepwater Horizon was so recent, we don't really know how these photooxidized oil compounds interact with those factors that I talked about earlier, specifically when it comes to biodegradation. So we know how those crude oil compounds biodegrade, but we don't necessarily know how the photooxidized oil compounds biodegrade. And so that leads me to my project. So I wanted to quantify the degree of biodegradation of photooxidized oil specifically, and then compare that biodegradation between oil that has and has not been photooxidized. And then finally, I wanted to see how the marine bacterial population was affected by enriching with this photooxidized oil. And so my experimental plan for the summer, I ran a preliminary experiment for 2 weeks in a small volume size, 500 milliliter bottles. And I wanted to find ideal variables for microbial growth and oil biodegradation. So I tested a number of different concentrations. I tested a number of different dispersion methods. And then based on the results of the preliminary experiment, I then ran a full experiment. This ran for 4 weeks, large volume size. We used 6-liter carboys. And I wanted to obtain as much photooxidized biodegradation as possible, again, with those ideal variables from the preliminary experiment. And what I found from the preliminary experiment was that we were going to use a concentration of 25 ppm and a high energy water acclimated fraction dispersion method, or HEWA for short. And both of the concentration and the dispersion methods that I chose are replicable to the natural environment. And so here is a schematic of my experimental treatments for the full experiment that I ran. There are 5 experimental treatments. All of them had 3 replicates except for our evap treatment. So if I direct your attention to the top left for our ox treatment, that was live seawater with photooxidized oil. The top right is our no-ox treatment, which was live seawater and just evaporated crude oil. Our bottom left treatment was autoclave seawater and photooxidized oil. Our evap treatment was autoclave seawater and evaporated crude oil. And then our no oil treatment was just our live seawater with no oil added whatsoever. And what The seawater that we used for our treatments were just collected off the Bigelow dock at high tide, and it was 5 micron filtered to try to obtain only the bacterial population. And so here's just some images of some methods that I ran this summer. Up at the top left, that is the solar simulator, which is what was used to prepare our photo-oxidized oil. So we actually took Deepwater Horizon source oil and spread it onto a thin film, which was then placed into the solar simulator for 24 hours in conditions that replicated natural sunlight in the environment. So 24 hours in the solar simulator equates to about 2 to 3 days in the natural environment. And then in the bottom left, you can see this is how we prepared our HEWAF. So we added oil and seawater into the blender at a ratio of about 0.5 gram of oil for 1 liter of seawater. And it was blended for 30 seconds on low and then added to a separatory funnel where it was allowed to separate for 1 hour. And then the extract was then diluted to the necessary concentration. Up at the top right, you can see an image of half of our experimental treatments. We added a stir bar and they were allowed to stir for the course of the experiment. And then at the bottom right, we have our sampling setup. So this experiment ran for 1 month, but for the first 2 weeks, this is what I was doing. I would take my experimental treatments down to the laminar flow hood in order to preserve the microbial community. And I would use a vacuum pump to remove 1 liter of seawater, which was then subsampled into different containers to preserve for later analysis. So for my project, I looked at TLC FID, which quantified the bulk fractions of the oil. We looked at saturated, aromatic, and oxygenated hydrocarbon fractions. We did a GCMS analysis, which specifically quantified the N-alkanes and the PAHs. And then we also did flow cytometry, which looked at bacterial concentrations over time for all of our replicates. So here are some TLC FID results that looks at the chemical difference between our evaporated crude oil and our 24-hour irradiated oil. So our TLC looks at 4 fractions, but for the purposes of what we're looking at here, our oxygenated hydrocarbon fractions were pooled. And so the saturated and aromatic, you can kind of equate to that leftmost column on the slide that I introduced earlier. And the OxHC fraction is kind of those photo-oxidized oxygenated compounds on the rightmost side. And so what you can notice is there is a much higher percentage of OxHC fractions in the 24-hour irradiated oil compared to the evaporated crude oil, which means that 24 hours was sufficient enough to produce an oil that was chemically different so that we were actually having differences in our oil for our experimental treatments. And so now I'm going to get into some of the results from our main experiment. First, I'm going to look at flow cytometry. So I'll direct your attention to the leftmost graph, which looks at absolute bacterial concentrations on a log scale for all 5 of our treatments. So on our y-axis, we have total bacterial concentration in cells per milliliter. And then on the x-axis, we have our 5 treatments: no oil, ox, control, no ox, and evap. And a blue bar is initial time point, red is 1 week, and green is 2 weeks. And so for our 3 live treatments, they kind of hover around 1 million. But what's important to notice from this graph is our 2 sterile treatments, our control treatment and our evap treatment, which had bacterial concentrations of less than 10,000 cells, which indicates that these treatments stayed sterile for the course of the experiment, as counts below 10,000 are usually not statistically significant enough to indicate the presence of bacteria. Now I want to direct your attention to the right, which looks at normalized bacterial concentrations. So for our 3 live treatments, we normalized our concentrations to time 0. And these 3 treatments showed generally the same pattern. You see an increase in bacterial concentration at time 1, and then a decrease at time 2. But what's important to notice is that we actually see a larger increase in bacterial concentration for our 2 treatments that contained oil, which therefore led to a higher concentration of bacteria at time 2 for those treatments as well. And so now we'll look at some of the TLC FID results, which again quantifies the concentration of the saturated, aromatic, and OxHC fractions for our 4 treatments. So we have our ox, no ox, control, and evap. And again, time 0 is initial time point is blue, then 1 week is red, and 2 weeks is green. And so what's important to notice here is I want to focus your attention on our OxHC fraction, specifically for our no-ox group. So if we were to suspect that there would be biodegradation, we should see a decrease in the concentration of OxHC over time. But what we actually see is an increase. which is especially when compared to our sterile evap treatment, which shows a decrease in OX-HC fractions over time. And this same pattern is seen in our OX treatment as well, but not to the same degree. And so in order to do kind of a further analysis into the saturated and aromatic fractions, we did a GCMS analysis. So these are some results from our GCMS. We quantified both N-alkanes, which is the leftmost side of every graph, and then PAHs, which are the rightmost side of every graph. And our N-alkanes were broken up by concentration into carbon chain length 10 to 19, which is green, 20 to 29, which is red, and then 30 plus, which is blue. And then PAHs were also quantified by how many rings they contained. So the purple bars are 2-ring PAHs, and then blue is 3-ring, and yellow is 4-ring. And again, this is all Hoppe normalized concentrations of these fractions. And kind of the most obvious pattern that we see is with our N-alkanes for both our OX and our no-OX treatment. So over the course of the experiment at time 2, we saw a drastic decrease in the concentration of N-alkanes that was not seen in either of our sterile controls, which indicates the presence of biodegradation. The PAHs were not such a simple story. We didn't necessarily see any evidence of biodegradation except for perhaps the 2-ring PAHs in our no-OX treatment. But given the fact that the concentration of PAHs for all of our other treatments either stayed consistent or increased over time, we cannot conclude that there was any biodegradation happening. And just so some conclusions of what we found, try to put all together, both oil types led to similar bacterial growth and they can be biodegraded. We found that saturated compounds degrade more than aromatic compounds for both oil types. And we also found that that oxygenated fraction, which contains the photoproducts, increases, which we think could due to the potential accumulation of oxygenated biodegradation products. And so I would just like to thank everyone who helped me, the Apley Lab, Chris, Brian, and Aaron. I couldn't have done it without you. I'd like to thank Laura and the Center for Aquatic Cytometry for running all the flow cytometry samples and getting the data turned around so quickly. I'd like to thank Dr. Cameron Thrash at the University of Southern California for helping a bunch of environmental chemists do a microbiology experiment. I'd like to thank the Countway Lab for their generous use of their cart and other equipment that made this experiment run. And I'd like to thank Liz for being my Apley Lab girlfriend. And I'd like to thank all the other REU interns for their help throughout the summer. And I'd like to thank the GOF Research Program and NSF for funding this project. And I will happily take any questions.
Hi, that was amazing. I have a question. I want to know how representative your experiment was to the actual Deepwater Horizon area. Like, did you have the same type of bacteria?
Yeah, so we think that there's hydrocarbon degrading bacteria all over the globe. So the specific species and the specific bacterial concentrations might not be the same as what's in the Gulf of Mexico, but through enrichment with this type of oil, we actually think that we're going to increase the presence of those hydrocarbon degrading bacteria so that if we were to run this experiment with water from anywhere, by the end of the experiment, we should expect generally the same type of microbial community by enriching it with that type of oil.
So you showed in your graphs that I think like the small alkane chains were biodegradated, but the large like PAHs weren't. Do you know why? Does it have something to do with like the steric hindrance or the aromaticity of the PAHs?
Yeah, it's probably a combination of both of those. It's been tested in previous research that the N-alkanes are greatly more readily biodegraded than the PAHs. But the PAHs can actually be biodegraded. So if we were to run this experiment for longer, which we do have another time point that we're looking at, we should expect that the PAHs should get degraded once the N-alkanes have been depleted sufficiently. So it's kind of like food choice preference. They like the N-alkanes more than the PAHs, but the PAHs can still be biodegraded.
Cool. Thank you. Great presentation.
Thanks.
So if I understand correctly, the oxygenated hydrocarbons don't get biodegraded as easily as the non-oxygenated ones. So my question to you is, if there was an oil spill, is it better for it to be a cloudy day or a sunny day in terms of biodegradation? Or does it matter?
So ideally, you would like to get to the oil spill. The bacteria would like to get to the oil spill before it becomes photooxidized, just because we're not really sure how persistent these oxygenated hydrocarbons could be. So if we were to start off with a lower concentration of them in the first place, even if some might be formed as like a microbial byproduct, their concentration would be lower over time. But realistically, for an oil spill, like the photooxidation happens so quickly in the field that you wouldn't be able to mitigate it fast enough, even with enriching with a lot of like hydrocarbon degrading bacteria. You wouldn't necessarily, it's not necessarily realistic. So we kind of have to figure out how these photooxidized compounds can get biodegraded, because we know for a fact that they're going to be there.
Thank you.
Hello, everybody. My name is Sam McNeely, and my name is Sam McNeely, and I'm a senior at the University of North Carolina, Wilmington, studying marine biology. And to continue on the same vein of oil research, I worked on a project with Maura Niemisto, Dr. Christoph Apley, and Dr. David Fields entitled Greasy Grub, Copepod Ingestion Rate at Sublethal Oil Concentrations. And so a recent survey estimated that there's over 90 billion barrels, or nearly 4 trillion gallons of undiscovered oil in the Arctic Circle. And as Arctic sea ice continues to recede due to climate change, this exploitation of these reserves will only increase, as will transportation across the Arctic, both of which increase the likelihood for an oil spill to occur. And so a common cleanup method from oil spills is to use dispersants, which are a cleaning agent that separates the oil slick at the surface and sends the oil deeper into the water column. And this can be beneficial for megafauna so that they don't get stuck in the oil slick at the surface, but it's actually harmful to macrofauna like copepods. And so Arctic copepods, which vary in size between about 2 and 5 millimeters in length, are the foundation of the Arctic food web. They eat phytoplankton and then are eaten by the succeeding trophic levels like fish larvae and even whales. And previous studies have seen that copepods have ingested dispersed oil, which is shown here in these pictures in bright blue fluorescence. And that indicates that the oil can biomagnify. So the dispersed oil can actually provide or be harmful in the long run to the megafauna that we were originally trying to protect with the dispersants. And because copepods are known to ingest oil, they experience both lethal and sublethal effects from exposure. And some of the sublethal effects include decreases in egg production rate, egg hatching percentage, and fecal pellet production rate, which all suggest that there's some sort of change going on in their ingestion rate. And so the purpose of my project is to investigate the ingestion rates in a common North Atlantic copepod, Calanus marchicus, in the presence of sublethal crude oil and dispersant concentrations. And my hypothesis is that copepod ingestion rates of phytoplankton will decrease in the presence of these pollutants. And so before I began any of my experiments, I created my mixtures. And these emulsions were stirred for 18 hours at room temperature. And the left jar contains only oil and filtered seawater, which made up the water accommodated fraction, or the WAF. And the middle jar was only dispersants and filtered seawater. Then the far right jar was the combined oil and dispersants with filtered seawater, which made up the chemically enhanced water accommodated fraction, or the CWAF. And these mixtures were extracted by draining the jars about 3/4 of the way to only get the soluble compounds from these mixtures and leaving the oil slick or the larger oil particles away from the extraction. And the extractions were then stored at 12 degrees Celsius. And so then moving on to my experimental design, I had a few preliminary experiments to gather some background data to answer my primary question. And the first experiments I wanted to run were looking at copepod ingestion rate of a centric diatom, Thalassiocyra westflogii, to determine the minimum concentration that produces the maximum ingestion rate. And the second experiment looked at or tested copepod survival at various concentrations of the combined effects from oil and dispersants in the seawater to determine my sublethal concentration. And then third, I looked at how the algae responded to the oil-only WAF, the dispersant treatment, and the oil and dispersant seawater. And then all of this data was used to set the conditions for my primary experiment that examined copepod ingestion rate under pollutant exposure. And so for all my experiments, I maintained the same experimental conditions of using 250 ml jars at 12 degrees Celsius for 48 hours and in the dark to limit algal growth. And any experiments that had copepods in it had 10 copepods per jar. And then all experiments with algae were run on this machine here, which is a plankton wheel to keep the algae in suspension. And then so now to look at those preliminary experiments, I wanted to find the ideal concentration to use. And so copepods were fed at various concentrations of Thalassiocyra westflogii, which is noted as the average cell concentration on the x-axis, and then ingestion rate is cells eaten per copepod per hour on the y-axis. And so based on this logarithmically fitted curve, I decided that 20,000 cells per milliliter, which is this X right here, I used that as my optimal concentration to determine if pollutant exposure actually decreased the ingestion rate. And so then I wanted to look at copepod resiliency. And so survival was tested on a concentration gradient of the combined effect from the oil and dispersant seawater. And so the first number on the x-axis represents the microliters of oil per liter of filtered seawater. And then the second number represents microliters of dispersant per liter of filtered seawater. And based on this data, and the y-axis is copepod survival as a percentage. And then based on this data, we found that Calanus finmarchicus as a species is very hardy, because even at the ecologically extreme highest concentration, which is this dark blue bar, there was no difference detected. And even because it's an ecologically extreme concentration, there was no significant die-off. And so that's what I used as my sublethal concentration. And then my last preliminary experiment looked at how algal growth was impacted. And so I put algae into a jar with the different pollutants, which are on the x-axis as the control, the oil-only WAF, the dispersant, and then the combined oil dispersant CWAF. And then on the y-axis is the algal growth constant, which was calculated by the Frost equations up in the left-hand corner. And there was no difference detected between any of these treatments, saying that algal growth did not need to be accounted for as a confounding factor in my primary experiment, which, given all this background data, I was able to start looking at whether ingestion rate did change under oil and dispersant exposure. And so for the control, only copepods and algae were put into a jar without oil or dispersants. And then for the WAF treatment, only oil was added. Then the dispersant treatment, only dispersants were added. And then for the CWAF treatment, it was the combining effect of both oil and dispersants. And all of these were run with 6 replicates, and then algal concentrations were measured by a MOXIE cell counter at the start and at the end of the experiment. And all of this data was statistically analyzed with an ANOVA test. And so results from this experiment are here with the x-axis categorically grouped as the control, the oil-only WAF, the dispersant treatment, and then the oil dispersant CWAF. And then ingestion rate is again plotted on the y-axis as cells eaten per copepod per hour. And so based on the data that was collected, there was no significant difference between any of the treatments. And so my conclusions for my project were, first of all, Calanus fumarchicus did not die off, even at the ecologically high oil and dispersant concentrations. And this just shows how tough Calanus fumarchicus is as a species, which is very encouraging. And then another conclusion was that Thalassiocyra westflogii was not impacted by oil and dispersant exposure, or their growth wasn't. And so this is encouraging as a food source for these copepods. And although it sounds encouraging that feeding rates did not change, there's the factor that maybe algae is acting as a vector for oil and dispersants to enter the copepods' bodies. And that leads into the future of this project, which we are planning on running a gas chromatography or GC analysis to determine if the copepods are actually ingesting algae with oil and dispersants attached to that algae, which could then biomagnify and accumulate in their predators, which is something I'm also interested in, is looking at how much of a pollutant is bioaccumulated in predators of copepods that have been exposed to certain pollutants. And so with that, I'd like to thank you for your time and listening to my presentation. And I'd like to thank the National Science Foundation for funding this project. And special thanks to my awesome mentors, Dr. David Fields, Dr. Christoph Apley, and Maureen Niemisto, as well as Hannah Sterling, Liz Westbrook, and Aaron Byrne for their help on the chemistry side of my project, which I don't understand quite as well. And then the Fields Lab, of course, all the staff at Bigelow, and my home institution, UNCW, and of course, my family for all of their love and support throughout my career.
Hello, my name is Mansi Desai. I'm a senior at the College of Worcester in Ohio, and here at Bigelow, I am working with Dr. David Fields, Dr. Christoph Apley, and Maura Niemisto on my project, Microplastics as Carriers of Polycyclic Aromatic Hydrocarbons, or PAHs, that are released from oil spills. I measured ingestion rates and bioaccumulation of these PAHs in copepods. So oil spills are a serious threat to the marine environment. As Hannah previously stated, on April 20th, 2010, the Deepwater Horizon oil rig in the Gulf of Mexico exploded with a total discharge of around 4.1 million barrels of oil spilled in the marine environment. Here's a picture of the oil spill. This is not an isolated event. Around 1.3 million gallons of oil are spilled in the US waters in a typical year. So when crude oil comes into contact with water, it partitions into hydrophobic and hydrophilic components. Out of these hydrophobic components, PAHs predominate, and they're found to be acutely toxic and carcinogenic to marine species. Here's a diagram of some common PAHs. They are organic compounds consisting of 2 or more fused aromatic rings. Another growing concern in the marine environment are microplastics. They've been detected in the marine environment due to a large influx of plastic waste being dumped from land. Around 4.8, 12.7 million metric tons of plastic waste is being dumped into the ocean. They're less than 5 millimeters in size, and they're made from a variety of polymers, such as polyethylene, polystyrene, etc. So what's the connection between these two concerns? Well, due to their nonpolar surface and high adsorption coefficients, microplastics can act as pretty good sorbents to contaminants. So as you can see in this diagram here, these pHs can actually sorb onto plastic marine debris. So essentially means that the molecules of the pHs and the molecules of the microplastics stick together and travel around the ocean like the best friends that they are. So what are the implications to the marine ecosystem then? Well, you have these tiny critters called copepods as small aquatic crustaceans, and they eat almost anything and everything that's within their food size spectra. So unfortunately, microplastics are within the copepods' food size spectra. Copepods play an important role in the marine food web. They serve as a link between primary producers and larger fish, marine mammals, etc. So if you have these copepods ingesting microplastics or even contaminated microplastics, these contaminants would then become bioavailable and possibly biomagnify up the trophic level. Copepods, their fecal pellets also serve as important sources of nutrients to the deep water ecosystems, so that fecal pellets could actually export microplastics and contaminants to the deep water ecosystem. So my research question is essentially asking if microplastics provide an ecologically important route for pHs to enter the food web through ingestions by zooplankton. So we broke this broad question down into 3 sub-questions. We're first asking if copepods ingest microplastics, indicated by the black dots here, and microplastics plus pHs, if copepods ingest them as well, which are indicated by the orange dots here. Our second question is then asking what's the body load of these pHs in copepods, if at all they ingest pH-loaded microplastics. Again, this has implications in the marine food web because these contaminants become bioavailable and can biomagnify. That is an increase in concentration. Our third question is asking if their fecal pellets contain pH-loaded microplastics. So here's a blown up picture of the fecal pellet, and the tiny spherical dots you can see are actually the microplastics that we found. So fecal pellets again they can act as a source for microplastics and contaminants to the deep water ecosystem and can act as a sink for these contaminants as well. So for this experiment, we chose 3 model specimens. We chose the polystyrene microbeads that are around 12 microns in diameter. We chose Calanus phenomarchicus as our model copepod, which is about the size of a small grain of rice. And we chose phenanthrene as our model PAH, which is a 14 carbon ring organic structure. So we first wanted to know how much of the beads can actually adsorb this phenanthrene. So we first made a bunch of dilutions of a phenanthrene stock solution from 50 microgram per liter to 1000 microgram per liter. And we ran it through a fluorometer to get this fluorometer curve. So on the y-axis, you can see the counts per second, and on the x-axis, you can see the wavelength in nanometer. So these 3 distinct peaks are indicative of phenanthrene. So we chose the values at around 345 nanometers, these values at this wavelength, and we plotted that against the concentrations of phenanthrene. So in this graph, you can see the emission values at 345 nanometers on the y-axis and the concentration of phenanthrene in microgram per liter on the x-axis. And we got a calibration curve. As you can see, we got a pretty good fit. So we chose 500 microgram per liters of phenanthrene, and we added that to around 10 to the 4th beads per mil of the polystyrene beads. And we let that adsorb for 4 hours, for at least 4 hours. So then what I did was I calculated the drop in background concentration of phenanthrene. So in this graph, you can see the concentration of phenanthrene in microgram per liter on the y-axis and the time on the x-axis. And you can see a pretty steady drop in around 4 hours to 16 hours. And so we calculated the concentration of phenanthrene disappeared, divided over the total number of beads to get a load on the bead. So essentially, these beads just sucked up all the phenanthrene. And we got a value of 26 picograms per bead. So then to answer our first question, if they ingest microplastics and pH-loaded microplastics, our experimental design consisted of two treatments, a beads only and a beads loaded with 500 microgram per liter of phenanthrene. We had 25 ml jars and we put 5 Copepods in them. We had 5 replicates of each treatment. We let them ingest for around 23 hours and we put them at a controlled temperature of 12 degrees Celsius. We fed an average bead concentration of 9 times 10 to the third beads per mil. Our control was beads only, and the bead measurement was calculated using a Coulter counter called the Orflomoxy, and the Frost equation was used to calculate the average ingestion rate. So what did we find? In this graph, we have the average ingestion rate on the y-axis, the beads consumed per copepod per day, and the treatments on the x-axis. The beads only, they consumed around 2.67 times 10 to the fourth beads, and the beads with phenanthrene, we saw they consumed 1.81 times 10 to the fourth. So Copepods ate significantly lower beads loaded with phenanthrene, of around a 32% decrease in ingestion. But the important thing is that they still ate the beads loaded with phenanthrene. That means they still have potential to accumulate phenanthrene in their bodies and possibly transfer that phenanthrene up the trophic level. So then we took these Copepods from the ingestion experiments and we wanted to extract them to detect their body burden of phenanthrene. So we followed the same extraction methods from Liz and Brian. We added NaOH and hexane to our Copepods and O-terphenyl, OTP, which is a recovery standard. The NaOH is a strong base that would basically break down the Copepods. We sonicated and beaded them just to basically decimate the Copepods. And so we expect that the phenanthrene would partition into the hexane layer. So then we took that hexane layer, we dried it off, and we transferred it to a GC vial for analysis. So we put that through a GCMS, a gas chromatography mass spectrometer that would essentially detect phenanthrene levels in the Copepods. So what we found was in this graph, the phenanthrene concentrations in picograms on the y-axis and the number of Copepods in the x-axis. So we found that as you add more Copepods, the concentration increases. We need a minimum of at least 2 Copepods to detect phenanthrene levels. And we found that individual Copepods can accumulate around 3,196 picograms of phenanthrene. So then we took their fecal pellets from the ingestion treatments. We took pictures of it, calculated their volume. We wanted to get a bead count in them. So we just sonicated them to break apart the fecal pellet, get the beads out in a known volume, and then use the Moxie to figure out a bead count, and we got 300 beads per pellet. And then we took these same fecal pellets and we extracted them using hexane and OTP to see if there's any phenanthrene that was adsorbed on the beads still that was passed down in the fecal pellet. And what we found was that in this graph, you can see the phenanthrene per bead in picograms on the y-axis. And if the Copepod passed everything from its gut, we'd expect to see around 26 picograms per bead. But we actually saw around 0.7 picogram per bead in our fecal pellets, which suggests that the phenanthrene just leaches off the bead. So what are our conclusions? Our first question, do Copepods ingest microplastics and pH-loaded microplastics? We saw that they do. There was a 32% decrease in ingestion, which suggests that the Copepods can reject particles based on the chemical content. Our second question is asking, what is the body load of phenanthrene on these Copepods? And we found that to be around 3,196 picogram per Copepod. A third question then asks if they contain pH loaded, if the fecal pellets contain pH loaded beads. We found that to be around 0.7 picogram per bead. So this suggests the phenanthrene is either in the copepod's body or it leached off the beads and is resolubilized in the surrounding waters. Why is this not true? Yeah. I'd like to thank the US Department of Homeland Security and NSF for supporting me with this project. Special thanks to my mentors, Dr. David Fields, Dr. Krista Vapley, Maura Nemesto, Erin Byrne, as well as my peers, Zaina C., Hannah Sterling, and Liz Westbrook for all their support in this project. And finally, yeah, I would be happy to take any questions now.
Move up.
Thanks, Mansi. Nice talk. So you know how much phenanthrene was on the beads, and you know how many beads they ate.
Yeah.
And you know how much phenanthrene was in the copepods.
Right.
Did you calculate what percentage of what they ate they actually assimilated? Like, do you have a sense if it was like 1% of what they ate, or 100%, or somewhere presumably in the middle?
I am unsure on that. I'd be happy to get back to you on that. But yeah.
I mean, I think you would have said if you had, but it would be cool. Maybe I'll say it a different way. It would be cool if you did, because I wonder like how efficiently that process happens.
Mm-hmm.
That would be really interesting to look at. But thank you. Great talk.
Thank you.
Great talk, Mansi. I was just wondering if you know if copepods have any bacteria living in their gut, and if those bacteria might be utilizing the phenanthrenes.
So your question was if they have bacteria in their gut. I do know that they have an acidic environment in their gut. We're still unsure on whether that acidic environment actually then leaches off the provides an attractive environment for the phenanthrene to leach off from the beads and then get absorbed in the gut. So those are like the follow-up questions that we would look at. I am not sure on the bacteria. David's looking, saying yes, but yeah, they have an acidic environment in the gut.
Really interesting experiment. Do you think that other types of plastic would show similar patterns as the polystyrene?
Yeah, so this polystyrene was used as a model microplastic that could respond like other microplastics. So we found that, I found in previous literature that polyethylene as well acts as a good sorbent to contaminants found in the marine environment. And microplastics as well could act as a proxy for algal populations as well. So one of the questions that Sam also had earlier of algae acting as vectors for these pollutants are some of like the future questions that we're looking at right now.
Great, thank you.
Thank you.
Hello, everyone. My name is Tejas Reddy. I am a rising junior at the University of Georgia. And here at the Bigelow Lab, I am being mentored by Dr. Jose Fernandez-Robledo and Dr. Peter Countway. And I'm collaborating with Dr. Michael Metzger and Rachel Kiersch of the Pacific Northwest Research Institute. And I'm also working with a fellow REU intern, Maria Jose Oriana Rosales, here at the Bigelow Lab. And the name of my project is a survey of bivalve transmissible neoplasia in Maya arenaria on the Casco Bay. So what is bivalve transmissible neoplasia, or BTN? So this is a type of transmissible cancer that infects many species of bivalves. And this is a very unique form of disease transmission, as there's only 3 other species outside of bivalves that actually have transmissible cancers. These species are dogs, Tasmanian devils, and Syrian hamsters. And on these images here, you can actually see what these cancer cells look like in comparison to the normal hemocytes. So in the top right image here, you can see the normal hemocytes are dark and tend to spread out more, while the cancerous hemocytes are white, rounded, and tend to stick out more to the background. And this way this cancer spreads is very unique. So when a clam gets a cancer cell, it proliferates in the hemolymph, or the clam blood. And when this happens, as the clam's filter feeding, some of these cells will go into the water column. Another clam, while it's filter feeding, can take these cells up, and then these cells will go into its hemolymph and they'll proliferate, giving this clam cancer. And so that's how it spreads in the environment. And so why do we even care about BTN? There's 3 main reasons why we care about it: ecologically, economically, and medically. So if I can direct your attention to the bottom right-hand screen here of this graph of the clam landings and values for the past decade or so, if we can see that the metric tons and landings values of clams has decreased, but the values increase, which just goes to show the per capita value of clams has increased in recent years in the state of Maine, and that it's still a very lucrative industry, and that this disease poses a serious threat to the economy. Additionally, clams such as my arenaria serve an important place in the food web for different species as their food for seabirds such as this herring gull. Additionally, they filter large volumes of water and drive lots of nutrient cycling. And lastly, they're studying animal cancers is very important for medicine, as this has the opportunity to lead to breakthroughs and possibly find other avenues for fighting cancer within humans. And so sampling. So we chose 3 sites within the Casco Bay: Gurnet Landing on Harpswell Island, the Dam in Quahog Bay, and Long Cove on Orr's Island. And we sampled each site twice, once in June and once in July. We then took a subsample of each to run qPCRs on them, which I'll talk about in just a second. And so right here is a diagram for a rough outline of my methods. I'll be talking about each one more individually later, but here I just thought I'd give a rough outline to start with right now. So to start with, we first did an eDNA analysis. So eDNA, or environmental DNA, is just DNA from the environment, from all the organisms and cells within that area. So as I told you earlier, the way this cancer spreads is by hopping through the water column and infecting different clams. So we thought we could just take a sample of water, quantify the amount of cancer DNA in there, and then relate that back to the prevalence within the population. The second method we were doing involves extracting the clam hemolymph. We then wanted to do a visual microscopy analysis, which involved taking images of the actual clam hemolymph. From there on, we wanted to run those images through a software called Cell Profiler. This software is supposed to identify the cancer cells, quantify it, and give us measurements on how infected these individual clams are. And lastly, we isolated the cancer cells from each individual clam, extracted the DNA, and ran 2 qPCRs on every clam's hemolymph, one for the normal hemocyte and one for the cancerous hemocyte, to then determine degree of infection amongst each individual clam. And so right here, you can see an image of my setup for how I withdraw the hemolymph. I set up the clams in rows of 10. And to the right, you can see a picture of me here actually withdrawing the hemolymph from a clam from the posterior adductor muscle. And so just to talk a little more about the histological analysis, why are we even doing it? So when most papers are identifying this cancer via pathohistology, it's a person looking through a microscope and individually counting every single cell. This can be tedious and is very subject to human error. We were hoping that by using this software, we could standardize this method and make this more accurate and accessible to different people, as the software is also free. Secondly, the eDNA detection of cancer cells. So as I said earlier, we took a sample of water from the environment of the clam population. We then put these through a suction filter on a 2-micron soup pore filter, and now we have the cells and the DNA. We then extracted this DNA, and then we're running a neoplastic or cancer cell qPCR to quantify that cancer DNA to then relate that back to prevalence within the population. And so how do we even detect cancer via qPCR? And so to really explain this, I have to explain what qPCR is, or quantitative polymerase chain reaction. So the way this works is that in a qPCR, for every cycle that happens, you're doubling your target DNA. And there's a dye called cyber green that binds to this double-stranded DNA. And so the more initial DNA you have, the fewer cycles it takes to see a brighter fluorescence from this dye, or CQ value. So if I had a heavily infected clam, it would take very few cycles or CQ value to see that fluorescence, and then I can relate that back to a standard curve we made to then get the number of cancer copies per microliter within that actual clam. Similarly, with a mediumly infected clam, it would have a medium CQ value, and then we relate that back to the standard curve, get a medium, and get the DNA concentration. And a normal clam will have no cancerous amplification, and we wouldn't even have to relate that back to the standard curve. And so here are some results from this experiment. And so if I can direct you to this graph of site and date versus BTN prevalence, we found that Gurnet Landing on each month had the highest amount of cancer using both sensitivity thresholds that we use. And this is really important because the last published data on BTN within the state of Maine was 10 years ago. So just this data alone shows that this cancer is still in Maine, still prominent. And within Gurnet Landing in July, as you can see, it got up to near 80% using our highest threshold, which is a lot, which is very high. And additionally, we found that one thing really interesting was that Long Cove had 0% prevalence of BTN in June using the high sensitivity threshold, which is very different from any of the other sites. And here are some results from the cell profiler. So in the top images here, you can see cell profiler correctly identifying the neoplastic or cancer cells of the heavily infected individual. So the top left image is an actual picture of clam hemolymph, and those white dots are cancer cells. And to the top right, you can see cell profiler identifying these cells correctly, quantifying them in a very rapid way than a human ever could. And then the bottom images here is actually, the bottom left one is a non-infective clam. But when withdrawing the hemolymph, if you touch different muscle areas, you're subject to get different debris and tissue matter within your withdrawal. And that's what happened in this non-infected individual. And cell profiler quantified those tiny little muscle fragments as cells. And so that produced a false positive. And so we found that cell profiler is great in identifying cancer and quantifying it within high to mid-infected individuals, but it suffers a little in the low infected ones with cellular debris. But we think by using nuclear staining next time, we can create a new filter almost for cell profiler to look and identify these cells in a better way. Okay, and then here are the results from the eDNA analysis. And so it's, the y-axis is eDNA copies per milliliter, and the x-axis is site and date. And as you can see, we were able to detect cancer cells in the environment. This has actually never been done before. One of our collaborators has been able to do this using a closed tank aquarium system with some cancerous clams, but in the natural environment, this hasn't been done. And as you can see, the July samples had significant more amount of cancer cells within the environment. And we think this has to do with the suspected seasonality of this disease, as it increases the most in late summer, early fall. And so there might be an onset of proliferation of these cancer cells within the water column in July. And so here's some conclusions. Cell profiler is successful in identifying cancer cells in highly neoplastic clams, and we just think using nuclear staining, we can produce more accurate identification of these cells within low infected individuals. And then Gurnet Landing has the highest prevalence of BTN, and it's very high in the July sample, near 80%, which is significantly higher than the last published data of 10 years ago of BTN in the state of Maine. eDNA can be used to detect BTN within the water column, and this has the possibility to save time and money in regards to sampling these clams, as normally withdrawing the hemolymph, running the qPCRs is very time intensive. Using eDNA as an proxy for prevalence within the population has the definite capabilities to both save this time and money. And lastly, the July eDNA samples had significant increase in concentration than those of June, and we think this has to do with the seasonality of the disease. And so here are some future directions. So first, we would like to investigate how and why these cancerous clams expel more neoplastic hemocytes, because it's not really understood why these clams are, putting more hemocytes into the environment. If it's temperature, it's salinity, it's just unknown. Next, we would like to examine whether there's tidal zonation or phenotypic preferences in cancer prevalence, so that maybe clam diggers will know, let's get rid of these clams because they have the highest risks to the population of disseminating this high mortality cancer. Third, we would like to explore how increased temperatures and decreased pH affect BTN prevalence, because as you know, the ocean's changing and we don't know how this will even affect the cancer in the environment. And lastly, we would like to do more work with Cell Profiler to get more accurate image analysis on these images with tissue debris. And now I'd like to acknowledge the NSF REU for supporting me during this project and other NSF grants that my mentors Pete and Jose have. I'd like to acknowledge the clam digger, David Wilson, for collecting samples for us. I would also like to acknowledge Sydney Greenlee and Dr. Robin Sleeth for assisting me in lab work and helping answer lots and lots of questions that I had. And lastly, I'd like to acknowledge the fellow 2021 REU interns for their support.
Hello. Okay. Going back to your results, what was the difference between the lowest and highest sensitivity threshold?
Let me just go back to that chart. Okay, so the highest threshold was actually determined by our standard curve. And so if we look at the standard curve, our lowest concentration for detecting the DNA, detecting cancer, the last point was 6.23 copies per microliter of cancerous DNA. So that was considered the minimum amount of cancerous DNA needed to have to say you have cancer for the highest threshold. For the lowest threshold, we decided that if you had cancerous DNA between 6.23 copies per microliter or 0, and if you amplified there, then you consider under the lowest threshold. Because there's a little debate, like if you have one cancer cell that produces 1 copy per microliter, are you really considered cancerous? And so that's basically the justification for using both thresholds.
Gotcha. Thanks, Rumi. Good job.
Thanks. That was a lot of great new results you had there. I was wondering, with your LC location, I forgot what that stood for. You saw almost no cancer cells in June, but it seemed like the highest amount of cancer in the water. So I was just wondering if you had any comments about how fast maybe the onset of the cancer could be, or maybe I am remembering those graphs backwards.
So Long Cove in June had a 0% cancer using the highest threshold, but in July it actually had a pretty significant cancer increase. So that kind of aligns with the trend you see in the eDNA of it being low to high. And I'm not really sure why necessarily it has the most cancer copies in the water, even though it didn't have the highest cancer prevalence. But we only had 6 data points to really compare prevalence to eDNA, so it was really hard to get a significant trend regarding that.
Great talk. I think you may have just sort of alluded to this question I was about to ask, but were you able to relate your eDNA copy number to your prevalence of cancer or your mortality?
We were not able to do that just because, as I said, we only had 6 data points. And so it was really hard to get a significant trend. We did see a trend though, but we just needed more data points to solidify it.
Last question.
Hi, great talk. I was wondering, with the algorithm that you were using to identify cancerous and non-cancerous cells, how do you know in the heavily infected individual that you weren't also getting false positives in that, or were you, and you were able to account for that?
When you have such a heavily infected individual, like what I showed you there, what was a very diluted sample of the cancer cells, because in these heavily infected individuals, typically you're seeing like the cells are so dense and so prolific that no matter if you have a little tissue in there, the difference you have compared to the cells is so little. And so it wouldn't really matter for these heavily infected individuals if there was even a little tissue in there.
Thank you.
Thank you. Start. And there we go.
Hi, my name is Asher Platts. I just graduated with an Associate's in Applied Science from Southern Maine Community College's Department of Marine Science. I've been working here at Bigelow in Dr. Orchid's lab. We initially set out to test heavy metal tolerances of deep sea microbial communities in relation to deep sea mining, but as we worked on it, our focus ended up shifting. This image you see over here is of the abyssal plain with potato-sized polymetallic nodules. This is about 3.5 miles below the surface of the ocean. And it's home to a poorly understood microbial community that would be directly impacted by deep sea mining, because deep sea mining is, they're after those nodules. Okay, that didn't work. I can't advance the slide. Here we go. The sediment that we started working with comes from the Clarion-Clipperton zone. It's located on the abyssal plain between Baja California and Hawaii, in between the Clarion fracture zone and the Clipperton fracture zone. And like all international waters, mining on the seabed here is regulated by the UN-chartered International Seabed Authority, or the ISA. And the ISA wants to make sure that any mining activity doesn't impart irreparable harm. And since, they recommend using ATP to track microbial community health. So why we're after them, the polymetallic nodules contain rare earth minerals that we need for green tech, like laptops, batteries for electric vehicles, and they have relatively low overburden compared with land-based mining. But they are also home to that poorly understood biological community. So we, well, why do we care about the microbes? They provide all kinds of vital ecological services. Whenever we think about nutrient cycles and food webs, microbes are there every step of the way. In the roots of plants, in our own gut microbiome, in the soil, they drive global nutrient cycles. And deep sea mining produces plumes of heavy metal laden sediment. So we want to know how tolerant of heavy metals produced by that mining are these deep sea microbial communities? And can we rapidly monitor microbial health using the Biothema ATP biomass kit? So we wanted to measure, using this kit, cellular ATP production, but the sediment is a complex environment, so we wanted to know if the readings were accurate doing just preliminary tests. One of our preliminary tests was just adding known quantities of ATP to the sediment to see if we were able to get out what we were putting in, and we were not. So the Biothema kit uses luciferase as a reagent and produces light when it comes in contact with ATP. So you see on the graph over there, we have, oh, be over here. This isn't working. Over here, we have the amount of light produced, and over here you have lower to increasing amounts of ATP added. So at this point, the project sort of bifurcated into two parallel projects. First, we needed to develop a method that would improve the ATP recapture from the sediment. And second, we moved to a model organism so that we could look at heavy metal toxicity and establish whether or not ATP could be used as a proxy for biomass. Part of the problem that we were running into with ATP recapture was there was a background signal from microbes living in the sediment. So to remove that signal, we used two different treatments, autoclave and desiccation. So we dried it out and then treated it with ultraviolet light to sterilize it. so that we did that to try to isolate any sort of chemical interference with sediment that might have been happening. We also changed the ATP extraction method from the kit. We began diluting 1 milliliter of sediment 9:1 with phosphate buffer saline, or PBS, vortexing that to resuspend and make sure that the sediment was fully coated by it, and then added our extractant to prevent so the PBS prevents the ATP from binding to the sediment, which is what we think was happening. So after that, we added the extractant, vortexed again, and then centrifuged the sediment out and transferred the supernatant. From that point on, we followed the biothema kit protocol as directed. So this thing here shows, these are two different versions of the same graph. The one on the right has live sediment removed so that you can better see the differences between the different treatments. These, of course, only have 3 sample points, so it's easy to get an R² value of 1, but so these are all straight lines, but all different slopes. So it's an improvement over what we got initially, but it's not perfection, and I think a lot more work needs to be done with that. We also see differences, and particularly with the Clarion-Clipperton zone sediment, there's still kind of big differences in the lower range, which is the range we were interested in working in. So that remains a problem, but anyway. So the second track that we followed was testing a model organism, Schuonella lodohica, and looking for the relationship between a better known proxy for biomass, optical density, and ATP production. We used a spectrophotometer set to 600 nanometers to measure the absorbency of light by cells in culture. So the more cells, the more light they absorb, the higher the biomass. We first ran a large growth curve with many different treatments to find our toxicity ranges. And that graph is here. This is our first 32-hour growth curve. And we found that at 10 millimolar cupric sulfate, growth was inhibited almost entirely. So we focused in on these ranges here, the highest dose that's close to the effective control, and the 1 millimolar cupric sulfate. Our first test was done in duplicate only because we kept running out of ATP reagent. So on the left, we have an optical density curve with light absorbance on the vertical axis and time on the horizontal axis. And as one would expect, the less toxic treatment is growing less. Oh, sorry, the less toxic treatment is growing more and producing more cells compared to the higher toxic treatment. On the right-hand side, we see that ATP production is spiking prior to the exponential phase and then dips later on when the biomass is actually high, which begins to kind of tell the story that maybe ATP is not a great proxy for biomass. There's also that weird outlier. So we ran the test again in triplicate and focused in on the 10 to 24-hour time gap where the exponential phase was beginning. And we see here the optical density for the 10 to 24-hour period. The bottom line, as expected, the 1 millimolar cupric sulfate treatment is in orange. Above that is the 0.1 millimolar cupric sulfate in blue. It looks just like you'd expect. But then looking at ATP, our ATP measurements were actually higher in the more toxic, lower biomass sample. So our conclusions so far, we were able to improve ATP recovery using phosphate buffer saline. And the inconsistencies that we were seeing were likely due to heterogeneous distribution of microbes in the sediment. Let's see. Based on the model organism study, ATP seems to peak prior to exponential phase, but also in response to toxic stressors. So ATP is not a good quantification for biomass. If ATP is your only measure, very few stress bacteria can end up looking like a lot of thriving bacteria. And since there's not a linear relationship between optical density and ATP, It's not a good yardstick to use. As two big caveats to all of this, the copper concentrations that we needed to produce the toxic effect in our model organism were too high to be relevant to deep sea mining, but also Schwannella loahica is known to be heavy metal tolerant. So we need to run this test more with more species of microbes and also different species of metals. And then as another caveat, the marine broth that we were using is a very nutrient-rich environment, which had a lot of dissolved organic material in it. And the way that copper produces its toxic effect is interacting with organic material. So it may have necessitated a higher dose to produce that toxic effect. I think another study in lower nutrient conditions might yield different toxicity limits, which may be relevant to deep sea mining. I want to say thank you to Beth Orchid, my mentor, and Stephanie Carr, who's in the Orchid Lab, David Fields, the postdocs in the Orchid Lab, who were all extremely helpful when I had no idea what I was doing early on. And Alex Michaud in the Emerson Lab, who suggested we use the phosphate buffer saline. And I want to say thank you to Autumn Pope. I cut the slide with the paper that talked about it, but she referred me to a paper that was helpful for explaining a different point that's not here. And then thank you to Lydia, Sam, and Brianna for coming in with me into the lab at all hours of the night, because when you're working with bacteria, you work on their schedule. And I want to say thank you also to my SNCC faculty and advisors who made me aware of the REU program and encouraged me to apply for this. And of course, thank you to the NSF for funding the REU program. This has been a tremendous experience. Thank you.
So you mentioned some shortcomings of using the S. loahica in terms of like needing high concentrations, and also anyway, I was just wondering why you chose to use that species in particular.
I didn't choose to use it. It was, I think, just what we had. I'm not. Beth says yes.
Cool. Thank you so much. What was a great presentation.
There's a question in the chat. In regards to deep sea mining, what environmental component would ATP be measured from? Crushed rock, sediment, water column? Is there some sort of priority from the deep sea mining community for which microbial community they care about? I'm actually not sure about that. I believe that during the mining, the nodules are crushed and turned into a slurry and then sucked up. And there are, I believe, microbes that live in there. So I believe that those would be important to look at. Certainly sediment as well. Certainly the water column. Yeah. As far as mining, I don't know why. Yeah, I don't know what interest mining has in the microbial community. I feel like, if anything, it might be a hindrance to them kind of doing what they want to do. But
is it possible that it's a time lag problem? So you ran your experiments for 24 hours, and I'm wondering if you would have run them for a week or would have run them for much shorter, if you would have got, or what you would have expected your responses to be, and if they would have been different.
yeah, so a big limiting factor for all of these growth curves was the reagent. We were like constantly running out and then having to order more. I think it, and I don't know, but what it looks like to me from the growth curve of the higher toxic treatment, it may just be like slowed down, and that we would see a similar thing but stretched out over time to the less toxic treatment, but I don't know. And we kept running out of the reagent, so I don't know. Time and supplies were an issue.
I got one more. Temperature. So what temperature did you run these?
Just room temperature.
Room temperature being 20 C.
I don't know, whatever it is in the hallway upstairs.
Yeah. And do you think that would be a very different kind of reaction than one at the 2 degree C temperatures at the bottom?
Yeah, I mean, yeah, I would expect that it would be different.
Yeah. Perfect. All right.
Thank you. Okay. Good afternoon, everyone. My name is Christiana Okafor. I am a rising junior at Bowdoin College in Brunswick, Maine. And this summer, I was working with Dr. McManus on trace metal composition of deep sea sediments and analyzing rare earth elements in the North Pacific. So one of our main questions was, can deep sea sediments supply trace metals to the ocean, specifically rare earth elements in ocean sediments? Seawater chemistry was originally thought to be only dominated by lithogenic material like runoff water carrying things like ash and dust, but now new discoveries are showing that trace metals may be supplied to the ocean from deep sea sediments, which are sediments that are basically beneath the seafloor and diffuse across the seafloor. Metals are very important for many biological and abiological processes. An example of that would be iron. Iron is a micronutrient for many organisms. It's toxic at high levels, but at low levels, that's just what we need. But if you have not enough, you become iron deficient and you have anemia. So that's just one example of one of the metals that we analyzed in this research. So another metal is neodymium. Now, neodymium is not essential for biological processes in our body. However, it is essential for tracing ocean water movements in the ocean. So neodymium has an isotopic ratio that's very interesting. One type of the isotope only travels with lithogenic material, which is material that's coming from land, whereas another isotope, it travels with biogenic material, which means if you have a sample of neodymium and you analyze it, depending on what type of isotope you have, it'll tell you whether it came from land or if it came from the ocean. And that is neodymium on the periodic table. So we studied samples from 3 sites in the North Pacific. So the first one being Station 3, which is the northernmost site closest to Hawaii. And then we have Station 5, which is in the middle, and Station 4, which is the southernmost site and closest to the equator. So we had 2 key hypotheses for this project. The first one is that our 2 extraction techniques that we're using to get these metals from the samples are going to produce similar concentrations, meaning that our 2 acids that we used, hydrochloric acid and dithionite, when we run them through our instruments to tell us how what concentrations of metals are in them, they should produce similar results. And our second hypothesis is that the area that's closest to the area that's going to be richest in iron oxides will have the most extractable neodymium and other trace metals, meaning that whichever station has the most iron, it is also going to have the most neodymium and other trace metals. So we have two
we have three key types of data that we analyze. The first one is pore water data. If you look at the diagram on the right, that is a sediment core. And a sediment core is an instrument that is dropped several hundred meters down into the ocean until it hits the seafloor. And then it's a tube that basically goes beneath the seafloor and collects a few centimeters of sediments. So the sediments that it collects, it's mud. So you see the brownish fluid in there, that is mud. And we perform a few different techniques on it, and we can get the water from it. We can separate the water from the sediments. If you think of like a salad dressing, maybe some sort of vinaigrette or Italian dressing, if you shake it, and you wait for it to separate for over a certain period of time, what you have on the top, that's the oil, that would be our pore water. And what you have at the bottom are the sediments. So that would be like your herbs and everything else that's in your vinaigrette. And then lastly, we have column water data, which is basically just ocean water. So you drop, another type of instrument, or it might actually be the same one. You just drop it several hundred meters down, and instead of letting it go to the sea floor, it just collects the water in the ocean, and that is the water column data. So for analyzing it for this summer, the data that I was analyzing, I analyzed the pore water, which remember is that oil in your vinaigrette, that water, and our solid phase sediments, which is your herbs in your vinaigrette. So our pore water, we put it through a machine called a centrifuge, which basically accelerates that separation process that I talked about earlier. And then that liquid we have on top, we pour that off and we filter it, and then we run it through a machine called an ICP-MS, or an inductively coupled plasma mass spectrometry, which is a machine that basically tells you what the concentrations of metals are in said solution that you put into the machine. So that is how we analyzed our pore water data. And then for the solid phase data, it was a few processes that we went through, but this kind of touches on our hypotheses that our two extraction methods were going to produce similar results. So we basically weighed out samples, we added hydrochloric acid, which is one of the acids, and then dithionite, which is the other acid, and we centrifuged it, we heated it, and did a whole bunch of processes until we were left with a liquid at the top. And that liquid is what we run through the ICP-MS, the machine that analyzes the metals, and it'll tell us the concentrations of metals that are contained in it and how much. So here is part of our results from this experiment. So if you look at the graph that says oxygen percent saturation, that's on the x-axis. That's basically the concentration of oxygen in the pore water. And on your y-axis, wow, okay. On the y-axis, we have, let me hover over. Okay. So on your y-axis, you have depth. So like I said earlier, in centimeters on your y-axis, that's how far the sediment, that's how far a machine that collects the sediments go. And if you look at the graph on the right, Station 3, that is the site that's closest to Hawaii. And that is also the site that has the most oxygen in it. And that is due to the fact that a lot of decomposition does not happen at those sites far from the equator. The way decomposition occurs when things are decomposing, it absorbs oxygen. So at the site that's farthest from the equator, you're going to have the most amount of oxygen because there's not a lot of decomposition occurring. However, Station 4, which is this site here, closest to the equator, and it correlates to this line here on the graph, it's very low oxygen concentrations. And that is due to the fact that a lot of decomposition is occurring there. So a lot of oxygen is being used up by that decomposing material. And that is just pointing out the three sites again. And then finally, well, not finally, but moving forward, decomposing material in these trace metals. Why is this important? So the whole point of determining the oxygen concentrations in our data is to see whether it tells us what type of material are these trace metals traveling with, and how much of it. So in the location here, station three does not have a lot of oxygen. So that tells us that the material there is in a crystalline state, meaning that it's going to be harder to extract. However, station 4, which is closest to the equator, it has it has a very low oxygen concentration because of all the organic matter there, and that organic matter is amorphous, meaning that it's easier to extract. And then here's some more results from our research. So if you look at the graph on the left, that is a graph of the water column data. So that is just the ocean water. That's telling us our concentrations of neodymium. So remember, the metal that's not essential for biological processes, but essential for telling us what type of things are coming into the ocean. And if you see the black dots, that is the, if you look at the gray dots, not the white dots, but the ones that are more gray, that represents station 4. And it's not a lot of neodymium at that location. However, if you look at these other dots, the more white dots that are kind of hard to see, there's a little bit more neodymium there. And now looking at the graph on the right, the main point of these two graphs is to show that there is more neodymium in the pore water, which is the water below the seafloor, than in the water in the ocean. So the main key is to look at this x-axis up here. This goes from 0 to 100. So it means that the most amount of neodymium that's in the ocean is probably around 30 or so. But when you come over to the pore water, which is the water below the seafloor, this graph goes from 0 to 1,500. And it looks like the most that's there is probably somewhere around 150. So this is very significant because, as I mentioned in the beginning, we previously thought that these trace metals were being supplied from sources outside of the ocean. However, from these graphs, we see that there is much more metals in the seafloor. So that means that below the seafloor, so that means that the metals in the seafloor is what is actually a source of the trace metals in the ocean. And then here's just one other graph that's helping us understand more about what type of sediments are in our, what type of metals are in our sediments. So if you look at this trend line, if our two extraction techniques work the same, then all of these dots would fall on this trend line, but it doesn't. And this is telling us that the type of material that our trace metals are being carried with are in different phases. So here is station 4. Station 4 is closest to the equator. And that means that material there, it's more organic and it's amorphous, so you can easily get it. So that's why the ratio between the iron, this is iron here with our HCl extraction, and then iron with our dithionite extraction. And here it's falling closer to the line, but when you move up to station 3, which is our site closest to Hawaii, there's much more iron being extracted with the dithionite. And this is telling us that dithionite is a more aggressive solution to get at the metals that are more in a crystalline phase. Okay, and here's just showing the graph again. And this is important to one of our hypotheses that both of our extraction techniques are going to work the same. As I mentioned in the previous slide, they're not working the same. Our dithionite extraction solution is taking out much more iron, which is telling us that
At some sites, we have metals that are in crystalline form, meaning that they're harder to get at. And in other sites, like in Station 4, where there's more organic material, there is metals there, and it's easier to get at because it's not in crystalline form. So we have a few conclusions from this data. So through our data, we showed that neodymium is in fact in ocean water and in our pore water and in our sediments. However, the important thing about that is that there's more in the pore water and the sediments than in our ocean water. Our northern sites, they have the most amount of extractable metals. So that is very important to this data. And in further research, we plan to analyze these sediments for more metals besides neodymium and iron. So wrapping up, I just want to thank a few people. So I want to thank Brian Haley, who was the chief scientist on the cruise, and he helped us in getting data. I would like to thank Aaron and Sarah for their great assistance in the lab. They were very helpful. Not sure how I would have interpreted those large numbers without them. I want to thank David Fields, my mentor, Dr. Jim McManus, and the entire Bigelow 2021 summer cohort. And I'd also like to thank my professor at Bowdoin for telling me about this program, Professor McKenna. I literally would not be here without you because I would not have known about this program. And I'm so happy you told me. So thank you. Okay, I think I see something in the. Oh, yes. So one of the questions in the chat is, how do I think that neodymium is getting into deep sea sediments? What could the source of deep sea sediment, deep sea neodymium be? So that is an amazing question. So there's a few ways that deep sea neodymium is getting there. One of them is coming from decomposing material. So for example, metals are very sticky elements, which means that they're going to stick to like plants, they're going to stick to different organisms in the ocean. So when organisms die, the ones that are more closer to the surface, and they decompose, that decomposing matter releases metals and different elements, and that is one of the sources of neodymium. Another way is, like I mentioned before, lithogenic material. So our sites are very close to the Asian, well, not very close, it's in the middle of the ocean, but close enough to the Asian continent to where dust can flow into the ocean, and that eventually also will attach to things and it'll sink to the bottom, and it eventually becomes deep sea sediments. Any more questions?
I have a quick question. It's actually kind of related to that. First of all, great talk. I really enjoyed hearing about the sediments, but it's kind of the opposite. So I noticed on your water column data, the trend kind of was lower towards the top, but then closer to the sediments. Is that what you kind of just mentioned? Does it kind of accumulate at these lower depths of the ocean, or is the sediment a source kind of coming back up into the water column?
Exactly, it's the latter. So the deep sea sediments, they're diffusing across the sea floor, and that's why the concentrations in the water column, it's higher at deeper depths, but at the top near the surface, it's much lower because the bottom of the ocean, the sea floor is supplying those metals into the ocean, and they don't really make it that. A lot of that does not make it far up. So,
sweet, thank you.
Yeah. Yeah. Any more questions? Oh.
Yeah.
Do we have time? Yeah. Okay. Great.
Was there one up here? Did you have a question? Yep.
So great presentation. When you showed the two different methods for extraction, I think the dithionite you said was extracting more than the other hydrochloric extraction. Could it be like possible other like metals though absorbing on, or are you sure that it's iron?
So if I'm understanding your question correctly, you just want to know why dithionite is better at extracting the metals than hydrochloric? Or are you saying that dithionite may be influencing?
Yeah, I guess I was wondering if there was like any other metals from the sediment that could be like pulled out by the dithionite that were coming through as iron.
Well, the instrument that we're using, when we run it through, it's able to decipher like between iron, neodymium, chromium, nickel, all the different types of elements. So it could be extracting other ones, but it's not going to like be a false positive and like detect something that's iron that's not actually iron.
Cool, thank you.
Yeah.
All right. Hello, everybody. My name is Danny Buckheister. I am an undergraduate student at the University of Colorado Boulder studying ecology and evolutionary biology. And this summer, I've been working with doctors Julia McGonigal and Beth Orcutt, looking at horizontal gene transfer in Lost City hydrothermal field bacteria. And I want to start us off with a question, and that is, where does life get its energy from? When we think about ourselves, we might think of the pizza we just ate for lunch or the air we breathe. When we think of plants, we think of photosynthesis. And sometimes we think of these as the two options, but there is another way. Some microbes are able to use chemicals from rocks around them to make food. And we're going to look at one example of an environment where this happens. We're going to head out to the Atlantic Ocean, all the way out in the middle of it, down about 750 meters beneath the seafloor, maybe, to the Lost City hydrothermal field. And this system is driven by a chemical reaction called serpentinization, which occurs when the crust mineral olivine comes into contact with seawater. And this chemical reaction makes the water hot, it increases the pH of the fluids, and introduces things like methane and hydrogen into the system. And in these fluids, microbes can use the things produced by the reaction to survive. Environments like these are unique and especially interesting since we may find similar ones in other places in our solar system. And they can serve as sources of heat and reducing power for metabolism. So learning about the life in these environments here on Earth can help inform our search for life elsewhere in our universe. As of right now, little is known about the microbes that live in these venting fluids and therefore in the subsurface. So the specific question I want to ask is this: what horizontal gene transfer is occurring in this community? Now, some of you may be asking, what is horizontal gene transfer? And it's the transfer of genes between species rather than from parent to offspring. This can facilitate adaptation and evolution, and oftentimes important genes are transferred. And this can be impacted by environmental conditions. So we know from previous literature that things like heat and anaerobic conditions can increase the incidence of this happening. This is also a topic that's underexplored in the subsurface environment. And so I wanted to look at this, and I expected to find evidence of metabolic genes that would help these microbes survive these relatively harsh conditions. And so to do this, the first thing we needed to do was actually bring some of these microbes up to the surface. And this was a 2018 Atlantis expedition using the remotely operated vehicle Jason to head down to the vents and suck up the venting fluids as they came out of the hydrothermal system. Those were then brought up to the surface where they were separated into single cells using fluorescent cell sorting. So first, those were stained with fluorescent dye, sorted with a flow cytometer, as you see on the right there. And then we ended up with single cells in separate wells. And so that's when we sort of enter the bioinformatic pipeline here. So thanks to Bigelow's unique single cell genomics center, we were able to get single genomes to look at and analyze. And this allows us to know precisely where the genes we're looking at are coming from. From there, we can search for genomic islands. And these are basically pieces of DNA that can move around within and between genomes. and that help facilitate horizontal gene transfer. And oftentimes you see things like integrase and transposase on the ends of those, further indicating that those are genomic islands. So I used a web-based integrated tool set called Island Viewer 4 to pull out the genomic islands from our single-cell genomes. And then from there, I was able to refine those gene annotations as well as identify functional categories with a web-based platform called Acknowledgements. And so just to get a sense of what we're looking at here, I looked at a lot of single-cell bacterial genomes, looked at 99 in total, and out of those, only 5 of them did not have islands. So 94 of those had genomic islands for us to work with. The total number of predicted proteins within those islands was over 6,000, so a lot of data. And when we ran those through Acknowledgements, about 60% of those had database hits. So we were left with about 3,500 genes to therefore categorize and look at. And then within that, we also looked at 3 bacterial groups. So Disulphotomaculum and Thermodisulfovibrio are both species that are known to inhabit environments like this one, hot anaerobic environments. And then we also looked at chloroflexi species, which we further split into 2 groups, high organic and ocean groups. And the reason for this, in addition, we added in 29 chloroflexi reference genomes. The reason for that is that when we mapped out these chloroflexi species, we saw that our genomes from the Lost City sort of grouped into 2 different categories. One in the blue there is the ocean chloroflexi, as we're calling them moving forward here. Those seem to be closely related to just general ocean chloroflexi species. And then we also had in the green the high organic chloroflexes. So our genomes grouped with chloroflexi genomes from other sort of high organic similar environments. And so the first thing I wanted to know was just in general, how much is this or has this occurred in the Lost City? And so I did this by looking at the portion of genomes contained within the genomic islands. So on the x-axis, I have my different Lost City genome groups, as well as my chloroflexi reference groups. On the y-axis, I have the portion of the genome contained within a genomic island. And what we can immediately see is these first 3 groups, Thermodisulfovibrio, Disulphotomaculum, and our high organic chloroflexi, all of which are suspected subsurface species, they all had a higher portion of their genome contained within those genomic islands compared to the chloroflexi ocean species we think are coming in from surrounding seawater, as well as compared to our 2 different reference genome groups. And that really helps indicate that there's something about the environment here that is causing so much of that genome to be located in these genomic islands, something that's making these genes move around. And then from there, I wanted to know what sort of categories these genes might fall into. So on the x-axis here, we have number of genes. On the y-axis are different functional categories. And you can see these are just some of the top ones here. I want to zoom in a little bit on a couple categories, but before I do, I want to point out that the function unknown means that we had some sort of hit to a specific protein, but we don't necessarily know what that protein does. Unknown protein is we had a hit to something, but we don't even know what the protein is. So there are a lot of unknowns, but we have a couple standouts here. The first one I want to point out is this energy production and conversion category. And in this category, we found things like genes involved in the TCA cycle, which helps some of these microbes or could help some of them process organics from the surrounding environment. In the Thermodisulfovibrio genomes, we found quite a few genes, nitrogenase genes involved in nitrogen fixation. And then we also found just some carbon monoxide dehydrogenases, which may help these microbes just get a little bit of supplementary energy from the carbon monoxide around them. So all of these confirm the idea that there were probably going to be metabolic genes being passed around in this system. And then I also want to point out our top group here, the replication, recombination, and repair. And so one, this does help validate that these are indeed genomic islands. You need some of these genes to move around genetic information. But on top of that, about 50% of those were transposases, integrases, and other phage proteins, all of which indicates that there is viral activity down there that may be helping to move around this genetic information in this community. And then finally, I wanted to look at these categories and see what the breakdown was between our different groups. And we can see that here. So again, the same functional categories on the y-axis, as well as a portion of the genes that we found in each category in the genomic islands over the total number of coding regions in their genomes. And already we can see that the thermodisulfovibrio and the high organic chloroflexi seem to, in general, have a higher portion of these functional genes in their coding sequences. And then in addition to that, we also have some standouts. For example, the thermodisulfovibrio in yellow under energy production and conversion, that really associates with those nitrogenase genes we found, really indicating that that group may have some capability to fix nitrogen in this system. So all in all, our findings here were that viruses really seem to be driving bacterial evolution in this Lost City hydrothermal field subsurface environment, especially compared to just the general ocean around that. There's definitely more work to be done and a whole lot of data to dive into. We're hoping to move forward with this and figure out maybe what types of viruses are maybe moving these around, where these genes might be coming from, and things like that. But this is the big takeaway and something that was a little unexpected to find, but really exciting. I wanted to quickly make some acknowledgements here. Just for support during the REU program, I'd like to thank my mentors, Dr. Julie McGonigal and Dr. Beth Orcutt, plus the entire Orcutt lab for being so welcoming, especially with me coming physically here so late in the program. I'd like to thank David and Roxana as well for helping me during my time here. I'd also like to thank the Lost City team. I was not here around in 2018 to head out to the middle of the Atlantic to collect these samples, but I'd like to thank the people who did that so I could be here doing this work. And then I'd also like to thank just others at Bigelow, especially the Single Cell Genomics Center that helped gather this data for me to work with. And then some of our funding sources are here. So thank you all so much for listening, and I will take any questions. I can take one or two questions. Yes.
Thanks. That was a great talk. I was just curious. Were you able to also look at recombination genes within that group of sort of your DNA processing genes? Because I mean, I guess an alternative to viral activity is that there could just be a certain amount of recombination going on between different related chloroflexi.
That could be. That's definitely why I'm careful to say gene movement sometimes rather than definitive evidence for horizontal gene transfer. I did not look for those specifically. I do know that out of that big category, like I said, about 50% of those were sort of transposases, integrases, and other phage-related proteins. So there could definitely be a recombination component to that, but it seems like the viral signal is pretty strong within that data set. And then it looks like I have a question in the chat. Are there other more studied environments where viruses are prevalent that also have bacteria with a high content of genomic islands? Can this help us better understand the hydrothermal vents? I did not look into that in particular. I do think that somebody yesterday during the poster session came and talked about the prevalence of viruses and evidence of viral activity in some like non-oceanic hydrothermal environments. So I wouldn't be surprised at that at all. And it seems just from my background research that these hot anaerobic environments might just be a place where this type of gene movement is common. So yes. Well, thank you all so much.
Okay.
Hi everyone. My name is Grace Berry, and I'm a rising senior at Boston University, studying ecology and conservation biology. And this summer, I've been working with Dr. Julian McGonigal and Dr. Beth Orcutt to determine the diversity of sulfate-reducing microbes and the subsurface fluids of the Lost City hydrothermal vent field. So first, let me introduce you to the Lost City hydrothermal vent field, located about 750 meters below the surface of the ocean on the mid-Atlantic ridge. So you may have heard of hydrothermal vents before and are maybe picturing a system similar to this one on the right, with thick black smoke, fluid temperatures reaching up to 350 degrees Celsius, and an abundance of tube worms, mussels, and other unique macrofauna like yeti crabs. This type of black smoker system is driven entirely by subsurface, and up until about 20 years ago was the only known type of hydrothermal vent field until the Lost City was discovered. So the Lost City is extremely unique because instead of magmatic activity, the energy in this system is generated by subsurface serpentinization reactions, which occur in the ultramafic crust. Serpentinization is a reaction that occurs when olivine rock reacts with seawater, producing methane, hydrogen, heat, and serpentinite. The heat produced by this reaction is around 90 degrees Celsius, which is much cooler than that of a black smoker system, but is still near boiling, so it's pretty hot. Also unique to the Lost City is the extremely alkaline fluid from the vents, whereas the typical black smoker is very acidic. So all of these conditions at the Lost City are able to support microbial life, and much of the biodiversity consists of methane and potentially sulfur cycling microbes. So there have been a handful of expeditions to the Lost City since its discovery, and the unique microbial community has been a major area of interest for research. Genomic studies have shown that the microbial communities inside the chimneys differ from those on the outer chimney surfaces, partly because the insides of the chimneys are continuously bathed in that really extreme serpentinite fluid. However, we aren't really sure what microbes are actually in this fluid and therefore existing in the subsurface of this environment. So for my project, I'm interested in characterizing the microbes collected from the serpentinite fluid in order to get a better understanding of the microbes that are living in the subsurface. Another layer that makes this project unique is that we're able to use a single cell genomic approach to the Lost City microbes, as opposed to a whole population approach, which most previous studies on the Lost City have utilized. So that's going to allow us to get a much more accurate, in-depth picture of the community, which is very cool. So to get our samples in 2018, ROV Jason collected these fluid samples as part of a research cruise. So this photo here is a picture of the fluid vacuum appendage that was specially designed to collect fluid samples from the Lost City. Samples were brought back to Bigelow, where activity-based sorting was done to determine the metabolically active community, and where the single-cell genomes, which are SAGs, were produced in the single-cell genomic center here. Looking at the genomes, protein sequences for each SAG and corresponding functions were determined. So I had two objectives for this project. The first was to determine whether dissimilatory phosphite oxidation exists within the microbial community. And the second was to investigate how the dissimilatory sulfate-reducing community at the Lost City is related to cultured sulfate-reducing microbes found in other environments. So for my first objective, dissimilatory phosphite oxidation, DPO for short, is the biological conversion of soluble phosphate to phosphite, where microbes are actually able to gain energy through this process. The reason we're interested in looking at this is because there's actually only 2 known species of microbes that can undergo DPO. It was pretty recently discovered, but more are thought to exist, just undiscovered or unannotated. So the gene we're specifically interested in is this PDTXD protein gene, which is located here in this chemical pathway of dissimilatory phosphate oxidation. The Lost City environment was a great candidate for potentially finding this pathway because DPO organisms are predicted to be chemolithoautotrophic and exist in low oxygen aquatic environments. So to search for this, I used the reference genes from the known DPO organisms to look for similar genes in our lost city SAGs. Unfortunately, after quite a bit of searching, there were no close matches, so there is no evidence of DPO occurring in the lost city, but that's good to know for future. For my second objective, looking at dissimilatory sulfate reduction, or DSR, is the anaerobic respiration using sulfate, where microbes are able to gain energy from this pathway. DSR is an important component of the global sulfur and carbon cycles. And the genes that we're focused on are in this DSR pathway are located here and consists of subunit alpha and beta. So together, these two subunits work together to create this DSR protein, which is able to facilitate this chemical pathway. So to characterize the DSR community, I searched through the annotations of the lost city SAGs to identify which ones contained the DSR-AB genes. I then pulled these out of NCBI's reference database. I was then able to align the lost city DSR-AB genes to reference genes and use this alignment to construct two phylogenetic trees, one for each subunit, DSR-A and DSR-B. So this is my phylogenetic tree for DSR-A. If you've never seen a phylogenetic tree before, it's just a visual representation of how genetically related each of these organisms are to each other. And so here on this tree, it's a little pixelated up on the big screen, but we see our lost city SAGs in this blue color. We can see our Disulfida maculaceae, Thermodisulfovibrio and gamma proteobacteria grouped in with an assortment of reference sulfate-reducing bacteria from other environments. In this tree, we used Archaeoglobus up here, which is included as an outgroup to anchor our tree because Archaeoglobus is an archaea and all of the other taxa represented on this tree are bacteria. We were interested in seeing if the lost city dissimilatory sulfate-reducing microbes group more with other lost city organisms or more with similar species from other environments. And as we can see here, they largely group with similar species from other environments. As we see the Thermodisulfovibrio grouping with the reference Thermodisulfovibrio and so forth with the Disulfida maculaceae as well. And over on the left, just see the counts of all of these taxa that are represented in these like collapsed clades here. So you get an idea of what we're working with. This is my DSR-B tree. So we can see pretty much the same pattern in this one that we saw in the DSR-A tree, where lost city genes group more with similar species from other environments. And ultimately, this suggests that there were likely multiple evolutionary events within the overall sulfate reducing community, which reflects the life history of sulfate reducers, as we were kind of already aware of. Had we seen lost city organisms more grouped with each other, it could have suggested that there was a unique evolutionary event in sulfate reducing microbes specifically at the lost city, but that's not what we have observed. So it makes sense to conclude that the lost city DSR-AB genes cluster more by organism than by environment. The lost city taxa present in both of these trees are very similar. Pretty much the same taxa present on both of these slides in this blue color here, except for in this DSR-B tree, there is one species not present in DSR-A, which is Dihalococcoidales right here. And that was just an interesting find because it's a chloroflexi, which is not super well known for being sulfate reducing, but there has been some literature on the potential of like chloroflexi and Dihalococcoidales being sulfate reducing. So it was kind of interesting to see that in this tree. Also, more lost city taxa seem to contain DSR-B than DSR-A genes, which is interesting because both DSR-A and DSR-B together are needed to undergo dissimilatory phosphate oxidation. So you need both the DSR-A and DSR-B subunit to use that metabolic pathway. Overall, we found that lost city DSR-AB genes cluster more by organism than by environment. Dissimilatory sulfate reduction is likely an important metabolic strategy in the lost city subsurface that we were previously unaware of, and that DSR-AB genes likely evolved from multiple evolutionary events that reflect the history of dissimilatory sulfate reducing organisms. And we also found that dissimilatory phosphite oxidation likely does not exist within the lost city microbial community. So that being said, I would like to acknowledge co-chief scientists, Dr. Susan Lang and Dr. William Brazelton of the Lost City team for going out on these expeditions and learning all about these microbes and bringing them back. Would like to thank Orcut Lab technician Tim D'Angelo for doing a lot of the single cell work here. Big thanks to REU Director Dr. David Fields for everything that you've done. My mentors, Julia McGonigal and Dr. Beth Orcut, who have been a great support for me here. And of course, all of the REU interns that have been so great. Okay, I can take like 2 questions. There's already one in the chat from Anne. Thank you, Anne. Anne asks, were you surprised that you found no evidence of dissimilatory phosphite oxidation in the Lost City environment? Is there something specific with this environment that made you think dissimilatory phosphite oxidation could happen? So we weren't necessarily surprised to find no evidence of dissimilatory phosphite oxidation. As I mentioned, there's only 2 known taxa that undergo dissimilatory phosphite oxidation. It was a relatively recent kind of discovery in the scientific field, but because the Lost City is kind of this anoxic, like very unique environment, we thought that there might be a chance of finding dissimilatory phosphite oxidation, just like looking at the other environments where known DPO organisms exist. We thought that there could be a chance that they would exist at the Lost City too. So it wasn't necessarily surprising that we didn't find them because there's so few of them out there, as we know now, but still interesting.
No. No, I just want to ask the question. Can I ask one? Okay. I'll walk back there. I'm wondering if you didn't find that the phosphite to phosphate because you were looking at so few cells. And is it possible that it's there and you just don't have enough single cells to find it?
That's possible. We did have 179 single amplified genomes from the Lost City fluids that we were working with, which I think is a pretty decent number of cells, maybe not. But it's definitely possible that we didn't have like a large enough sample size to find dissimilatory phosphite oxidation. It's like definitely possible, but also unlikely that it would be there. Who knows?
And I'm going to walk back and I'm going to ask a question while I'm walking back to David. So you found more of the subunits A as opposed to B?
Yeah, we found more subunit B than opposed to subunit A.
Do you think that's because there's just a bunch of gene transfer that's going on down there and it's just incomplete? I mean, we saw it was a cesspool of Sodom and Gomorrah.
Yeah, no, that's actually possible because I mean the taxa that Danny found that have high amounts of horizontal gene transfer in the disulfide maculaceae and the thermodisulfovibrinales are also the taxa that I was looking at for dissimilatory sulfate reduction that we found evidence of. So definitely possible, but we don't know.
Well, David took my biblical Sodom and Gomorrah question, so I won't ask it, but it was a great talk anyway. I've learned a lot about Lost City.
Thank you.
Oh, thank you.
Okay. Hi, my name is Alexis, and I'm a rising junior at Bowdoin College in Brunswick, Maine, and I'm studying Earth and Oceanographic Science there. And I'm really excited to share my work this summer, where I assess stage-specific thermal tolerance of American lobster larvae. And I'd like to thank my mentors, Eric Annis from Hood College and Doug Rasher from Bigelow, for helping me through this. So to start off, our story begins with egg-bearing females, also known as eggers. And thank you to the Maine Department of Marine Resources for helping us get those. But anyways, we are thankful to them for supplying the larvae necessary for this project. And to start out, when the eggs hatch to larvae, we categorize them as stage 1. And then once those molt, we categorize them as stage 2. And this continues on. And I wanted to point out here that I included a 1 millimeter scale bar here to demonstrate the increase in biomass growth from stage 1 to stage 4, and also point out that our stage 4 are also known as post larvae because they settle down to the benthos during that stage. So yeah, the degree to which larvae are able to tolerate different sea temperatures influences their ability to survive and develop. Development here refers to an individual's ability to molt to the next larval stage and also grow in biomass. So temperature is hypothesized to determine larval settlement. And I'm going to show here a stage larvae settling down to the benthos, which impacts the distribution of both juveniles and adult lobsters in the ocean. So why is this important? Maine is home to North America's most lucrative single species fishery, and studying the thermal tolerance of lobster larvae can help to predict future distributions of the fishery. There we go. So to start out, we have our lobster larvae in the water column, and here are 4 pelagic larval stages. So 20 degrees Celsius was the average sea surface temperature in the Gulf of Maine last summer. And we know that 18 degrees is the optimal rearing temperature for larvae in lab, in which they have low mortality and successful development. And so if we were to test within the optimal temperatures for larvae, which is around 18 degrees, we would likely see no stage-specific differences in development or survival, meaning that larvae would have low mortality and be successfully molting and growing. And so this is why we are testing larvae in this experiment at 8 degrees and 26 degrees, temperatures that are hypothesized to be near the lower and upper thermal tolerance limits for lobster larvae based upon a bunch of work in the past that has been done on larvae. So it takes an incredible amount of effort to rear healthy larvae in lab. And in order for us to single out temperature as the variable responsible for the death and development we were seeing in this experiment, it was crucial for us to limit other stressors such as handling mortality and lack of food. And so we decided to raise larvae individually in 400 milliliter glass jars, which you can see here in our 18-degree incubator. This was when we were near full capacity with 1,000 larvae in the system for our broader project this summer. And so water was changed 3 times a week with filtered seawater at the treatment temperature that larvae were being raised in to avoid a temperature shock. And larvae were fed brine shrimp and checked daily for development and mortality. So this flowchart highlights the process of this project following the hatch of stage 1 larvae. So once larvae were hatched, 30 stage 1 larvae were placed into 3 different temperature treatments here, 8 degrees, 18 degrees, and 26 degrees. So other larvae from hatch were then placed into the optimal rearing temperature of 18 degrees. And once there were 30 newly molted stage 2 larvae and 30 newly molted stage 3 larvae, those larvae were placed into the 3 temperature treatments. So larvae were checked daily for signs of molting or mortality, and they remained in their respective temperature treatments until they either molted or died. So molted larvae were removed from the incubator
And then they were photographed for carapace length and frozen for dry weights. So here's an example, one of the graphs I'll be showing for my results. Let's see how to do this. So yeah, so our 8-degree temperature is shown here in blue. Our 18-degree temperature is shown here in orange, and 26 degrees is shown in pink. And down here we have the days since the start of our treatment. And we also have our percent survival here on the y-axis. So I wanted to point out that the ending of each of these lines indicated that all larvae in the treatment had either molted to the next stage or died. And also show that an ontogenetic shift is a change during the course of development. And this can signify a change in thermal tolerance between two larval stages, for example, from stage 1 to 2 or from stage 2 to 3. So showing our results, we had very high mortality here in our stage 1 larvae in our 26-degree temperature treatment. And this signifies that there's likely an ontogenetic shift in thermal tolerance from stage 1 to stage 2 larvae. Because as we can see here, both stage 2 larvae and stage 3 larvae had higher survival rates there. So they were more able to withstand that temperature treatment than the stage 1 larvae were. Something else I wanted to point out is that stage 3 larvae had a higher mortality in our 8-degree temperature treatment, our cool temperature treatment, than stage 2 larvae did, which also signifies that this may be an ontogenetic shift in thermal tolerance. For molting, our larvae experience extremely fast development times in 26 degrees. And as you can see by the long lines here, the number of days in treatment, all of these larvae in the 8-degree treatment for stage 1, stage 2, and stage 3, they were staying in that 8-degree temperature treatment for so long because they were having difficulty molting, which signifies a slow development time. Oops. So in terms of our growth rate results, we found that carapace length growth rate increased significantly in stage 1 and stage 2 as temperature increased. And then we didn't find a statistically significant difference in growth rate between our 18-degree and 26-degree treatments for stage 3 larvae. And we found similar findings in terms of doing growth rates using the dry weights for these larvae. So I like to use an analogy to Goldilocks and the different, and so Goldilocks and the 3 bears. There's this, too hot porridge, there's the too cold porridge, and there's the just right porridge. Well, that's very simplified, but in terms of our results, we found that for the 26-degree temperature treatment, while it wasn't lethal for stage 2 and stage 3 larvae, it did show increased mortality in stage 1, which signifies that this may be nearing the upper thermal tolerance limit for lobster larvae. And for our 8-degree temperature treatment, we saw extremely slow development there. Low mortality overall, but then stage 3 experienced a higher mortality there at that 8 degrees. And then we found that in our 8-degree temperature treatment, which is known to be the optimal rearing temperature, and so was our control, larvae of all stages were experiencing low mortality and relatively fast development times. And this led us to our conclusion, there we go, sorry. So this led us to our conclusion that as waters warm in the Gulf of Maine, larvae are likely to shift lower into the water column to reside in seawater closer to 18 degrees Celsius while still avoiding temperatures around 8 degrees where we know that they have slow development slow development times that may inhibit settlement. And I'd like to thank the National Science Foundation for their funding for this project through Eric and Doug's 3-year project on larval thermal tolerance, as well as the REU program funding. I'd like to thank Aubrey and Hannah, the other lobster gal pals of the Seawater Suite, for their help on this project. They were doing water changes with me as well, and helping organize the system. And thank you so much to Eric and Doug for making this project possible, as well as the Maine Department of Marine Resources for providing the egg-bearing females.
Thanks for a nice study and presentation. A question for you about the choice of temperatures. Can you remind me again why you wanted to go for 26? It seems high for the Gulf of Maine, but maybe we're anticipating that being more common?
Yeah, so most of the larval research has focused mainly around 10 degrees to 22 degrees. And in terms of looking at stage-specific tolerance, kind of the bulk of research has shown that more around lower temperatures than that, so around 8 degrees and 26 degrees, may be around the lower and upper thermal tolerance limits. And that's where we would start to see differences in the larvae that would hint that they're experiencing, cold or hot stress. Yeah. Thanks.
When you showed it at the end that the larvae might go down in the water column to the correct temperature, would they be missing out on any other benefits of living more towards the surface, maybe nutrients that they feed or food that they feed on?
Yeah, that's a really great point. Something I was talking about with people yesterday at our poster presentation is where does food availability come into this question? And we don't actually know too much about what larvae feed on, but we were talking about if this temperature is warming at the surface and larvae are shifting down in thermally stratified systems, it's also likely that that's pushing down the thermocline where food source may be able to go deeper down. And so they may still be able to feed, but then again, we don't really know that much about what they're feeding on in the ocean. Yeah.
So you suggested that this data is going to be shifting these larvae to colder water so they can avoid the heat stress. And so there is still studies going on right now to try to find where these larvae are existing within the water column. So do you think as time goes on and the warming of the Gulf of Maine is going to increase, will we be seeing these larvae shifting to colder waters, whether that means into deeper stratified water or maybe offshore?
If I get what you're asking whether we think that they're actually going to shift down into waters. Are you talking about over down below this 18-degree range? Or what specifically were you thinking?
Yes, exactly what you said.
I think our thought is just at the surface of 26 degrees, we can see that the stage 1 are already experiencing high mortality. And then at the 8 degrees, they're not able to develop. They're not able to get to that stage 4 where they settle down to the benthos and populate at the bottom. And so our thought is that if they're experiencing this heat stress at the top, they're going to shift down more towards where they're able to develop. I'm not sure if I answered your question. I'm sorry.
No, absolutely.
Thank you.
Yes. Yeah.
Okay. Yeah, you're good. Okay. Hello, I'm Autumn Pope. I'm a rising junior at Hartwick College studying biology. This summer, I'm working with Dr. Stephanie Carr from Hartwick College and Dr. Beth Orcutt from Bigelow Lab. And we are investigating pili structures in the marine crustal subsurface. So, okay. The subsurface area that I'm focusing on is an area off the coast of Oregon in the Pacific Ocean called the Wandafuka Ridge. And this is a very hot, low energy environment. So we're wondering how these microbes living in this environment are surviving. So in the marine crustal subsurface, cold water in the water column filters down and enters an outcrop in the ocean floor, and then enters this area called a crustal aquifer
And that cold water is then heated by the lithosphere. And then that hot water then circulates back out another outcrop and enters the water column. So the crustal biosphere is a really important area for us to study because it is 2 × 10 to the 18th cubic meters in size, and it contains 10 to the 25th cells. So if you lined up 10 to the 25th cells, you would be able to go to the moon and back 300 times. So going back to the Juan de Fuca Ridge subsurface, this area, like I said, is a very extreme environment. So has very little dissolved organic carbon. The microbes living there don't use oxygen as an electron acceptor. They use sulfate, so that provides them with a lot less energy. And to go on top of that, it is 64 degrees Celsius, which is very, very hot. So we are wondering how these microbes living in this crazy environment are surviving, and how are they getting energy to do a bunch of things. Okay, so to sample the marine crustal subsurface, we're able to insert these underwater borehole observatories, which are a lot like wells that we're able to put into the ocean floor and sample fluids from the subsurface. So then we're able to extract those fluids, and scientists can count the number of cells in the fluid. They're able to culture microbes, well, they can try. And we can look at the genomes of the cells down there. So looking at genomes from the different areas of the subsurface, we can see that in the crustal aquifer, where that water is being heated, there is a high abundance of motility genes in their genomes. So those motility genes are able to help a cell build structures so they can swim around in the ocean. And even though 2% seems like a really small percentage, comparing it to really motile organisms like Salmonella or E. coli, which have 2.5 to 5% of their genomes dedicated to motility, that's a pretty substantial number for these microbes living in such an extreme environment. So, like I said, motility could mean that these microbes are using motility structures like pili or archaeolum to swim around in the subsurface fluids to try to catch nutrients, or they can create biofilms through attachment. So they're able to swim through the subsurface, attach to a surface, and then create microbial colonies to make biofilms to obtain nutrients. Smith et al. Was able to show this by putting rocks into one of these boreholes, putting it in the ocean, taking it out a few years later, and they were able to see that biofilms did start to form on those rocks that were in the subsurface. So the big question I have is, how are these microbes able to power motility and attachment if they're in such a low energy environment? And this is really important because if we're able to recognize motility and attachment, we can understand how energy is used by these cells in the subsurface. We can understand how cells are working together, like to create a biofilm, and we can understand how nutrients is cycled in the crustal biosphere. So that could be either using their pili or archaeolum to swim, searching for nutrients, or they can use them to attach to a surface and create a biofilm. So my project is focusing on researching pili structures in crustal archaea. So pili structures are these wiggly lines. They're a lot like flagella, but they're in archaea, they're hollow, and another structure similar to them is archaeolum, which is flagella, but for archaea. So they're able to use those to swim around or create biofilms like this picture. So the two main questions I have is, what portion of the Juan de Fuca Ridge archaeal community has these pili structures? And I also want to know if motility in biofilm was an adapted trait that was evolved early in life's history, or if it was acquired through horizontal gene transfer, like Grace and Danny have talked about. So focusing on question 1, I was able to use the Joint Genome Institute's database to search through Juan de Fuca Ridge genomes for archaeal organisms that had some type of motility genes. And I was able to find that I was able to find 42 archaeal genomes from the Juan de Fuca Ridge. And then I used that same database to search for this protein in a pili, which is called RI, but that doesn't mean anything to you. But it's actually an ATPase that is able to secrete the building blocks for these motility structures. So then I used the 42 genomes that I found, and I used a system called Protein Blast to search through those genomes to see if they had that RI protein. So looking through those genomes, they were single cell amplified genomes, so SAGs. I was able to break them down into 2 phyla, so TAC and Euryarchaeota. And out of those 42 SAGs that I looked at, 41 of them had that RI protein. The only one that didn't have it was this methanopyri. And these circles represent how complete the genomes are. So like in Archiglobus, they have a bunch of very complete genomes, but in methanopyri, it's not very complete. So that probably explains why we didn't find any motility genes. So then moving on to question 2, I was able to make a phylogenetic tree to understand the evolutionary history of this ATPase. But before I show my tree, I wanted to show a simple version of a archaeal tree of life. So Depan, Euryarchaeota, TAC, and Asgard, those are all phyla in Archaea. And you can see that they branch separately from bacteria. And it's hypothesized that eukaryotes may have branched off of Archaea. So then to understand like what horizontal gene transfer is, or if those proteins evolved early, we have to talk about what that stuff is. So here there are 2 Euryarchaeota groups right next to each other. So if a pili protein showed up twice in a genome, that could mean that that gene was duplicated. So that would represent gene duplication. But if there are 2 groups that are separated, and the pili protein was shared in between them, that could mean that genes were shared through horizontal gene transfer. So to make my tree, I was able to use a base tree from a Makarova paper and they used ATPase proteins as for their base tree. So then I was able to take those sequences along with my Juan de Fuca Ridge sequences and align them using a process called Muscle. And then I took those sequences and was able to actually make my tree with a program called RAXML. And then I took that tree and used programs called ARB and ITAL to analyze it and make it look very pretty. So this is my tree. So in the Makarova paper, they separated their tree into 4 clades. So clades 1, 2, and 4 represent sequences that have pili proteins. And then in clade 3, those sequences have an archaeolum protein. So then, also, these orange dots represent all of my Juan de Fuca Ridge sequences, and the red dots represent hydrotherm archaeota sequences from the Juan de Fuca Ridge, and those are important, and I'll talk about them in a bit. And then I separated the branches, the blue branches. It's kind of hard to see on there, but I'll do a zoom in later. The blue branches are the Euryarchaeota phyla, and the green is the TAC phyla. So an interesting thing that we found was all of those orange dots they're all in all 4 clades. So that shows us that those Wandafuka Ridge sequences have pili and archaeolum genes. Then going back to the hydrotherm archaeota, in clade 1, we noticed that there was a group of them all together and they were very deeply branched in that clade. So that could indicate that that was an early evolved clade 1 ATPase. So that gives us some hope to better understand how this ATPase has evolved. And then breaking down the Euryarchaeota and Tachyphylla, we can see that they grouped separately. So there wasn't like random blue like Euryarchaeota sequences anywhere. It was interesting that they all branched separately. And we also noticed that when there were 2 copies of an ATPase gene in one genome, it looks like it represented gene duplication and not horizontal gene transfer, like going back to those previous simple trees. So that was interesting. So the conclusions that I have from this tree, so focusing on question 1, we found that almost all archaea had some type of external pili structure. And we noticed that many groups had more than one type of structure. And for question 2, since we see that the Wandafuka Ridge sequences are in all 4 clades, that suggests that swimming and attachment are very important for life in the crustal biosphere. And going back to how the genes were next to each other, we can assume that gene duplication was present. So I would just like to say thank you to the entire ORCUT lab group. You were all so helpful and kind. And I've had just a really great summer, and thank you for last summer too. Thank you, Dr. Carr, for introducing me to this wonderful place. And they're not, I don't know if they're here, but we have some collaborators in Hawaii who have helped out a lot with the genomes. And I want to thank NSF for the different grants for providing the funding for this project. So are there any questions? Okay, so Anne, she asks, do you have a hypothesis for why Wandafuka Ridge archaea would have both an archaeolum and pili? Do they serve different functions? So great question. We're trying to figure out what they mean. We tried to do some culturing, but it's not going well, just because they're really hard to grow and they're very picky. So hopefully we can figure out some way to induce some type of like biofilm production, just to see if we can even see the different types of structures. But yeah, hopefully we'll find out.
Yeah.
That was really cool. Do you have a sense of, so you showed that phylogeny and how like the eukaryotes evolved from or seem to have evolved from the Asgard archaea. Do you have a sense or do they know if is the archaeolum or any parts of it homologous to the eukaryotic flagellum? Or is it kind of a, because I know the bacterial flagellum spins like a motor, like a rotor, it moves completely differently than the eukaryotic one. Do you know like are there any conserved parts that seem to exist between those
I'm
systems?
pretty sure they rotate like that.
Yeah.
But I don't think they're like eukaryotic. I think they're more like bacterial.
Okay. So eukaryotes like probably evolved it independently.
Maybe.
Cool.
I don't really
know. Yeah, you don't have to figure that out now.
Yeah.
I know it took like 1 billion years to evolve, so.
Yeah.
Great presentation. I just wanted to confirm, when you say gene duplication, you mean that they have 2 copies of the gene in their genome, or do you refer to a different thing?
2 copies in their genome.
Gotcha. Thank you.
So I got one. So the Earth is spitting out a ton of these things all the time. And do they survive once they leave that environment, or are they constrained to living in there? And this is just, then it's just spewing this organic carbon into the ocean.
I'm not sure, actually. I don't think.
I mean, it seems like a lot of cells.
Yeah, because like we, with our samples, like we've been focusing on one species called Archiglobus sulfaticalidus. And they have to be grown at 65 degrees Celsius. Like they're very, very picky, and that's why they're so hard to grow. So yeah, I don't think they can just survive. Yeah. Yeah. Great. Thank you.
Cool.
Okay. Okay. Hello, everyone. My name is Paxton Tomko. I'm a student at Purdue University. And this summer, I was working with Jacob in the Stephanakis lab on a project where we were using a unique data set to investigate the role viral infection plays in phenotypic expression in marine microorganisms. Oh, hold on. So viruses are the most abundant and diverse biological entity in marine ecosystems and in all ecosystems. To get an idea of their abundance, this photo shows a fluorescent stained drop of seawater, with the larger spots being prokaryotes and the teeny tiny specks being viruses. Viruses outnumber bacteria by about an order of magnitude. In addition to their abundance, they are also major drivers of evolution and genetic diversity, and they play a key role in geochemical and nutrient cycling in marine environments. This is the marine food web with arrows indicating the flow of carbon of trophic levels. Viruses inhibit a portion of this carbon from reaching organisms at higher trophic levels through a process called viral shunt, in which the virus lyses its host cell and returns the cellular components back to dissolved organic matter. Now, some of these cellular components can aggregate together and become food for organisms at higher trophic levels. In addition, viruses themselves can be prey for these higher organisms, which is shown here through this viral link. Now, in addition to this, viruses are also known to change the metabolic potential of their host. This has been seen in some species of cultured cyanobacteria, where the cyanobacteria physically get larger when they're infected with viruses. So this study was done in a cultured system, and so this is one of the things we're testing to see if this pattern holds true for natural environments. So for those of you that don't know, traditional methods of studying virus-host interactions primarily rely on culture-based approaches, but this poses limitations as a very small percentage of species can actually be cultured. And so to bypass this need, we can use a technique called single-cell genomics, which allows us to sequence the genome of individual cells taken directly from an environmental sample. This increases the resolution of viral investigation as it allows the observer to directly identify the virus and host in the system and observe genetic and metabolic characteristics or interactions between the two that can't be seen on a community level. So the data set I'm working with, as I mentioned, is pretty unique. It combines these single-cell genomes with phenotypic measurements for those individual cells. And specifically, we are looking at respiration rate and cell size between our infected and uninfected populations. So prior to my involvement in the project, 6 samples were collected from the Gulf of Maine. They were collected right off the dock here at Bigelow between the years of 2017 and 2019. They were tagged with one of 2 fluorescent dyes, redox sensor green, which we calculated oxygen consumption from, and Cyto9, which just stains all DNA containing biological entities. Cell diameter was estimated using a forward light scatter and flow cytometer. And from there, cells were randomly selected from the environmental sample and sequenced using short-read Illumina sequencing. So my involvement in the project was completely computational, and so I want to give you an overview of that workflow. We started by identifying viral sequences present in our genomes and performed a quality assessment to remove all contamination. From there, we did our phenotypic analysis where we were looking at estimated cell size and oxygen consumption changes between our infected and uninfected populations. So here are the results from our viral identification analysis. What's really important about this is we identified at least one viral sequence in roughly 43% of our genomes that we had in our data set. So everything that contained at least one viral sequence is what we're considering to be infected, and everything that doesn't contain a viral sequence is our uninfected population. Now getting into the phenotypic analysis, we started by looking at respiration. And so these are the violin plots that David was talking about if you were here earlier this morning. So this is showing changes in distribution with the widest point on the chart containing the most sample sizes. So here we have our infected cells shown in blue and our uninfected in yellow. And our respiration rate is shown on the y-axis on a log scale. So at the population level, we did not see a statistically significant difference in respiration. And so we broke this down by day to account for seasonal variations. In our October 2018 day and our April 2019, we saw a similar trend in the sense that there was no statistical difference. For our July 2019 collection day, we did see a slight difference with the infected population displaying slightly greater respiration than the uninfected. But still, we weren't done looking at this because different genera can have quite variable respiration rates between them. And so we ended up breaking this down by genera to try to account for some of that variation. So this is a 16S tree that shows the taxonomic breakdown at the genus level. Statistical significance is indicated with a red star. Our median infected respiration rate is shown in blue and our uninfected in yellow. So for our October date, we identified 6 cases in which we saw a statistical difference. We identified 6 for our April date as well, and 5 for our July. Now, I know this is a lot to look at, and it might be kind of hard to see in the back, but what's really important here is that overall, in the cases that were significant, we saw the majority of genera decreased their respiration rate, meaning the uninfected population had greater respiration than the uninfected. And that is shown by everything highlighted in a red box. So next we looked at cell diameter, and once again, we looked at this for the total population. Here we did see a quite significant difference between our infected and uninfected, with our infected cells being much larger than our uninfected. It's kind of hard to see this by this plot, but you can see a slight shift in the change in distribution. But once again, this was statistically significant. So similar to our respiration, we broke this down by genera. So here's another 16S tree. And again, everything that is significant has a red star. Here, this is not plotted on a log scale. So the median infected estimated diameter is in blue and the uninfected is in yellow. Again, it's a lot to look at. But the main takeaway is that of the cases that were significant, the majority of genera increased their cell size when they were infected. So the infected population was larger than the uninfected. So in conclusion, we found that viral infection does have an effect on host respiration rate and cell size, with the majority of genera getting larger and decreasing their respiration rate once infected. Now, there's still quite a bit of work that has to go into this project, and so I'm very thankful to be continuing this work with Jacob in the fall. Our next step is going to be looking at this from the viral side of things rather than the host side. So we will do this through viral clustering and gene annotation to see if there's any metabolic genes in our viral sequences that may be impacting the host metabolic potential, which leads us to see these trends in the data. So I would like to thank my mentor, Jacob, for helping me with this project, as well as Ramunis and Julia for all their feedback. I would also like to thank Nicole Poulton for preparing the data set, as well as the NSF for funding of the REU and the Genomes to Phenomes project. So are there any questions? Okay. Yep.
Great talk, Paxton. I just wanted to know what the implications for this research is that you found this and plan to continue on with this research.
What do you mean by that?
So with this research that you've done, are there any, can you apply this to something that is prevalent in this study, or is this pretty niche of just trying to find out these nuance interactions, I guess?
Yeah, so I mean, the data we have, it is representative of like the natural environment because we took it directly from an environmental sample, but it's still a pretty small data set compared to the entirety of the ocean. So I would imagine you would need a much larger data set to draw any conclusions.
Thanks, Paxton. You showed the table and talked about how at least 43% of your single cell genomes had evidence of viral genes in them. And you mentioned all the different programs you used to try to classify those genes. And I know some of that can be a little subjective, right? So for instance, VirSorter gives you like classification 1 or 2 or 3. So I'm curious, did you have to go through and like manually evaluate all those 3,500 or so genes, or what level of confidence do you have in those classifications?
Yeah, so that's a great question. We didn't go and manually evaluate those. We ran a program called CheckV, which checks the quality, and it gives you like a score of like between 1 and 3. But we just use CheckV to remove contamination because of the nature of short-read sequencing and single-cell genomes. We didn't see a whole lot of high quality viral sequences identified, but that could just be because we're using short reads. And so what we really have is a bunch of longer viral sequences that just get cut up, and so they don't read as high quality.
That was really interesting. I was just curious, and sorry if I missed this, but so you mentioned that the majority of the genre showed increased cell size when they were infected, and the majority also showed decreased respiration rate. And I was just wondering, was it the same genre, or was there an association between those two effects, or was it kind of just random, no association between those groups?
Yeah, so we saw a decrease in respiration rate, but an increase in cell size. We didn't go and compare the genre yet. That's going to be the next part of this project.
Thanks.
Yes, we would.
Were you shocked at 43%? And did you happen to find seasonal differences in the in the amount of virally infected cells?
Yeah, so we didn't look at seasonal differences for the viral sequences identified in the cells. The 43% wasn't completely shocking considering the magnitude of viruses in the ocean, but it was exciting that we didn't just have like one or two. We had something to work with, so.
Thank you.
Thank you.
Okay, thank you. Hello, everyone. I'm Jace. I go to California State University at Monterey Bay. I'm working in Nick Record's computational lab. And for my project, I've been helping the Single Cell Genomics Center create a model to help sort their single amplified genomes utilizing AI, specifically a neural net. So we're currently at a point where we can extract more data from the environment than we can accurately classify. As we've seen in some of these talks, there's a lot of blast categorization going on, and we can't always get exactly the results we want. And this is especially true for the ocean, marine environment, where a single drop of ocean water contains over 1 million cells. So I'm interested in coming up with a novel model to help predict some of these categorizations that we're trying to make. The data set I'm working with is the Gorg Tropics data set. It was sampled all across the globe, and over 10,000 prokaryote genomes were sequenced. A lot of work went into classifying all these genomes. I'm going to talk a little bit about that right now. So as we heard in the last talk, these tools we use to categorize these genomes are aligning our sample genomes to reference genomes we have in a database. And that's good because it gives us a certain degree of certainty when we make these classifications. But if we don't have a genome similar enough, we can't make these classifications. And this is especially true for prokaryoplankton, which is a very big group because they're very diverse, even within species and between different genres. And especially if we want to categorize something like virus presence, where there's more viral DNA in the ocean than any other type of DNA, we're not at a point where we have all this DNA readily available in these databases to make these classifications. So a method that's currently being used in the Single Cell Genomics Center to help classify make some of these categorizations without actually aligning sequences is with this principal component analysis. And in this case, this principal component analysis is comparing the variability of DNA tetramers, which are 4-letter nucleotide combinations, along the genome. So every point you see on this plot is a small portion of the genome, and we get its coordinates by comparing its variability of these tetramers to the rest of the genome. So it's like a way to 2D visualize the genome in a 2D method. So this is a simplified version of that last plot, and it can help us understand what we're actually looking for when we look at these plots. On the left, we have a typical single amplified genome cloud. There's no significant deviation from the center, and it doesn't really tell us a whole lot about this cell. But in the middle plots, we see that there's 2 distinct clouds. And this could be because when we isolate a single cell, we get 2 cells in the well by accident. And so when we start to plot the genome from that second cell, its variability is different enough from the other cell that it starts to make its own cloud. And we can see that in these plots. On the right, we see a single portion of a single contig that deviates significantly from the main cloud. And this could be for a number of reasons. It could be the presence of a viral prophage. It could be a preserved gene that doesn't mutate like the rest of the genome. It could be a past virus prophage that now lives in the genome and that species reproduces with this prophage. I can't tell specifically what it is, but we can see it with these plots. So the Single Cell Genomics Center, to categorize these cells, it can take them weeks to look at these plots for every cell and look for the things that we just look for. And I'm interested in automating this process using AI. So AI has been around since the 50s. Traditionally, it's been what's called rule-based, where these programmers are entering these functions and rules into the AI so that it uses the input data and makes these predictions that the programmers want. That's quite different from machine learning, where we're not entering any rules or functions. We're entering in training data and expecting it to make these rules and functions. So that's why it's so important that I had a large data set that was already labeled. So neural nets are kind of the new thing in machine learning. They're used in a lot of image classification tasks like facial recognition or even language translation. And this problem of visually identifying these plots seemed like a really good problem that can be solved with a neural net. So my workflow was taking this PCA data and formatting it in a way that's uniform for every cell so that I can input it into this model and test this model. These models in themselves are very variable. There's a lot of different options you can have when you make these models. So I would format the data, input it in the model, and test it to see if it was predicting accurately. Most of the time it wasn't, so I would go back and change something about the model, or I would go and reformat the data altogether. So this was the first data format I used. It's really simple. It's a 2D histogram, and it's basically like those plots that we saw where each point is being accounted for in one of these bins. It didn't perform well compared to the others, but I changed it to entering multiple of these histograms into, instead of a 2D data format input, I use 3 dimensions, which helps the AI pick out patterns. So I tried 2 different formats where instead of using 2 principal components, I used 8, and instead of single histogram for the whole genome, I would split the genome up and give each portion its own histogram. The last format I tried were these images, which are the most similar to the plots we saw that the Single Cell Genomics Center uses to make these classifications. It's a little bit more data because we can see the how the contigs align to each point with these lines. But ultimately, none of them predicted as well as I was hoping. I tried 4 different classification problems on all these models, and here are my results. Ultimately, I thought that the most help would be to alleviate all these cells that the Single Cell Genomics Center has to go through. So I kind of categorized them by cells that would require further analysis after they look at these plots, and cells that they wouldn't necessarily need to look at these plots for because they looked good enough. And So these are the results for that classification problem. And even then, it's a little hard to say how well they do, because there are some plots where we can look at it and it can go either way. We could say, well, we could look at it more, or we couldn't. Could go either way. So that's why I think some of these scores aren't very well. But something like virus prediction was especially hard to predict. That's where I spent most of my time trying to figure out, and it proved to be the most difficult category to predict on. Something like contamination was a little bit easier, but nothing like in the high 90s, which is what I was hoping for. Something that kind of surprised me was predicting on these lineages of all these cells. So random guessing for these 10 lineages that I was training on would be 10%, but there must be something about these plots that these lineages have its own signature that this model can pick up on, because 70% is a lot more than 10. So going forward, it would be interesting to add some blast data to these plots. So instead of just a black and white image, we could color code some of the contig strains to give the model more information to work with. That'd be interesting to see how it predicts after that. Before I take questions, I want to thank NSF for providing the funding for my research, and Nick Record for taking me into his lab and letting me play with AI this summer. Ben Tupper was really great resource to have for coding and just talking about my project in general. Julia Brown provided the data, and Jonathan Evan was really good help with neural networks. Thank you. Cool.
Really interesting things to think about here. For evaluating these tetramer plots, right, and trying to decide, okay, this is probably spurious, maybe it's 2 cells. How does the tetramer plot approach deal with the idea that some cells may have extra chromosomal DNA, like a plasmid or something like that? Will that be detected, or will it just show up in the big clump of everything else?
Yeah, it's a great question. So like I said, this is the first step that they do when they make these analysis. So it is like a way to weed out suspect genomes that they can perform further analysis on. So yeah, there's really nothing that these plots specifically can say like for certain, but it does make its own, it does have its own visual identity, which is kind of interesting.
So with machine learning, the better the sample size, the training data that you feed in, and the more data you feed in, the better the predictions get, right? So did you, I guess, how many sample like data sets did you use? And did you monitor at any point like an improvement with time, like maybe that 70% that you had with the screening accuracy will improve as you add more data sets in?
Yeah. So to put it in perspective, out of the over 10,000 genomes that were in my data set, about half were actually labeled with lineage. And that goes for most of these other categories I was doing. So typically how I would try to go about things is I would use maybe a quarter of the data available to me to create something that I thought would work well. Like I would see an improvement on like experimenting with the format or the format of the model. And then I would use all the data for the final model and see how that predicted. And yeah, I would see an increase, but with more data, it would only be better.
Great talk, Jace. Beyond the detecting a possible virus or contamination in the PCA plots, is there anything else that the neural net can pick out for us from what we're seeing in those plots?
it would be really interesting to kind of expand this past prokaryoplankton into, I mean, every cell, every genome would make some sort of signature, and it would be really interesting to see like at what kind of classifications you can make. Like, could it be mammal or, you know, arthropod classifications? I'm not sure, but like how it picked up on lineage would make me assume that it is possible. Yeah.
Thanks.
Can it spit out the rule once you do that? And
I wish it could, because then I could.
Right, you can't. I mean, the end point is interesting in itself, right? I mean, that means it's a pragmatic thing, but intellectually, it'd be interesting to find out how it got there and the steps that it took to divvy up the different populations.
Yeah, that's a great point, and just a great criticism on neural nets in general, because these models are being used in all sorts of fields, and yeah, we can't look inside this box and see how it works. And there's a lot of fields where that is a big problem. And for this specifically, like seeing what it was looking at would be an interesting thing to compare to, but no, we can't.
Nice talk.
Thanks.
Would you expect the same result if instead of for the nucleotides you put 5, 6, 7, why 4?
Yeah, I mean, it's kind of, there's a lot of arbitrariness in every step of this process, like to the data format, to the number of the size of the windows and the steps making this plots. And yeah, using DNA tetramers is another arbitrary step. It would be really interesting to see how it does with other combinations of nucleotides.
Thank you.
Oh yeah, thanks.
Hello, everyone. My name is Maria Jose Reana Rosales. I'm from Southern Maine Community College, and this summer I'm working with Dr. Jose Fernandez Robledo in the project expression of the SARS-CoV-2 RPT spike gene in the marine protozoan Perkinsus marinus. As you can see here in this picture, this is Perkinsus marinus expressing the green fluorescent protein GFP. We will all remember the year 2020 when the SARS coronavirus 2 caused a major global health crisis. And in this electron micrograph here, you can see the SARS coronavirus 2 in yellow and its spike protein in red. The SARS-CoV-2 spike protein, especially the S1 subunit, also known as the receptor binding domain, it's the one that interacts with the ACE2 receptors in our cells, mediating the entrance and fusion of the virus into our cells, which then it makes us sick. The world is still fighting and struggling with the pandemic, as you all may know. As of April 2021, only 5% of the world population have been vaccinated. And novel vaccine technologies like mRNA vaccines have made it possible to get back to normal in places like here in Maine, and made it possible to have events like this in person. But countries in development, specifically around the world, lack the infrastructure to distribute vaccines. According to the World Health Organization vaccine tracker, right now, as of July 2021, there's 108 vaccines candidates under clinical trials. Most of these vaccine candidates all use many different strategies, and most of them are using protein subunits from the virus or using coding information for the proteins like DNA or RNA, or inactivated virus particles or non-replicating virus. None of these vaccine candidates are using a marine organism as a vector. Also, vaccines are usually in a form of injection. But if you're like me and hate needles, you will probably will be better taking an oral vaccine in form of a pill than an injection. And now here I present to you Parkinsus marinus, the marine protozoan. If you have eaten oysters before, you have most likely already ingested this guy here, Parkinsus marinus. Parkinsus marinus is an oyster parasite that causes a dermal disease in oysters. Parkinsus marinus is very unique because unlike other parasites, it does not require a host to fulfill the life cycle. And we can grow it in vitro in an incubator and in defined culture media. Our previous research from our lab showed that mice that ingested Parkinsus marinus had an immune response towards it, meaning that it recognized it as a foreign cell. But it did not cause any pathology or symptoms that made it seem that the mice were sick. Also, another special thing about Parkinsus marinus is that it can be genetically modified using a plasmid, which, sorry about that, which is a circular piece of DNA used to introduce foreign genes into the cells. Previous research from our lab also and other people around the world have successfully achieved gene expression from pathogens like Ebola and malaria in primary rhinos through a plasmid. And my mentor before and his collaborators were able to create this plasmid specifically for Parkinsus marinus under a highly expressed gene promoter called MOE and tagged with a GFP, green fluorescent protein, and an anti-puramycin gene, which act as a selectable marker. We are going to call this plasmid from now on PMGP. A recent article from Sakamoto et al. Also described a new plasmid that can be used to transfect Parkinsus marinus with organelle-targeted genes. When the SARS-CoV-2 genes became available, we were able to acquire a plasmid from AdGene, and AdGene is a plasmid repository. And that plasmid that we got, contained the RBD spike gene, as you can see here in blue, tagged with GFP, as you can see here in green, under a CMV promoter, which means under a cytomegalovirus promoter, which is intended for expression in mammal cells. But we decided to try this plasmid in Parkinsus marinus. So we transfect the Parkinsus marinus using the plasmid I showed you previously, using electroporation. And electroporation uses an electric current to create holes in the membrane of Parkinsus marinus, allowing to the plasmid to go inside the cells. After the transfection, we analyzed the Parkinsus marinus cells under a confocal microscope to search for green fluorescence. And before I show you some pictures of the confocal microscope, I want to do a quick view of Parkinsus marinus morphology. So here is a Parkinsus marinus cell, and like any other eukaryotic cell, it has similar organelles. But what is specific or particular of Parkinsus marinus is that it has a large vacuole, which is a storage organelle. And within that vacuole, it has a structure called vacuoplast, which is also a storage structure. And also Parkinsus marinus has a cell wall and a relic plastic. So we analyzed our cells under the confocal microscope. Sorry for the people here seeing very pixelated pictures. And we see a lot of fluorescence, as you can see. We saw fluorescence at different intensities and also cells of different sizes expressing fluorescence in different parts of the cells. As you can see here in this Parkinsus marinus cells, most of the fluorescence is located in their vacuole, especially in their vacuoplast. Also, we do not know if the fluorescence will be transient, meaning that it will only be expressed for a few days, and lost after cell division, or if it will be permanent, meaning that the cells will keep it, the daughter cells will keep it after cell division. And in this picture here, you can see it seems that this cell is undergoing cell division and there's is still fluorescent. And here, these two cells seem to be of a very similar size, and they're very close together. So we think that they just went under cell division and they're both fluorescent. And also, we see here some cell debris, most likely caused by a lysed cell, like a cell that ruptured. And we can still see the protein fluorescently green. So now we did this, we tried this plasmid intended for mammal cells and it worked. And now our next steps are moving these RBD spike gene in that plasmid specific for Parkinson's minus PMGP. And we are approaching this in two ways. The first method we are trying to do is called restriction cloning. And restriction cloning works by amplifying through PCR that RBD spike gene, incorporating NDE1 restriction sequences. And NDE1 is a restriction enzyme that cuts specific DNA sequences. And then we linearize PMDP and then incorporate those two together. The next method that we're trying to do is called gypsum assembly, which works similarly. But this time we amplify the RBD gene through PCR, but adding about 20 base pairs overlap with our plasmid PMDP. And then we linearize the plasmid and then we integrate those two together. And then we do the same process of transfecting via electroporation. After that, if we have NC fluorescence, we would do an immunofluorescence assay, also called IFA. And this assay uses human antibodies against the spike protein as the primary antibody. So we will expose ourselves to antibodies against the spike protein. And then we will also use as a secondary antibody that is fluorescently tagged. So it will bind to that primary antibody. And then we will use a confocal microscope to look for that fluorescence from the secondary antibody. And this assay will tell us if Parkinson's minus is expressing the gene, and also if the protein that is making is properly folded so that antibody is recognized. So now we are in the experimental stage of this project. So right now, our next steps are, like I said before, cloning the RBD spike gene in PMGP and transfect Parkinson's minus. Then we're going to select those transfectants to grow clones from those cells that are fluorescent. And then we're gonna do the IFA through a collaborator with brand new spike gene antibodies. And then we hope to test those clones in mice to see if they get any immunity against COVID-19 by being exposed to Parkinson's minus, meaning that they will ingest Parkinson's minus cells and see how they react to COVID-19. Also, we will try to use the same approach that as I have explained, we're using other Parkinson's minus plasmids. And then we will, in the future, hopefully develop a Parkinson's minus as an oral vaccine alternative. And here is like a schematic of our process. And these hopefully could also work with other pathogens of medical interest and create other vaccines from other pathogens, and also use pathogens or viruses that affect other industries, like aquaculture industries, like viruses for fishes, and so on. And for last, I want to acknowledge my mentor, Dr. Jose Fernandez, who has been incredible support. And also, even though they're not here anymore, I would like to thank Ragath and Umeda for their setting up all these methods and teaching me how to do that. And Dr. David Fields for coordinating this REU program, the Bigelow Laboratory staff who was there every day in the lab, and REU Interest of 2021, who made this experience much better. Also the NSF for supporting this project. Thank you very much, and if you have any questions.
Really nice and inspiring work. The idea to try this plasmid that was more designed for mammalian, was that just because it was available and was already connected to having the spike protein in it, or was there another advantage to picking that one instead of trying to get it already into your MOA?
Okay, so we first because we had it and it was available, and also because we were having trouble creating the PMGP plasmid specifically for Parkinson's marinus, so we decided to try it and see if Parkinson's marinus will actually, accept it and express the gene, and so it was mainly because it was there. Yeah. A question from the chat. Why did you choose to use electroporation as your method of transfection for Parkinson's? How does the efficiency of electroporation compare to other methods? So my mentor, Dr. Jose Fernandez Robilla, has done a lot of work in figuring out and developing methods to use for Parkinson's marinus, and I think they, based on previous journals that they have posted, or articles, sorry, they have chosen electroporation as the most efficient method to get plasmids inside of Parkinson's marinus, as opposed to like other transfection methods.
That was really interesting. I'm not sure if this is within the realm of stuff that you've studied as much, but I know one of the big things about vaccines is sort of needing to be at a certain temperature and going bad if they're not at a certain temperature. Do you know that if this theoretically would have the same sort of limitations, or would temperature be a problem for it?
We think that this is why we're exploring this, because this will take away all of those variables. So if Parkinson's marinus is already, so when they feed it to mice, it was able to go through that digestive tract and still produce an immune response. So that's why I think that's what we're trying to develop it as a alternative to delete all of those variables that pose like difficulties of vaccine distribution around the world. All right, thank you so much.
And how do I scroll down the presenter notes if I have like a lower one? Oh, I think I got it. Okay. And just leave the chat. Should I push my hands? Yes.
Hi, everyone. I'm Hannah O'Loughlin, and I go to Vassar College. My mentors this summer were Dr. Doug Rasher here at Vassar and Dr. Eric Annis from Hood College. The title of my project is Effect of Larval Acclimation Temperature on the Thermal Tolerance of American Lobster Larvae. Thermal tolerance refers to the range of temperatures at which an organism can survive. Thermal tolerance is important for lobster larvae because it dictates where larvae will settle. This then informs the distribution of juvenile and adult lobsters in the water. The goal of our overarching NSF-funded project is to collect metrics from larvae during acute and chronic temperature treatments. and use these metrics to calculate thermal tolerance range of the larvae. This relationship to larval settlement will then be used to predict larval settlement in the field and anticipate the distribution of juvenile and adult lobsters. This has implications for the fishery industry in the Gulf of Maine, which relies heavily on the monoculture of the American lobster. Research relating to lobster distribution can help maintain the ecological stability of the species and therefore the economic stability in the Gulf of Maine. Understanding how larvae respond to different temperatures will be important for predicting the distribution of lobsters in the future, considering climate change. Distributions have already been shifting as the waters in the Gulf of Maine warm at a particularly accelerated rate. In my project specifically, I looked at acclimation temperature. In other marine species, such as crabs, there is evidence that acclimation temperature, or the temperature at which larvae develop, affects the thermal tolerance of the larvae. However, in the field of lobster larvae research, the impact of acclimation temperature on the thermal tolerance range remains largely unstudied. Differences in thermal tolerance based on acclimation temperature can be used to help understand the mechanisms behind the much broader range of thermal tolerance that we see in wild-caught larvae versus lab-reared larvae, who are usually reared at 18 degrees Celsius. So my research question for my project was, do larvae reared at different temperatures exhibit different thermal tolerance ranges? Here's the workflow of my project. So to start, the Maine Department of Marine Resources collected and then provided us with local egg-bearing females. As soon as the larvae hatched off of the mothers, they entered a rearing temperature. So we chose 18 degrees Celsius, which is the optimal lab rearing temperature used in lobster larvae research in terms of efficiency, and ambient temperature, which follows the seasonal temperature changes seen off the dock where we source the water. So that starts around 13 degrees Celsius and kind of approaches 16 or 17 degrees Celsius. And lastly, we have 14 degrees Celsius, which is the new rearing temperature in question. Larvae were reared individually in jars of filtered seawater and were fed Artemia brine shrimp with their water and food replaced every other day. Between hatching and stage 4, larvae molted through these larval stages pictured here, and we tracked their development with daily checks of mortality in larval stage. Metrics were collected on these stage 4 post-larvae during acute temperature treatments at these various temperatures. Due to constraints, we only collected data on stage 4 post-larvae. Stage 4s were chosen because they make the transition to the bottom, so their thermal tolerance will dictate how many larvae settle and where they will settle. For acute temperature treatments, half of the larvae entered resting trials, while the other half entered active trials, and their oxygen consumption was recorded via microrespirometry. The metric scope for activity was calculated by subtracting the average resting oxygen consumption of several individuals from the average active oxygen consumption of the individuals in the active trials. Scope for activity is used as an indicator of what a larvae's metabolism is undergoing at a particular temperature. And we then collected dry weights to correct each metric for the weight of each individual. And so these are the raw data from the microrespirometry trials at all 3 rearing temperatures. Each data point represents an individual that underwent an acute treatment at a particular temperature, which is indicated on the x-axis. And then on the y-axis is their oxygen consumption. So these graphs can show us the spread of the data. We can also get an indication of where their scope is going to be positive versus negative. For example, we can see here in the 14-degree larvae that entered a treatment at 18 degrees that their resting oxygen consumption tends to be lower than their active oxygen consumption, which indicates that they're functioning normally. Then we see at 4 degrees Celsius that the resting oxygen consumption is actually higher than their active oxygen consumption, which tells us that they were not doing so well during the trial. You can also see there's overlap of data points between individuals who are in an active trial and a resting trial. So lots of variance there. And so these are the compiled results based off of the raw data that we saw before. So here, each data point is the scope for activity for all the larvae who entered an acute treatment at that same temperature. So the points are colored differently based off of rearing temperature, and we fit a polynomial trend line for each rearing temperature to create predicted thermal tolerance curves.
Negative scope means that this was outside of their thermal tolerance range. So we can see that larvae reared at 14 degrees Celsius had a colder, lower thermal tolerance threshold than larvae reared at both 18 degrees Celsius and ambient temperature. Essentially, they could survive at these colder temperatures. There's also some indication that larvae reared at 14 degrees might be slightly more heat tolerant, as we see this greater y-intercept over here. And these are data that were collected last year on wild-caught larvae. So this graph gives you an indication of how much broader the thermal tolerance range is for wild-caught larvae compared to lab-reared larvae at any of the 3 temperatures that we reared them at. So wild-caught larvae experience positive scope at 6 degrees Celsius, whereas the 18 and ambient reared that we looked at had negative scope at that same point. The 14-degree larvae bottom out at a similar temperature to the wild-caught larvae, but they were nowhere near as heat tolerant as the wild-caught larvae, who have positive scope all the way up through 30 degrees Celsius. You can see over here. The first major takeaway is that larval acclimation temperature does have an impact on the thermal tolerance of the larvae. So larvae reared at 14 degrees Celsius were more cold tolerant than those reared at both 18 degrees Celsius and ambient temperature. Further, the 14-degree reared larvae exhibited a thermal tolerance threshold lower end that was similar to that of the wild-caught larvae, but not as near as high of an upper threshold. And this suggests that factors other than rearing temperature account for the broader thermal tolerance range that we see in wild larvae compared to all lab-reared larvae. We can also hypothesize, based on the relationship we saw between acclimation temperature and thermal tolerance range, that as ocean waters warm, lobster larvae will be more likely to be heat tolerant after having developed at these warmer temperatures. These findings are very exciting because the relationship between larval acclimation temperature and the thermal tolerance range could be used to better predict the future distribution of juvenile and adult lobsters in the Gulf of Maine. Support for this project was provided by the NSF grant titled Linking Physiological Thermal Thresholds to the Distribution of Lobster Settlers and Juveniles, as well as Bigelow's NSF grant for undergraduate research experience in the Gulf of Maine and the world ocean. Thank you to our collaborators, Jess and the Maine Department of Marine Resources, as well as University of New England. Thank you to my mentors, Eric and Doug, for their leadership and guidance. Thank you so much to Aubrey, Alexis, and Zai, without whom this project would not have been possible. And lastly, thank you to every kind soul who Alexis and I roped into water changes.
Oh, sure.
Good job. You did so good. So you took the respiration measurements at different temperatures for the 3 treatments. So why was that just a time limitation, or why was the one treatment have so many temperature measurements but not the other ones?
Yeah, so I'd say it's partially due to time constraints, and we looked at lots of temperatures for the 14-degree reared larvae because that was kind of the specific treatment that we were honing in on. And then also for the broader project, we're collecting more data on those reared at 18 degrees Celsius because that's where we have the majority of our larvae being reared. And so kind of just trying to create a curve for this new rearing temperature that we had looked at before is the reason that we had more acute treatment temperatures for the 14-degree larvae. And then, of course, just a few for the ambient. And we tried to target the lower and upper thermal tolerance thresholds to try to get a good picture.
Great talk. I was curious, so since we see an increase in the tolerance at warmer and colder temperatures for the lobster reared at those temperatures, would we expect that future generations have the capacity to continue lowering and increasing their threshold tolerance of temperature?
Yeah, that's a good question. I would anticipate that because we looked at acclimation temperature, that the temperature that the individual was reared at or developed at in the wild as an individual would maybe impact their ability to withstand other temperatures. I'm not sure if there's a genetic link to that, but we are looking at different molecular data in the overarching project. But I would say for acclimation temperature, possibly if they did develop at a lower temperature, then that would then affect their ability to tolerate different temperatures.
Did you source your eggers from a single temperature or vary temperature, and were all of your replicates individual from the same mom or different moms?
So all of the larvae came from a variety of different egg-bearing females. I think we had about 10 over the course of the summer, but they were all sourced from the East Bouquet region, so over in Management Zone D, I believe. So those would be all sourced from the same area. I'm not sure if it would have been the exact same water temperature because we see the increase over the course of the summer. But yeah, different larvae from different egg-bearing females, all from the same region.
Great.
Click on my slide first. Hello, everyone. Thank you for sticking around for this very long day. My name is Molly Spencer. I am a senior at the University of Southern Maine. And this summer, I've been working with my mentors, Dr. David Fields and Maureen Nemisto, in my research concerning the ingestion rate of larval lobster, Homerus americanus, on copepod species in the Gulf of Maine. And so my research is focused primarily on the natural diet and feeding ecology of Homerus americanus, or the American lobster, and factors affecting these dynamics in their early life stages. And so the diagram I have depicted above on this graph is called a trophic cascade. And so this is a diagram that shows the relationship between organisms and how their food dynamics affect one another. And so at the very top of this trophic cascade, we have our primary producers. And in the water column, that's going to be the phytoplankton. And then right below this, we have the primary consumers with the zooplankton and pelagic larvae that will be feeding on the primary producers. And then lastly, we have our secondary consumers, which here I've depicted as the pelagic lobster larvae. And so the early life history of the American lobster is spent in its planktonic form within the pelagic water column in the ocean. And on here, we have the planktonic forms as stage 1, 2, 3, and 4, also known as the post larvae. And to give an idea of what these sizes look like, stage 1 is about the size of a ladybug, all the way to stage 4, which is roughly the size of a grasshopper. And so as climate change continues to warm the Gulf of Maine at an increasingly alarming rate, factors affecting zooplankton assemblage and trophic interactions could prove to be detrimental to pelagic larvae like the American lobster. But although I've depicted the American lobster as feeding directly on the zooplankton and pelagic larvae, research actually has yet to determine exactly what species, size range, or prey concentration that is attractive to these larvae, if at all. And so this gap of knowledge has large implications for what might be an underlying driver in larval transport concerning recruitment into our coastal nurseries in Maine. And so this gap in knowledge is currently being studied within a much larger project ensuing to understand what's known as Maine's Great Disconnect. And so ultimately, the GOM is seeing an abundance of breeding lobsters, but the young of year recruitment is decreasing. And this is often what dictates the young of year that we're seeing settling to the bottom and then being recruited back to our coastal nurseries. And so this is decreasing. And so this disparity suggests that there is a decrease in the survivability throughout larval development in the American lobster. It could possibly be due to their diet within nature. And therefore, clarifying the relationship between the larvae and the pelagic food web is of utmost importance to this developing narrative concerning the survival of larvae within the water column. And so our lab has conducted field surveys last summer and this summer to see what is living within the pelagic water column and coexisting with the lobster larvae. And so on your left, we're seeing these are the zooplankton that are coexisting with lobster larvae. And then within the water column, roughly 25 to 30 animals per liter within the height season of larval lobster, June through August. You see on the right here is what they're coexisting with. And so our data shows that out of these zooplankton species found in the water column, roughly 75% of it is made up of a subclass of zooplankton called Copepods. And so the dominant prey species coexisting in nature with lobster larvae are Oithona, Pseudocalanus, Centropodes, Temera, and then specifically for this study's research, Acarcia. And so my questions for this summer's research was, one, what is the max ingestion rate of early stage lobster larvae on copepod species? Second, do one of the major copepod genuses in the Gulf of Maine, Acarcia, serve as adequate nutrients for larval lobster in the early developmental stages 1 and 2? And thirdly, do early stage lobster larvae display a preference for specific copepods depending on their developmental stage? And so this experiment was looking for the ingestion rate of larval lobster and varying prey densities of copepods. And so what I've displayed here is one of 3 traditional functional response curves. The response curve depicts the intake rate of a consumer, and in this case being the lobster larvae, the rate being the slope of this curve of the ingestion as a function of prey density. And so on the x-axis, we have our prey density. In this case, it's going to be the zooplankton. And so as we go across the x-axis and prey density increases, a predator will eventually hit an encounter rate limitation. And so as prey density increases and the lobster larvae meet this little blip in their ingestion rate, that is when their natural improvement of development in catching their prey is limiting their ingestion rate of the species. But as prey density increases, we expect the ingestion rate of this prey to also be increasing until they hit a max ingestion rate. And so this is where we assume that they're satiated. And so the key objective for our study here was to find the max ingestion rate of the lobster on common pelagic prey to help us understand the larval lobster's feeding threshold in the natural environment, and therefore help us characterize how sparse prey densities in nature must be for larvae to be considered food limited. And so for this study, we obtained our zooplankton prey items via net tows off the coast of West Bay Harbor, Maine, and off the pier of Bigelow Labs. On the right, we have said zooplankton net tow. And so these net tows are catching very small animals, roughly 0.5 to 2 millimeters, or roughly the diameter of a pinpoint needle. And then our lobster larvae were reared here in our saltwater suite in Bigelow Labs. And so the stage of lobster we used in this study, again, were stages 1 and 2. And unfortunately, we were not able to get to stage 3 for our molts within the timeline of this project. And so for the study's ingestion rate experiment, we have 3 prey species that we worked with this summer. And so data was obtained from a previous study done by Sarah Risley on the early stage larval lobster ingestion rate on a species Artemia, or brine shrimp. And so Artemia are a lab-reared species and are very slow moving. And so this was just to get us the baseline ingestion rate of this species to know when stage 1 and stage 2 can be satiated, or at the very least know that they are ingesting. We also use, of course, Acarcia species. They're similar in size to Artemia, but incredibly fast moving. And we also wanted to compare the abundant Acarcia species found to be coexisting with the pelagic larvae with a not so much abundant, but dominant nonetheless species in the Gulf of Maine, Calus venemarcus. And so Calus are a much larger copepod species who in comparison to Acarcia are roughly twice its size, but this is also a very fast-moving organism. And so in this way, we can observe the capabilities of each developmental stage of the larval lobster in terms of what size and capabilities that they have in catching these prey items. And so the prey densities we used in this experiment were Acarcia and the Calanus species, whose individual concentrations were paired in altering volumes, seen here as a 1 liter jar, a 2 liter jar, and a 4 liter jar, with our most dense concentration being 50 copepods per liter to our least dense concentration being 5 copepods per liter. And so to help visualize what these grazing experiments look like, we have the grazing of prey being conducted in a dark incubator where we put the lobster larvae in for 6 hours of grazing. And at the end of each grazing period, experimental chambers were then immediately surveyed for the remaining prey upon retrieval of that lobster to calculate the number of copepods ingested. And so to assess the amount of predation attacks in the experiment, we looked not only for what copepods were missing, but also the state of the copepods that they were in within the vessel, as in were there any mauled copepods still left in that vessel. And so for our results, we have to start with the ingestion of Artemia. And so Artemia, again, were used here as a benchmark to just show the ability of the lobster stages grazing. And so on the x-axis, we have our prey concentration of number of animals per liter. And on the y-axis, we have the number of animals ingested per day. And so the ingestion rate of stage 1 lobster on Artemia show a max ingestion rate is being met. And so we know that they can be satiated. And then similarly in stage 2 lobster larvae, we're also seeing a max ingestion rate. But notice on the y-axis that this is a larger scale. And we see that stage 2 lobster larvae are able to handle much more prey than stage 1 were. Whereas in stage 2, they were able to start off with about 40 prey being eaten per day. Whereas in stage 1, they're eating roughly 20 at the start. And so this data was indicative of how easy it is for these lobster larvae to reach satiation on a slower moving lab-reared prey item. And so next we have our lobster's ingestion rate on a wild-caught Acarcia species. And so as we can see, the amount of ingestion of Acarcia in both stage 1 and stage 2 are grossly below that of Artemia, with stage 1 being able to handle at least 9 copepods per day, and while stage 2 can ingest around 14. It seems as though at stage 2 larval development, they're able to catch and or handle Acarcia better than stage 1's development capabilities. And so this draws a contrast between the Artemia and Acarcia, who, although are the same size, only one of them display evasion tactics, which would be the wild-caught Acarcia. And then finally, we have our ingestion rates on stage 1 and stage 2 larvae on calinus concentrations. Both stages of lobster larvae appear to be able to ingest more calinus Phenomachus individuals than they were Acarcia, with stage 1 larvae ingesting a maximum of 12 prey per day, while stage 2 ingested a maximum about 16 per day. And so this data tells us that despite being twice the size of both Artemia and Acarcia, as well as being a fast-moving prey item, stage 1 and stage 2 larvae are capable of capturing and handling these copepods. And then this is all the data together. And so looking at this data, we can conclude that we did not, in fact, find a max ingestion rate in stage 1 and stage 2 lobster larvae on wild-caught zooplankton species selected in this experiment. It seems that we didn't even breach the start of our ingestion rate curve for these species of copepods. However, introducing higher concentrations of these species to the larvae would not be ecologically representative of what is coexisting with them in the water column. Remember that in the water column currently, about 25 to 30 prey per liter are with the pelagic larvae, and our most dense concentration here, 50 per liter, so twice the amount of concentration that we're seeing in the ocean. They're not eating these wild-caught zooplankton. And so therefore, based on these concentrations in the study, if found in nature, we would expect these lobsters to perhaps be food limited. Additionally, we found that the early stages of development in these larvae were capable of capturing and handling the less abundant species found in the pelagic, the Calinus Phenomachus, more so than the abundant Acarcia species found in the pelagic. And so both species are considered to be fast prey items and display well-equipped predator evasion tactics. However, we are seeing that for some reason, the lobster larvae are ingesting the larger copepod as opposed to the smaller, more slim Acarcia species. And so this could be indicative of a preference from the larvae, where they will spend maybe more energy on the hardier, more filling prey item that they need less of to become satiated. And so what can we draw from these stark contrasts in ingestion rates versus the Artemia and the wild-caught zooplankton? Well, as stated prior, the larvae could be having a unique experience catching these prey items. Artemia are thought to have so little of an escape mechanism that when faced with a predator, they're basically floating donuts. So as the larvae are swimming through their experimental vessel, they're able to pop these little guys in their mouth and then move on to the next one, and therefore their ingestion rate is not being limited. And so therefore, it is expected that the lobster is able to stuff itself with these floating donuts in mass quantities without limiting its rate of ingestion as seen on the data. Acarcia, however, does display fantastic escape abilities, and thus the 6 hours that the lobster larvae were introduced to these prey densities maybe was not enough time to develop a good mechanism to catch these prey items. And so these prey items could have been too zippy for these developmental stages to waste their energy on, and therefore don't display a high rate of ingestion. Similarly, calanists have a well-equipped predator evasion tactic. However, they might have been at a disadvantage for being so big that the lobster larvae found an easier time getting a hold of them. And additionally, the lobster larvae could have also had a unique experience handling these prey items. Whereas again, it's easy to just pop the Artemia in their mouth. The lobster larvae might be spending 2 hours eating that big calanus species because it's so much hardier than the size of the Artemia and the Acarcia. And comparatively, the 2 wild-caught copepod species found may have presented a more robust composition compared to the lab-reared Artemia, and thus presenting a longer handling time. And then finally, the nutritional value presented by each prey item may have been a source for such a large contrast between ingestion rates. Artemia are known for their nutritive deficiency and are usually packed artificially with several nutrients when fed to lobster in aquaculture or research purposes. Thus, the larvae would have to eat tons of these prey items in order to reach satiation, whereas comparatively, wild zooplankton are saturated, and thus larvae could be satiated on just a few prey items, therefore providing reason for such a low ingestion rate. And so what's next? Well, thankfully, Acarcia isn't the only dominant prey item within the water column. So hopefully new research will be looking at a bunch of different prey items that are coexisting with lobster larvae. And so this project hopes to repeat these experiments targeting other dominant prey items coexisting with the lobster larvae to determine their feeding rate and behavior on them. And so I would like to thank those who funded this research, including NOAA and the National Science Foundation. I'd also like to give a huge thank you to my mentor, Dr. David Fields, Warren Mesto, and my advisor, Dr. Rachel Lasley Rasher, which just had her baby, but she's not here today, but for good reason. And so for providing their valuable expertise, guidance, and support throughout my research endeavors. And I will take questions.
Hi, Molly. Great job. So it seems that the zooplankton are very important to the lobsters. Does the zooplankton assemblage in the Gulf of Maine affect the ingestion or behavior of any other animals?
What a great question, Liz. So actually, previous research has been shown that zooplankton assemblage in species composition has known to affect the trophic interactions with several commercially important fish, such as haddock and herring and cod. But these effects have not really been studied for crustaceans, including lobster, which is such a lucrative source in Maine for our economy. So this is why this is such an interesting study to look at, because zooplankton assemblage with the increasing temperatures in the Gulf of Maine, driving these assemblages to different places, might be affecting this transport in this larval lobster, and therefore affecting the harvesting of this lobster, and again, the survivability of those larval stages.
Hey, you did a great job. So something our lobster project was looking at is that wild larvae are actually way more thermally tolerant than lab larvae are. I'm wondering if you guys are doing any experiments of the same kind with this, but using wild larvae, and if there's any evidence that wild larvae have different consumption patterns than larvae reared in lab.
Well, that's super interesting to think about, and hopefully we'll be able to be catching more wild lobster larvae in the future to do those experiments. But right now, the larger project is catching these wild lobsters in the field and using them for diet analysis in like dissecting them and looking at their gut for eDNA. There's also video work being done to see how lab-reared lobster are handling their prey and how they're eating it. So I'm not sure right now if we'll be able to get wild lobster to do these ingestion rate experiments, but that is something to definitely look at a later time if we can get those abundance of larvae.
Yeah, thank you. I know it's really hard to get the wild caught ones.
Really interesting talk. I was wondering, so at the end, you showed all the other zooplankton species that are out there. and that could be important too. And all the, all the other factors that lobsters might be deciding between when they're eating. Can you learn anything from doing a similar experiment with like a mixed natural community of zooplankton? It just kind of seems like it would take a long time to go through every species one at a time. And then when you get to the end of it, you don't really know the answer because you don't know the proportions that they might encounter in the real world.
Yeah, absolutely. So while in this project, since the timeline of our project is so short, we definitely wanted to hit those dominant prey items first, because all the prey items found in the pelagic, a large chunk of that is just that subclass of copepods, but there are like bivalves and barnacle nuclei and abadmi that are also swimming around in there. So right now there's some choice assays going on to try different species altogether with the larval lobster to see if there's a prey selection there, but also in the water column, there is a lot of turbidity, so there are more interactions happening than our still water experiments. Not to mention that a lot of the zooplankton out there aren't necessarily evenly distributed in the water column. A lot of patchiness is occurring as well. So that is something to also think about. But absolutely trying to get those different prey species acting together and to introduce to these larval lobsters is definitely of importance to this project.
Cool, thanks.
Thank you. Thank you for one more question.
Great talk, Molly. I wonder if the different stages prefer different prey. Like, do you have any insight on that? Like, stage 1 lobsters, larvae will prefer like a copepod species from the other, or like from stage 2 will prefer like a bigger one or something like that?
Yeah, so stage 1 larvae and stage 2 larvae seem to prefer the Calanus over the Acarcia species just in our preliminary experiments. We're still trying to figure out why. Some carbon analysis would be really nice to see that, that distinction in nutritional value, to maybe see if they're picking prey items based on their nutrition. For lack of a better word, but definitely looking at different species to introduce to different stages of lobsters of interest to us, just to see if they're hindered by their capabilities at that stage. So maybe stage 1 larvae can't handle anything bigger than a Calanus copepod because they're so small. The Calanus is roughly half the size of a stage 1 lobster larvae. So it was just impressive in and of itself that it was able to even handle that massive of a prey item. But definitely looking more at those choice assays in the future to see if each stage does select on different prey species.
Okay. Hello, everyone. My name is Lega Rocha, and I'm a rising senior at the University of the Pacific in Stockton, California. This summer, I was working in the lab of Dr. David Fields alongside both Dr. Fields and Maura Niemisto. And the title of my presentation today is Feeding Fleas, Ingestion Rates of Marine Cladocerans. And these images on the left-hand side here are marine cladocerans, also known as water fleas. These are individuals that belong to the genus Podon, which is the primary focus of my study. So to start, I just want to introduce you to cladocerans. There are over 600 species that belong to this superorder Cladocera, and that includes both freshwater individuals like the genus Daphnia and marine individuals like the genus Podon. Cladocerans are, though, more overwhelmingly freshwater adapted than marine adapted. In fact, there are only about 8 confirmed cladoceran species that are marine dwelling, and they all belong to this family Podonidae. Despite the obvious lack of diversity in the few marine species as compared to the hundreds of freshwater species, marine cladocerans have been recognized as contributors to the marine food web, both as consumers and as prey. They have been known to be a part of the primary diets of some larval fish species, and they themselves are consumers of some primary producers in phytoplankton, although we're not sure which ones. Sometimes their consumption can rival that of copepods when conditions are favorable. Despite the fact that they play such a role in this food web, we don't know nearly enough about them to keep them alive in the lab. And before I talk about why that's so important, I want to orient you to the animals themselves. So, like I mentioned, these individuals belong to the genus Podon, and they're the focus of my study. They're recognizable by their one large eye spot. You can see the striations of the muscle that they use to control its movement. They also have these two arms that extend outward from their body for swimming. And along the middle of their body is their gut, which you can see in this individual on the right is full by the dark gray-green coloration. And you can also notice that this individual is a female by this large pouch on its back, which is the brood pouch that holds the developing eggs. And it's this pouch, or more specifically, its contents, that makes marine cladocerans so important. So cladocerans can reproduce both via parthenogenesis and through sexual reproduction. When they're in favorable conditions, they're more likely to reproduce through parthenogenesis, where they're making genetically identical clones of themselves that are already suited for a particular environment. So you can see in this individual, there are clones developing in the brood pouch, recognizable again by their distinctive eye spots. So you can count how many are developing in this individual. There are 3. When conditions are not so favorable, though, they'll reproduce through sexual reproduction, and they'll form this one large egg, which is known as a brood or resting egg. That's the result of sexual reproduction, and it's more differentiated. And this egg can remain viable in the sediment for centuries, hopefully hatching when conditions become more favorable. However, it's the production of the genetically identical individuals that can be really useful and important in studies of genetics and toxicology and even evolutionary research. So recreating these favorable conditions to get them to clone themselves is ultimately our goal. But it's a very difficult goal, and there are a lot of aspects to account for in order to achieve it. And my project focuses on one of those aspects. Specifically, what do they eat? What is it that they're consuming out in the wild that is able to sustain them? And what could we provide them for food in the lab that can help keep them alive and keep them cloning? So I started by looking at some background data of the abundance of cladocerans here just off the Bigelow Dock in the Damariscotta River estuary over the past year. So these dots are representative of 2 genera of cladocerans common in this region, Podon and Avodne, and they correspond to the individuals per liter axis on this left-hand side. The gray line shown here is the average water temperature that corresponds to the y-axis on this right-hand side, ranging from 0 to 20 degrees Celsius. And the time is shown along the x-axis, starting in February 2020 to February 2021. And you can see in the middle of the graph around July, August, cladocerans start to increase in abundance with this increase in water temperature. And this was useful information for our experimental design, telling us that they're more abundant and could possibly prefer these temperatures above 14, near 16 degrees, which is the temperature that we use to maintain them for the duration of our experiments. So when coming up with the experimental design for this study, we first tried broad samples of prey and made attempts to maintain the Podon for long time periods, and we realized very quickly that that was not going to work. So we switched gears to shorter time periods and more specified samples of prey. But we still wanted to provide a varied distribution of the prey based on their size and concentrate it enough so that the Podon could be a little bit picky and they could choose the preferred prey to consume. We would then work from there to measure that concentration of prey at 0 and 24 hours, both before and after they were fed to the Podon. So we concentrated our sample by taking 500 liters of whole water just offshore from the Damariscotta River estuary and filtering it through meshes to get prey between 53 and 100 microns. And we took 4 liters of water, again from the same location, filtered through meshes to get prey between 5 and 53 microns in size. And we resuspended all of that prey in 1 liter of filtered seawater. And we put that concentrated prey mixture into 20 milliliter jars. Our first was the jar serving as our initial concentration of prey, which was immediately preserved with 10% ethanol and is represented in blue on the left-hand side. The next was our control, which contained the prey mixture and sat in a 16-degree water bath for 24 hours before being preserved, and is represented in gold in the center. And the last was our experimental jar, containing both the prey mixture and 20 podon, which also sat in the water bath for 24 hours before the podon were filtered out and the sample was preserved. And this sample is represented in dark green on the right-hand side. And this trial was performed in triplicate, and all of those samples were then run through the flow cam, which you may remember hearing about very early in today, that provided us with images and size distributions of the prey in our samples. And this was the data that we were able to observe. So in this graph, you can see area-based diameter in microns along the x-axis increasing as you move from left to right. The y-axis is prey abundance per milliliter, increasing from 0 to 550. The initial concentration of prey, again, are the blue bars, the control concentration are the gold bars, and the concentration that had the 20 podon are the dark green bars. So this data is averaged across all of our replicates, and as you can see, the prey abundance drops in the samples with 20 podons as compared to the controls and or the initials in nearly every size bin. So this shows us that the podon are grazing. They're actually eating something in our samples, which was fantastic news when we first saw that, because when we started this project, we weren't confident we were ever going to get to see them eat anything in our lab. We also took this data and we ran it through the Frost equations to determine ingestion rates of the podon. The Frost equations here, where K is the growth rate of the control from the initial and G is the grazing rate of the podon compared to the initial. In the bottom equation, F is the feeding rate and N is the number of individual podon. And when the results of these two formulas are multiplied together, you get the ingestion rate. So in this graph, along the x-axis again, we have area-based diameter of the prey increasing as you move from left to right. The y-axis is the ingestion of individual prey by each podon per day. And as you can see, they appear to be ingesting the greatest amount of prey in the smaller size ranges, and they're ingesting few, if any, prey in the larger size ranges. In order to get a better idea of what kinds of prey they could be ingesting, we converted our data into biovolume, which hopefully I can get to show you in a little bit. Oh, it's the best. We're building up the dramatics a little bit.
Could you do this in a performance dance? I'll be your next wife. Oh, is it just one? Because I still see it. You're zoomed in.
Mm-hmm.
Do you see it on your screen too?
Yeah. Well, I see the slides.
Your projector's tired. Yeah, our projector overheated and we're not going to be able to use it tonight. So the alternative is for Allegra to finish her presentation.
Yeah, and it's working. Do you mind? Just
Oh, what do I do?
we can set it up.
Do I go over there too, or do I stay here? Okay.
Yeah, and it'll just project.
Okay.
So weirdest thing though.
Never happened. We never had
Oh,
a
that's
presentation
true.
so long.
Uh-huh.
That's true. Everybody can log on. You guys all log on to that? Yeah. Do you guys know the Zoom number? Yeah. Oh, that's great. Amazing.
I know. It's going so well.
Can I sit next to you?
Just let me know when to start.
Are you guys all on?
Oh yeah, so the projector overheated, so everyone in person here can't see anything on the screen, but I'm sure everyone on Zoom already can. So you might be confused as to why there was a lull.
Are you guys all logged on? I'm so glad you say that's great.
Just let me know when to start. Okay, so yeah, just to reorient to where I was talking before.
Somebody's got their audio on.
Okay, so looking again at the ingestion rates. Good. Okay. So yeah, again, reorienting you to this graph of ingestion rate. So this is showing individual prey consumed by the podon per day. We saw a lot of grazing happening on the smaller end of the scale as compared to the larger end. So in order to take a closer look at what the podon could be ingesting as prey, we converted our data into biovolume. And this was the really exciting graph that everyone here was waiting to see. So this graph, the area base diameter again is in microns along the x-axis, increasing as you move from left to right. The y-axis is now the biovolume of the prey based on its abundance per milliliter in cubic microns. Again, and this axis is in millions. So just for reference, the biovolume is calculated using the area base diameter, assuming the individuals are spherical. So you can think of it as the larger size ranges here are like cupcakes, and the smaller size ranges are like cupcakes. The larger size ranges are wedding cakes. So although there may be a lot of individual cupcakes, having just a few wedding cakes is going to make up a lot more in terms of biovolume. So after the 24 hours, you can see that in these smaller size bins, the controls and experimental samples dropped compared to the initials. But as you move towards the right, the controls start to increase compared to the initials, which suggested to us that there may be some background grazing of the larger prey on the smaller prey items. However, we can still see a decrease in biovolume in the samples that had the 20 podon compared to the controls, which tells us that there is a difference between that background grazing of the large prey on the smaller prey and what the podon are eating. So again, we know that the podon are eating specifically somewhere in these size ranges, and that was super exciting to see. So we went a little further and calculated the ingestion rate of the podon based on biovolume this time, rather than just the number of individuals. So this blue data, again, is data that you had just seen of that ingestion of prey, individual prey per day by the podon that corresponds to the y-axis on this left-hand side. The x-axis, again, is area-based diameter, increasing as you move from left to right. And the red data is the ingestion of biovolume per day by the podon, corresponding to the y-axis on the right-hand side. And this axis is in millions. So we can see a lot of consumption of individual prey in these smaller size bins, but it's not making up a lot of the biovolume that's consumed. But in the larger size ranges, particularly this point here, between 60 and 70 micron area-based diameter, we notice a peak in the biovolume consumption by the podon, even though the ingestion of individual prey in that size range doesn't look very significant. So again, you have cupcakes on the smaller side, and you have wedding cakes on the larger side. Eating dozens of cupcakes is not going to get near the amount of biovolume as a handful of wedding cakes. So we want to know specifically what those wedding cakes are. What are the podon consuming that's contributing to the most of their biovolume? And in order to start this process, we did some analysis using the images we collected from the flow cam. So this image is an output from the flow cam showing individuals in the sample within the size range of 60 to 70 microns with an initial concentration of prey mixture. So some of the more abundant individuals within this size range that we were able to identify included Thalassiocyra, which is a very common photosynthetic centric diatom. We also saw Gyrosigma, which is another common diatom found all over the world's oceans. And we even saw a lot of bivalve larvae. So think developing oysters or clams. So we have a mix of both producers in our diatoms and early consumers in our larvae. So it's possible that one of these or some other prey item in this size range could be the preferred prey item for podon and making up most of their consumed biovolume. So looking at all the data collected and the research that we conducted this summer, we were able to make a couple of conclusions. In terms of prey ingestion, podon ingests the highest amounts of individual prey that are under 50 microns in size, and there is no observable grazing taking place on prey larger than 100. In terms of biovolume, most of their consumed biovolume is coming from individuals within the 60 to 70 micron size range. And obviously, there's a lot more that can be done in terms of future research, including doing some more isolated feeding experiments in that kind of 50 to 70 micron prey size range at different concentrations, which would hopefully lead us to specifying the prey of interest in that range. Calculations on this data could also be performed to determine the carbon content of the prey that's being consumed based on biovolume to determine how much of what they're consuming is being reinvested into the podon for the purposes of reproduction. And obviously, there's still so much more beyond this that could be done and more to learn about these organisms and how to keep them alive in a controlled lab environment. But we'd like to think we have a good place to start here. So very quickly, I would like to thank and acknowledge NSF for funding this research. I want to thank Bigelow Laboratory for putting on the REU program again this summer. A special thank you to Dr. Polton, to Laura, and to Mike for helping me learn how to use the flow cam. I want to thank the entire Fields Lab for all of their support, my home university, and especially my mentors, Dr. Zachary Stahlschmidt and Dr. Courtney Lehman, and of course, my family for supporting me moving to the complete opposite side of the country for 10 weeks to do this research. And thank you all for listening despite the very long day and the technical difficulties. If there are any questions, I would be happy to answer them.
Great presentation, Allegra. You know, I'm a Cladostrum fan. I just wanted to know if you were you able to see any of your Cladostrums developing like eggs at all, or is that something that you looked at, or maybe you saw them in the microscope and found out that they were doing that?
Yeah, so we looked when we collected them under the microscope, so we were able to see if they had any developing eggs, either as clones or as the brood resting eggs. But over the 24-hour period that we had them inside their water bath, we really were just looking for mortality. We weren't able to observe any differences in development of eggs.
David? So what do you think the primary consumers out here in the right off the dock of Cladocerans, who do you think are eating those?
So it could be a pretty wide range of consumers. I know that there's along the eastern coast that there's fish larvae that are primarily consumers of these marine cladostridans, but really anything significantly larger like fish or other larger zooplankton could be consuming. And even Molly just was talking about that lobster larvae could also be consuming some of these cladostridans out in the bay.
And what do you think they use their eye for?
Well, I doubt they would be able to see actual images, but it could just be a light dark sort of sensitivity so they could determine their movement up and down the water column.
So when they go through parthenogenesis and create smaller versions of them, cloned versions of themselves, what is the size difference between the large and the small ones?
So the larger ones, based on just the observations that I was able to see this summer, they're around 600 microns in size. And the smaller ones, I mean, I haven't been able to actually see them hatch or develop from the brood pouch, but I would imagine based just on observation, visually looking at them under the microscope, they would be about maybe 200 microns in size.
Because my question was sort of, do you think that they would eat different things depending on if they had just come out of the brood pouch versus if they were a fully formed larger one? And how would you go about studying that?
Yeah, that's definitely very possible. In order to study that, I would think we would need to do a little more research to be able to keep them alive in the lab so that we could get them to produce those clones and then kind of recreate these types of feeding experiments to observe that behaviors.
You made a comment at the beginning of your presentation that blew my mind, that the eggs that resulted from sexual reproduction can last in sediments for a crazy long time. How do they not get predated in the sediment? Or do we know how they last that long?
Yeah, so I'm not entirely sure. I haven't looked into too much of the specifics of the brood resting eggs, but I imagine just settling underneath the sediment would offer them some protection, and then just the sheer amount that they would be able to produce can get them to last at least until they're in more favorable conditions to get a good amount to hatch and kind of repopulate the species.
It is crazy. The oldest hatched egg was 400 years old. And they got it from the core. They aged it in stratigraphy and then they hatched it. Mm-hmm. Yeah, crazy. I'm not sure how many eggs they hatched. There was a science paper on it. It was really, it was amazing. So.
Great presentation, great talk. Correct me if I'm wrong. You said that marine cladostridans they've not been extensively studied because they're difficult to keep in labs. Do you have any idea how easy it is to keep the freshwater cladostridans? Why was it that they could keep them alive in lab or get those experiments done on freshwater cladostridans? Do you have any idea?
Right. So that is kind of one of the big mysteries, because freshwater cladostridans like Daphnia have been able to be maintained in the lab for decades. And for whatever reason, we haven't been able to figure out why marine cladostridans are so difficult to keep alive in the lab. It is suspected that cladostridans, just as a superorder, they're just not very well adapted to living in marine environments. That's kind of why there's that discrepancy between the 600 plus freshwater species compared to the 8 marine species. So whatever it is, wherever that kind of marine species deviated from the cladostridin lineage, it just hasn't, I guess, it might not have developed to the point where they're easier to maintain, if that answers your question.
Yeah, thank you.
Woohoo!